हाँ भाई हितेन, क्या चल रहा है? हाँ हितेन।
हेलो।
हेलो हितेन, क्या चल रहा है?
मेरा आवाज आ रहा है क्या आपको?
हाँ, आ रहा है, आ रहा है।
ओके। अ, एक मिनट, मैं एक बार स्क्रीन शेयर करके देखता हूँ।
हम्म।
ओके, आपका स्क्रीन ऑलरेडी शेयर है एक्चुअली।
नहीं, वो बाद में आपको कर देंगे, कुछ नहीं होता, प्रॉब्लम नहीं हो रहा है। सबका ही आराम से हो रहा है।
ओके, ठीक है। आवाज तो आ रहा है ना, क्योंकि मेरे लैपटॉप में कुछ इशू है तो मैं कंप्यूटर से कनेक्ट किया हूँ फिर।
हम्म।
ओके, आप बताइए, सब अच्छा तो चल रहा है?
हाँ बस यार, फिजिकल मीटिंग हो जाता तो अच्छा रहता।
हाँ, वो तो तब अच्छा होता, बट फिर भी अच्छा ही हो रहा है।
तो बहुत अच्छा हो रहा है मीटिंग।
हाँ।
नेक्स्ट नेक्स्ट देखते हैं, विंटर। इस साल एंडिंग में देखते हैं, कुछ कराएंगे।
हाँ, मे बी ठीक होगा, तब वेदर भी ठीक होगा एंडिंग टाइम में। सर का एक्चुअली एक बुक अभी भी रह गया है।
हैं?
हमने सर के लिए एक बुक बनाया था ना उनके बर्थडे पे।
हाँ, वो छपा देते हैं यार, वो अमेजोन में छप जाता है।
नहीं, वो अभी भी हमने किया नहीं है फाइनल, मतलब प्रिंटिंग।
नहीं, वही मैं बोल रहा हूँ। बहुत सारा पिन सेट बोल के एक कंपनी है, उसको
हाँ।
पीडीएफ दे दो तो वो बुक बना के दे देता है।
अच्छा। ओके। ठीक है।
है क्या? वो वाला काम कुछ मतलब वो दयाकर आ रहा है ना, रेगुलर? दयाकर।
कहाँ, सीसीएमबी?
हाँ, वो कमल के साथ कुछ कर रहा है ना।
राइट राइट।
तो
वो
तो उसका
आई।
मोस्ट प्रोबेबली एक डेढ़ महीने में हो जाएगा, तो उसके बाद
ओके।
हम पेपर भेज देंगे।
ओके, ठीक है।
स्टोरी तो अच्छा ही बन रहा है।
ओके। सही है। आप घर से कनेक्टेड हैं ऑफिस में?
हैं?
घर से कनेक्टेड हैं ऑफिस में?
नहीं, अभी तो घर पे हूँ।
सेकेंड हाफ में भी ऑफिस से करेंगे?
हैं?
सेकेंड हाफ में ऑफिस से करेंगे क्या?
हाँ हाँ, वही मैं सोच।
कैसा है फंडिंग अभी आपके पास?
खास नहीं है यार, वो प्रोजेक्ट का पैसा भेज ही नहीं रहे डीबीटी वाले।
आएगा, मैं भी डिले है लेकिन आएगा फिर।
कब आएगा पता नहीं। और सीएसआर तो, बाप रे बाप, सीएसआर का एक प्रोजेक्ट है, अभी तक नहीं दिया।
ये सर यार, हमने लास्ट मोमेंट में अभी आया हमको दिसंबर में, आपको कैसे दिया नहीं उन्होंने?
अभी मीटिंग के लिए कुछ पैसा बचेगा क्या, दिसंबर नवंबर दिसंबर में?
हो जाएगा, अरेंज हो जाएगा।
Good morning, Shubhodev. You are muted. You are muted.
Myself, my photo is visible.
Your photo is visible, but you possibly is logging through mobile, right?
Yeah.
Yeah, but I can see you very well. So the meeting is going very nice.
Yeah, very nice, very informative, and so much of nice discussions are happening. It's,
So
other meetings we don't see that very formal type. So this is more like kind of a group, like people are expressing different thoughts. Even all the talks were very good, and particularly like the discussion session. So many are participating.
that is true. So actually, I have logged through two different computers. Sometimes,
Yeah. When there is a conflict, then it creates a problem.
but no, I have kept one computer totally sound off, so there will be no conflict. But
Achha.
times, it gives a trouble at NIPGR, this Wi-Fi. So therefore, it's handy. Yeah.
So since today is Saturday, I thought first half I will do it from my home, then go to lab late, like around during the lunchtime or so.
That's fine.
Because nowadays, because of the still I am feeling a lot of weakness, so not able to handle too much of uh, this discussion, like feeling very tired.
But still, you could make this kind of a meeting after getting corona and all. That's really very common.
So mine was not that severe, but still it is not going off 100%. Like although it's okay, but still sometimes pain, other things are
Good morning, Devi.
Good
Yes,
morning.
Sunil. Good morning, Subhadeep.
Good morning.
Yeah.
Good morning. Good morning.
Good morning, Ashita.
Hello, Ashish.
Good morning. Good morning.
Shubhodip making us all gathered together
Yeah.
early in the morning on Saturday. Only thing there is no tea, Shubhodip.
Yeah, that's the problem. Actually, we thought of yesterday, we were discussing in CDFD that since we arranged, we thought of having a physical meeting and got good arrangements also that we had to cancel. So we
That
were
you
even
told.
thinking of tying up with Swiggy and deliver the food and beverage to the respected
No,
speakers.
that will be too difficult because
Yeah, that's what logistics we thought of that also.
Shivaji, someplace you will have only few people and then
Few people,
all
that's
this,
true.
city and everything. That's too much. We don't expect that.
So actually, we thought of that also. Zomato or Swiggy, some food like this thing, and also for some refreshment. So then me and Tangaraj also was having that idea. Then we thought that logistics will be too difficult.
It's too difficult.
Anyway, it is becoming normal now, looks like. So I think maybe in
Yeah,
another.
maybe next year it will be very fine to that physical meeting.
Good morning, Praveen.
Good morning. Good morning.
Good morning.
Good morning. So whether we are going to miss, that you are talking about the food and other things. No, we will have someday.
Yeah, that's what I was talking to Sudha and Prasunda that.
And not only that, right from the MIPAM, when we started long back, so we do have a local excursion also. All the time it is linked. So that is, that also we are missing.
In Hyderabad, actually, we planned for a trip in Nagarjuna Sagar, that Buddha, that extra, there is a big, when they were digging the Nagarjuna Sagar, they got a big, that monastery kind of thing. And that they preserved in an island in the Nagarjuna Sagar, which is around 150 kilometers.
So I think 19 people have joined. So can we wait for 1 or 2 minutes more?
Yeah, 1 or 2 minutes.
I think it's Saturday morning, so maybe people are reluctant to.
Then what will happen for tomorrow? The Subhadeep has kept whole day tomorrow without any break.
No, but that is a student
thing,
so they will attend. No, you will see.
Actually, it's so many students we can't.
वो स्टूडेंट बहुत ज्यादा है ना तो वो अटेंड करेंगे वो देखते रहे.
पोस्टर सेशन हमने we thought.
Hi Hitendra. Nice to see you.
Good morning.
And yesterday, I could see the photograph of Devanand. So that I really loved that photograph. So and I was looking at one of my Facebook or earlier Facebook the things there. I think 2020 Hitendra has given a poster for MIPAM.
Right.
And because of that COVID, क्या करें सब पागल हो गए.
Okay.
But you definitely you look like Devanand. इसमें कोई शक नहीं है.
Hitendra even booked almost booked all the rooms like in CDFD.
Yeah. And this time also I booked my flight tickets and then I cancelled. So although it was flexible, so I could only lose around 600 rupees. It's all right.
Subhendra also have joined, looks like, but he didn't turn on the camera. Hi, look. Good morning.
प्रोफेसर साहब आप आजकल कुछ डिफरेंट दिखाई दे रहे हैं. You are looking much more smarter and बहुत बढ़िया दिख रहे हैं.
Your kind wishes.
आप बहुत बढ़िया दिखाई दे रहे हैं आजकल. But we are MIPAM जो है वो तेजपुर में ही मजा आता है, because that is नहीं आशीष.
Yeah, we had a really great time.
Even Amarkantak was very good, because Amarkantak was very confined area, so we couldn't do much after sunset. So we all enjoyed that time.
It was 100% pure air.
Yeah. It was very nice. There I remember Subhendra's night ka pravachan for 3-4 hours in guest house.
We will be having, I think, again, it will be actually good fortune to having you all again. I think I'll also request to Isan to come and help.
Yeah.
Because Isan is great.
He is not visible this time.
He told us on that day, that train
He is actually head of the department of
Cotton University. He has moved to Cotton University.
So he is very busy there.
Cotton Institute.
Cotton University. Now it has become university.
I see. The
in
Guwahati.
Guwahati.
Oh, so he's there. He's a head of the department there.
Yeah.
He is very busy.
I see. Okay.
Subra madam has joined.
Yeah, I think we should start.
We can start.
Subra, you are ready?
Yeah.
Your name looks like Sriya. No.
These people are doing, I don't from yesterday I'm changing. It's actually controlled by CDFD.
Okay.
Oh, CDFD made you Sriya.
Why should I make me Sriya?
Okay, I thought it was your
Happy
daughter's
with
name.
my own name.
I thought it was your daughter's name.
No.
Because
just a moment. Let me try renaming. Yesterday also I did it, but it again went out.
No problem, man. We all know you.
Yeah.
So
now, see,
now it's okay, right?
Right.
Yeah.
Okay.
So welcome all of you for this new session of MIPAM. And Subra need not have to give any introduction. Everybody know her. Subra is right now director of National Institute of Plant Genome Research. So Subra, can you start?
Yeah. So just a moment. Let me try sharing the screen.
Yeah.
So can you see my screen?
Yeah. It's not full screen yet.
Yeah, I'm doing that. It's okay.
Great.
Yeah. Okay. So good morning, everyone. So we had a very vibrant last 2 days meeting, and today is the third day. So today I'm going to talk to you about something which is related to the fungal pathogenicity, and the interest of my group is to understand the multi-host resistance against fungal pathogens. So with that, my talk is divided into a little bit of an introduction, why we are interested. Then I'll take you through two of the model. Since I'm talking about multi-host, one is in the animal, how the fungus is infecting, and the same fungus, how it is infecting, and what is the immune pathway in plant. So little bit of a comparison. So 3-part, my talk would be divided. So as you see from this data, although this data is little older, this ProMed is not actually giving the data beyond 2014. But as we all know that because of this impending change in the global climate, coupled with increased frequency of very high complexity disease, not only for the human, but also for the plant, that has resulted in challenge related to food and food security and also human health. Now, as we could see that the plant infecting fungi is also, just a moment, sorry. Where is this? Okay. Yeah. So as we could see that the plant infecting fungi is also increasing, same as the animal. But if we look at the Indian scenario, although we have some of the means fungal infection in human is also there, but there are a lot of disease of this fungal infection in the plant. So now, having said so, if we try to see what is the fungal disease, why it's important and worldwide emergence, that is in the next slide. We know that there are almost 1.5 million species of fungi which are inhabiting this planet. However, out of those, only 10,000 fungal species are known to infect either plant or animal or human. Now, in that case, if we see crop destroying fungi, in case of the plant, it causes perennial yield loss almost 20% worldwide, with in addition to the 10% loss in post-harvest. So fungal disease cause more than 2/3 of all crop loss worldwide. A paper came by Caroline Dean from John Innes way back, and she said that there are 5 topmost fungal pathogens which are major impediment, as you could see from this slide, and Fusarium, which I will be talking today, is one of those. Now, looking into the scenario in case of the animal also, fungal diseases are having their moment in also wildlife. 2 cases that I'm showing here. One is a citrate fungus, which has led to extinction of more than 200 species of frog that we have seen. At the same time, we also have heard about nose syndrome, which is because of the white fungal growth, and that is also been spreading very fast in North America, which is also transmitted by bat. Now, when we come about the human, so global mortality, this paper also came a few years back and from Royal Society, which says that global mortality due to fungal infection will be exceeding either malaria or breast cancer, even equivalent to HIV and TB in coming years for human. So in that case, when we look into this, that means fungal species can infect across kingdom from the plant to animal to human. But there are very interesting study came first time from Brandstad's group who said that almost in 2001 that there are several fungal pathogens, and including Fusarium, they have the ability to infect not only one kingdom, but across kingdom. So now the concern, why we are so concerned, because what we see, all these fungal species are basically controlled by fungicide. Now these fungicides are coming from either we talk about the, the human control of the medicine that we are taking, or in the agriculture field, it is only one chemical, and that is azole. And all these things that we have, that comes from azole. And this antifungal chemical has been actually used like nystatin or Pauline, and they were discovered in almost in 1950s. However, for the human, but for plant, we know that azoles have been used as a copper and sulfur fungicides, and they were first used long back, almost if you look into the literature, it will go back to the 150 years ago. And therefore, what is happening because of this antimicrobial resistance that is coming, it is because we do not have a source where we have tapped till so far, which is not a derivative of azole. So that means that there is a parallel driver of fungicide resistance that we are seeing, and we know about AMR very much in the field as well as in the clinic. So now, therefore, how human and plant health is connected. If we see in the animal disease, or what we see, basically we see 20% production of animal husbandry is very much threatened by these global diseases, as well as it is for the 30 to 40 years that resulted in ecosystem loss. And ecosystem here means mostly the agricultural ecosystem. And because of the wild land use, demography, antimicrobial resistance, many other reasons. Similarly, in human health, we see 60% of all human disease have their origin from animal. And that connects the whole human health, animal health, and ecosystem, which we now say it is inextricably linked. And that there comes a concept of One Health that we all know. And this One Health concept came 10 years back, which is a paradigm shift to understand the interactions between plant, animal, human, and their environment. And in such case, what we are going to talk today is a fungal pathogen with respect to the fungal pathogen. And in that case, all life forms are connected. Understanding the interaction between them, therefore, becomes very critical to protect not only human life, at the same time, the agricultural productivity. I'm going to talk to you today of one such disease, which is fusarium disease, and it is a host-specific immunity. Now, as you could see, this small fusarium, which is Ascomycetes fungus, it came almost 50 million years before on this planet. However, we have the chickpea, which we are using as a model to understand this whole biology, that came almost many more million years before than fusarium, so as the worm, that is the C. elegans, that we have taken as understand the animal or human immunity, because you know that C. elegans immunity is very same, innate immunity is very same as of human, and also the innate immune pathway is very conserved amongst the C. elegans, Drosophila, even Arabidopsis. A very nice paper by Jeff Dangle and Jonathan Jones way back in 2006 in Nature has shown that pathway. So this particular fungus also can infect the animals as well as human. So the question came, how this multi-host response occurs. So in order to understand that, what we have tried to see how this fungus infect basically the plant. It actually makes a conidiospore that traverse through the root, and so it's a root-borne pathogen, and through the collar region. When it goes, it actually goes to the vasculature, and in this case, it is not like phloem through phloem, but it is through the xylem vessel, and it makes a vascular necrosis. And then due to that, there is a water transpiration pool that gets affected, and therefore the plant droops down. And therefore we see at the ultimate effect is the wilting effect. Therefore we call it as a vascular wilt disease as well. Now, what happens then if we try to see in case of animal, what we did, we used different Fusarium oxysporum pathotypes, because this is a host-specific resistance. So the fungus have different former species like Siceri, which infect the chickpea. We have Mathioli, which infect the Arabidopsis. We also have Lycopersicki, which infect tomato. In literature, when we started developing this animal model system, we found Lycopersicki have been shown also can infect human. So what we did, we tried to see out of all these species which have been known, which one can be a natural, food for the C. elegans. So as you could see from here, very surprisingly, but also very interestingly, we found that this Siceri becomes the most devastating pathogen, and it kills the worm once you grow them onto the fusarium media. Here the fusarium is giving the carbon and nitrogen source, unlike to the normal food, which is OP50, that is the E. coli. So now what happens when it ingests it? What we found that if you see it is after 24 hours, when you infect it, what happens? But then after 96 hours, you see the total worm is completely dead. Now, there is a very interesting observation that we found in this case is that, which should be noted, that in this case also, the fungus is traversing through the xylem vessel, which is actually the route by which the plant takes its nutrient and water. At the same time, when the animal is ingesting this fusarium, it actually goes to the intestine, and it dissolute the intestine completely. So in that case, there is a parallel between the animal and the plant, which is that the whole root of the invasion is to the kind of a place through which either the animal or the plant takes the food. For many years, our group, in collaboration with Dr. Chakrabarty's group at NIPGR, have interest in order to understand how organelles can actually give the regulation of any kind of cellular processes. So today I will be talking to you about one such organelle, that is ECM, while I will be speaking in the plant per se. So these are many papers that we have over the years published. So what we did, we took up a very, very detailed study, starting from the transcriptome to proteome to phosphoproteome to metabolome. And we took ECM and the nucleus, because ECM, where the fungus is perceived, nucleus, which actually controls all the activity in the cell. And using this, we then tried to make the integrative data. And from there, we tried to identify the hub proteins, and we go for functional analysis. I'll be showing one such gene function or the protein function for worm and one for the plant. So in that case, what happens when we were doing this thing from the NGS data, what we found that if you do the whole NGS data through the RNA-seq in case of the worm over the temporal kinetics, then we find there are almost 1312 dysregulated transcripts, which represent almost 1024 genes, which becomes deregulated. Now, if you try to pull out our interest was the transcriptional regulation. Therefore, we tried to pull out which are the topmost 5 class of transcription factors which gets enriched. And in that case, what we found in the worm, it was homeodomain, ZF and hormones. So these receptor family, which actually were the 3 topmost class of transcription factor which got enriched. Now, when we look the same thing in the plant, in that case, what we found that we had earlier a microarray data, which was published earlier, where we had 1200 deregulated genes, and we call them IRF, immune responsive factors, and there were 75 fungal genes as well. Very recently, we have done an RNA-seq analysis also, which showed almost 3,554 genes gets deregulated. We tried to develop the enrichment of the transcription factor. Very surprisingly, what we see, the enrichment in the plant is not any of these 3 classes which we see in the animal, but the topmost 5 class belong to BHLH, ARF, MIB, Werke, and H disease. So this gives us some clue that maybe the whole pathway of the transcriptional regulation of this particular disease might be very different. Some parallels will be there. However, there will be a divergence in the transcriptional regulation. So now I will be talking first to what we found in the worm, and then last part I will be concentrating in the plant. So what happened? We took the whole data, and what we made a kind of total disease responsive gene networks in the worm. As you could see, when we built this big network, we found there are totally 5 different clusters. One is related to cell fate and disease progression. Another is the development regulator, immune response, deregulated molecular machine and organ patterning, and lipid metabolism, stress signal, and the homeostasis and co-signal regulatory. I have actually shown here some of the genes which I'll be talking later, which we think are very, very important thing based on their interaction. So these proteins became the most interactive hub proteins like CLEC2, NHR6, which are the transcription factors. In the lipid metabolism, we have as AH1, LIPS5, CLEC3. Here we have TKR3, ILIS5, serpentine receptor. Now, in this, we have almost 245 genes came into it as nodes, and there are several ages. we tried to then pull out that in the worm, which are the organs which gets mostly affected. What we found that it is a neuron and it is the intestine, they became mostly regulated. And very interestingly, what we observed that although we had a forked family transcription factor, which is DAF16, but if you look to the network in the neuron, there are 227 genes comes in this network, of which 37 are found to be the target of DAP16 and 32 found to be the target of SAR2.1, which is a histone deacetylase. Now, in the intestine also, what we find out of 139, 21 are DAP16 regulated, 32 regulated. So very interestingly, what we found, although the DAF16 and SAR2.1 by themselves are not showing any kind of differential expression, however, their regulated genes are very much deregulated. That gives us a clue that possibly DAF16 might be the major regulator which is regulating this pathway. With that, if we try to see, DAF16 have been associated with many of the other biological pathway or cellular processes in the worm. For example, it has been shown that it is regulating the transgenerational epigenetic inheritance. It is also known to have the effect on the cellular plasticity. We know that it helps in the protein homeostasis network in the worm. And then it is also of late have been shown that it is involved in the stress signaling, and very two new data that has come after we published our data that came in that showed that DAF16 also is actually regulating the innate immune pathway. Now, in this case, the pathogen is the Pseudomonas syringae in one of the cases. So then we tried to look into, we tried to see that if we could see that how these networks are having the subnetworks. In that case, we tried to figure out the DAF16 regulated, as you could see over here, where you find these three genes are becoming the most interactive hub protein. And at the same time, the SAR2.1 regulated network at the bottom, where we find that serpentine receptor and transferrin receptors are the major interactive hub proteins. So this you could see from the right hand side all the other genes. So if we don't go here, then what we can say is that this RNA-seq analysis, can someone please mute their mic? Okay. So the RNA-seq analysis further then identified the shared and the unique signaling pathways. Those are regulated by DAF16, which is FOXO, and SAR2.1, and which we can say that possibly linking the neuronal stress, which facilitates the negative regulation of intestinal innate immunity. That was our hypothesis at that time. So we went ahead to prove that hypothesis. And in that case, what we did, because when we work in the plant, we don't have this, luxury and this kind of a thing, but working in worm, we understood it's so easy to do genetics, and because of RNAi and because of getting all the mutants available. So in that case, the question was, how then DAF16 influence? Does DAF16 influence the other immune pathways during this fusioresis? So if you little bit know about the CA ligands immunity, there are 5 individual immune pathway which are controlled by DAF16. One pathway by TOL or TRF. One goes through the PMK and SERC or JNK. Other is SMA. And you also have the pathway which is by TGF-beta. So what we did, we actually took the DAF16 RNAi and we made all these under a double mutant study. And I'm not going to detail each of the study. I'm telling you what conclusion we got. What we saw that when you fed the worm with this DAF16 RNAi, that significantly die earlier than the normal worm. And when we actually do all these double mutational analysis, what we found, that ultimate conclusion was that this insulin IGF-1 pathway is majorly responsive to the fusarium infection, while DBL1, TGF-beta, TOL, and ERK1 are involved in behavioral response through pathogen avoidance. So that was a very nice understanding that we understood from this whole genetic study. Now, after that, then we tried to focus more on those kind of neurons. So when I showed you the data, you saw there are SRD, there are GCY5, GCY4, those were the neuronal genes. And if you go through the literature, back to the literature, in case of the worm, which is known that there are many neurons in the worm, more than 300 neurons. Out of those, there are two different kind of function, which is AC and ASI. So if you see the type of genes that we are getting, those all belong to either AC or ASI neurons. So now we then wanted to go ahead with a genetic screen by doing the double mutant analysis. And we found in our transcriptional data that all these genes are target of either DAF16 or SAR2.1. And therefore, we tried to set out a detailed screen, genetic screen, using double mutant analysis. And in that case, what we found that this AC and ASI neurons possibly is regulating this DAF16 mediated immunity. And in this case, I would be right now showing you that DAF16, which of the DAF16 is important. So as you could see from this whole study, what we understood that the DAF16 exerts its function in the signaling cascade. We examined the transgenic worms expressing the DAF16 GFP in this case, and as a reporter, and found and tried to see in the next slide that where it gets localized. As I said, that we showed that non-neuronal DAF16 regulate avoidance and immune response in C. elegans. And to prove that, what we did next, we took VP303 with a DAF16 background and with its own background. And what it shows that we found that VP303 worms, when fed on DAF16 RNAi, that dies significantly earlier, as you could see from the killing assay, than the control worm. Thus, we show that non-neuronal DAF16 regulate the avoidance and immune response in the worm. Now, the question was that, in worm, you would see there are total 6 ortholog of DAF16. And these DAF16s are located in different organs of the worm. So the question next was that which of these orthologs basically are involved in fusarium disease. So in order to understand that, we actually set out another experiment. And in this case, we took the DAF16 GFP from our data. We had earlier what we found that it is possibly this DAF16 is a non-neuronal DAF16, and it is possibly the intestinal DAF16 which is working here. And it is not unlikely because, as I showed you in my first slide, that the worm is going through the total gut system of the pathogen, and therefore intestine, and it's through the intestine, and therefore it is activating the intestinal DAF16. Now, what happens when you have a DAF16 GFP, in case of the control, you could see that this DAF16 GFP is there in the whole of the intestine in the cytosol. The moment you challenge it with the fusarium and it engulfs the fusarium, this DAF16 goes and gets localized in the nucleus. So therefore, we prove that nuclear localization of the intestinal DAF16 is very important in order to evoke the intestinal immunity through their downstream target genes, which are the AC and ASI genes, particularly the SRD, GCY5, and also SAR2.1 regulated genes, that is GCY5, GCY4 as well. So when we say that, in that case, then it conclusively proves that during the initial phase, what we find that AC works upstream to the intestinal DAF16, and later what happens is the nuclear localization of DAF16. Then we see the SRD1 in ASI primes the invasion. And this study then indicates the crosstalk of insulin IGF-1 signaling pathway and neuronal stress in the response that facilitate the negative regulation of the intestinal immunity in the pathostress form. I might point out here that in case of fusarium, it is intestinal immunity. But if you see in other fungal species like Cryptococcus, it is not the intestinal immunity. There also DAF16 is involved, but there it is the epidermal DAF16 that evokes the whole pathway. So therefore, if I try to conclude our work in case of the worm, what we actually understood is that that in this case, when there is fusarium, it downregulates the ASC genes, which are GCY5 and GCY4, and it also downregulates ASI, SRD1. This pathway goes to the attraction and to the death. However, in this pathway, we don't know this missing link, which we are trying to understand now, but the non-neuronal nuclear intestinal DAF16 goes in the nucleus, and that evokes the immunity through the downstream genes, which are IRIS5 and CLEC193 from our data. And parallelly, we have a SAR2.1, and that SAR2.1 actually is regulating, which we have also proved by genetic screen through the UPR pathway and HSP pathway. And this actually is also linked to this DAF16 pathway. We don't know how SAR2.1 actually regulates DAF16. That is what we are right now trying to understand by many of the other mutant study and many of the study, because SAR2.1 is a histone deacetylase. So whether any kind of acetylation, deacetylation is controlling this DAF16, that is where we are right now focusing on. However, this particular pathway also has an effect on the lipid metabolism through NHR17, which is another transcription factor. And when we have this kind of immunity, we see the longevity, but through a PCD. So we are trying to connect also the PCD with the longevity and also the whole PTM, how the DAF16 PTM is regulating the DAF16 activity may not be only the phosphorylation, but also in this case, what we find from our data, possibly the acetylation. So therefore, if I try to conclude what we understood in case of the animal immunity against this multi-host pathogen, which is the fusarium, we possibly could show for the first time that how functional interplay of neuronal genes with the intestinal DAF16 regulate the component of innate immune system of the fungal pathogenesis, and potential mechanism include the dysregulated expression of neuronal gene, translocation and combinatorial expression of DAF16 and SAR2.1 regulated genes, and perturbation of the upstream and downstream signaling cascade. So finally, we could detect many of the C. elegans orthologs in the human genes in various innate immune pathway. For example, we could found the ASP12, which is actually the ortholog of NAPSA in the human. We also have something else which is involved in the Williams-Buron syndrome, which is a neuronal syndrome in the human, and that is here the ortholog is, I haven't yet marked it because right now we are working on that, R08F11.4, and also we have another one which is a pro-oncogene in the human, that is FES. The ortholog, what we find in our data is C18H7.4. So these orthologs then might possess a translational potential to understand fusiosis in human. Actually, we are trying to see some if we could get some kind of collaboration with the medical group, and that is where we are heading. So two things: one is to expand this to a medical group in a human fusarium infection, and other thing is to understand more mechanistic way of regulation through this intestinal DAF16 and how PTM can regulate this whole pathway. So with that, I would now switch over to the plant. Let's see what happens in the plant in this kind of a disease. So as we all know, chickpea is a legume crop, and this is the third most important legume. And as you could see, India is the largest producer of chickpea, but we always import chickpea still because we cannot have whatever we require, we cannot have it. So why this happens? So if we see, one of the major cause of this chickpea yield loss is coming from, again, the fusarium wilt, and that affect not only India, but globally. So this is a global disease across world, and that is the first most major impediment of chickpea production in the agricultural field, and 10% of worldwide crop loss happens every year due to this Fusarium oxysporum. Now it is a soil-borne hemibiotrophic ascomycetic fungus, and if you see, Fusarium ranks almost fifth among all phytopathogen. That limits the growth of more than 100 plant species. So what we started long back, in that case, my students, they took many of the chickpea varieties which were known from the genetic study and also from some of the studies which came from other data. And we took out these all 9 to 10 chickpea cultivars and we subjected them into the fusarium and we tried to see which one is the most resistant and which one is the most susceptible. What we found WR315 is the most resistant genotype, whereas JJ62, which normally in India we use as a Czech variety, that is the most susceptible genotype. So we set out a screen in the lab based on test tube. However, I must tell you there is a old paper by Haware and Nene from ICRISAT, very, very old paper in 1960s. And they actually set up this whole screening method, which we actually adopted. And in that case, whatever this method is, if you take a 10 to the power 6 spores per ml and you see exact phenomena, what you see either in the field versus greenhouse versus lab. So what you find that when you see this kind of infection, WR315, the plant actually can even flower and seed, even if there is a infection in the field. But in case of the JJ62, almost by 15 days, the plant is dead. Now, definitely we tried to see the fungal load, and as you could see, fungal load is much more in case of the susceptible genotype. So this vascular wilt is a very, very complex disease, and it is governed by host-specific resistance, which the genetic study always told. So therefore, we wanted to investigate the genotype-specific cellular response during this host-specific resistance in case of the plant. And our screening, as I told you, that also said that although the WR315 and JJ62 are two extreme conditions, but we have in between cases where K850 and ISOCV2, they showed a moderate response. So these are linked to the R genes which are there in case of the chickpea. We have another work which is very mature right now, but I'm not going to speak. Maybe in the next MIPAM, I would be focusing on that we have actually cloned the R gene. So now here I'm more focusing on the transcriptional regulation, so I'm not touching upon that. So now, what we did, based on the earlier data that I've showed you, the microarray, we then set out this whole network analysis. In this case, what we found from this modular network, we call this left-hand side network as a diseasome because it is developed from the JJ62 data, and the right-hand side is the immunome, which we have developed from the WR data. So in this case, as we could see, that you can actually group them into three major groups. And in that case, one group have all the genes which are known to have colonization function. The other group is having known genes which have disease development function, and the other group has penetration related function. And there were three genes which are perception related. So very nicely, it shows that in JJ62, our data shows that how all this disease is happening. But very interestingly, we see a complete change in these hub proteins. And in that case, we find it is the ETI related proteins, PTI related proteins, and only 3 proteins which are related to chitin triggered immunity. So here I would like to draw your attention. After this, what we tried to understand, because there were many other papers where people have tried to look into fusarium disease in other crops, particularly in tomato, and many papers in the PTI and ETI. However, there were no single paper in the CTI, that is chitin triggered immunity. So we tried to understand. But then if you know that the chitin and chitosan have been used extensively as a fungal disease control. So we try to focus on how this chitin or chitosan treated immunity evokes and what is the pathway. As this slide tells you that this is in this audience, I need not to say, only thing I have to say that we wanted to focus on chitin triggered immunity, and next few slides I would be focusing on that. Now, in this case, what we find that, this chitosan is nothing but the deacetylated form of chitin. So now chitin is a PAM in this case, but chitosan is not a PAM. Chitosan gives a protection that we know. So this chitin oligosaccharide, it is an archetypical general elicitor, that is the chitin, and typical fungal MAMPs that induce defense response in many broad host range, including plant, insect, mice, human, and therefore it suggests there shared occurrence in chitin-mediated defense machinery in higher eukaryotes. Since we are interested in multi-host response, and there is a very nice connection with the chitin and chitosan, therefore our interest was in this particular pathway. Now, notably, this chitosan has been shown, if you see, has been shown to form physical barriers around penetration site and inhibits the hyphal growth of many phytopathogenic fungi, including fusarium. This data came long back, and then also it has been shown from El Hasnei's group that in date palm, chitosan treatment leads to enhanced peroxidase and polyphenol oxidase, and in response of this FOSC, and that provide the vascular fusariasis resistance. We also have papers where people have shown that P-fusarium solani interaction is dependent on phytoalexin synthesis, and that is also inhibit this chitosan, inhibit this fungal growth through this phytoalexin biosynthesis. So therefore, this also tells you that chitosan does not go through only one pathway. It in some cases it is polyphenol peroxidase, in some cases phytoalexin. And what we found, I'll be talking to you in next few slides. So in our data, we found, as I said, that we have already developed the nucleus and the ECM, but I'm not going to speak to you on nuclear proteome data. I'll be talking to you from the transcriptional angle of the nucleus, but from the proteome, we initially targeted in our first data as a perception, the chitin-triggered immunity, what happens in the ECM, where it is a major player during fusarium-induced resistance in vascular vein. In another paper, we also showed that this whole microarray data, the RNA-seq data is just came and it's not yet published, but I'm going to show you that unpublished data as well. So what we did first, we actually set out a screen, and in that case, we grown the fusarium onto a different concentration of chitosan concentration on the plate. As you could see, the moment you have a 0.6 milligram per liter chitosan on a plate, you see the fungus is more than say 50% is inhibited. That tells, yes, fusarium is also chitosan responsive. So what we did here, please note that I would be taking in all my slides these terminologies. So we say C minus F plus and C plus F plus. What does this mean? It means it is not chitosan pretreatment, but fusarium challenge. In this case, it is chitosan pretreated and then fusarium challenge. So as you know that this JJ62 is a susceptible genotype, if you don't do a chitosan pretreatment and you challenge it with the fusarium, you see after 120 hours, the plant is completely dead. The moment you do a chitosan pretreatment of the seed and then grow and give a challenge, and that means there is a memory which is there in the plant, it actually reverts back the JJ62, just like something like WR, where even without chitosan, with chitosan, there is no death. So it's very surprising that only one molecule is giving almost more than 90% resistance to this otherwise completely susceptible host. Now, we tried to map the disease severity. Wherever you have no chitosan, you see very high amount of disease. With chitosan, the disease is very less. We tried to see the fungal biomass. Similar was the case. We also, because I told you it is a wilting syndrome, so we also measured the electrolyte leakage, and it becomes a similar story in this case. So then what we wanted to see, since we were interested in ECM, and as you know that, the first response will be in the ECM and the membrane. And the question was, when the plant is challenged by the pathogen, what happens to the ECM? So as you could see from the scanning data, microscope data, that in the chitosan minus not pretreated, if you challenge with fungus, there is a total dissolution of the cell wall. However, the moment you do the chitosan pretreatment, the cell wall comes back to its normal shape, just like the immune plant of WR315. Then the next question was, then what are the chemical compounds which basically is giving this strengthening in the cell wall? So we measured by Raman spectroscopy all the compounds starting from pectin, cellulose, lignin, and many other carbohydrates. What I'm showing you here, that in case of this, we found that not the cellulose or lignin, but it is a pectin, which actually is more when you do a chitosan pretreatment. And therefore, we find that there is a major role of pectin in the cell wall strengthening upon chitosan treatment. So in that case, what we understood that chitosan-induced immunity is getting regulated by the pectin as well as a metabolite. Now, what we then did is that we took out total metabolite profiling data. And as you could see, this data comes from GCMS analysis. It's so interesting. Just looking at the heat map, you could figure out that the whole heat map where you didn't do a chitosan pre-treatment, but you have challenged it, it's very different. The moment you do a pre-treatment and in the susceptible genotype, you do the challenge, you now look into it with the treatment with the WHU-5 at the end, that is 480-hour HPI, and also with chitosan treatment, it looks so similar. And so this part is totally downregulated, whereas this part is totally upregulated. We did this pathway analysis, which pointed out the galactose metabolism, amino acid metabolism, glucose, and few six of the amino acid metabolism were getting enriched in this. Now, with that, we developed a diseasome, ECM diseasome. Here, the major protein hub proteins became the germine and APX2, and we also have the hub, which are wall architecture remodeling and disease response. What we see in the immunome is completely a different story, and in that case, it becomes beta-galactosidase, purple acid pyrophosphatase, and we saw ETP signaling, EATP signaling, extracellular ATP signaling, and ROS homeostasis is evoking. So in that case, next what we did, we actually tried to see that when you do this treatment, whether a ROS generation is very differently regulated. As you could see that in chitosan, where no pre-treatment, there is no ROS generation. The moment you do pre-treatment, you see a huge amount of ROS burst in the cell wall, just like the immune plants. So in that case, we measured all the ROS metabolic enzymes, and we saw in the chitosan pre-treated JG62, the ROS metabolic enzyme activities are very high. So in that case, what we found, there is a stomatal immunity that is evoking, which was known in case of this. The stomata closing and opening is regulated by all these metabolic pathway. Now, this is our mechanistic model where we have actually found out that EATP stomatal signaling, NO signaling, ROS signaling, EATP and oligosaccharide, they all are playing major role. So from here, I will just last part concentrate on 3 or 4 slides where we went back to our microarray data and we tried to see which are those transcriptional regulator which can be chitosan induced and chitin induced. We found out, we named it IRF1, which is immune responsive factor 1. And if you see, this is a nuclear localized gene. From our earlier data, we actually saw all of them, but we found that this is very highly expressed in chitosan. So this is basically belong to a BHLH domain containing protein. In our data, we have 6 of those protein. We concentrated on IRF1 based on its very expression change. And as I said, it is a chitin responsive gene. And we found that this has 2 domain, and we already patented this gene. Now this BHLH in plant have been actually shown to regulate iron homeostasis, cell elongation, nodule senescence, JA signaling, and also immunity. In very, very recent paper in PLOS Pathogen also showed BHLH 84, but in our case, it is a other ortholog. So this gene is actually very much root specific under FOC1. So it's an induced gene, very early responsive gene. And if you see, it is not responsive to the dehydration. It is very specific to fusarium expression and the chitin expression. The hormones, it's salicylic acid, jasmonic acid, SEC and BR that regulate possibly these pathways, and these are not unlikely but downregulated. So we actually went ahead and tried to see whether it is localized in the nucleus. Yes, indeed, this shows you that it is localized. We did it by, the biopsy, as well as we did it by East2 hybrid, and you could see that it is a nuclear localized gene. It also becomes a dimer, and that dimer so it is a homodimer basically. And then we tried to see, since it is a transcription factor, that which of the motif it binds. It is a G-box binding motif, and it interacts with G-box in a very sequence-specific manner. As you could see from here, the abrogation when you do a cold challenge. We also found out that this works as a transcriptional repressor, and in that case, what next we did, we set out a pull-down experiment, and from the pull-down experiment, we have almost 29 nuclear localized proteins, and this IRF1 interacts with all of them. We validated till so far some of them. This work is still ongoing. What we find, it actually interacts with its orthologs, which are IRF9, IRF10, and IRF8. It also interacts with MADS box. So here we are trying to see how this IRF in Arabidopsis, it's known that BHLH in 3 different BHLH can make a complex, and that drives the development. But here also we are getting almost 3 and MAD box, 4 BHLH and MAD box. So we are trying to see how this whole pathway is regulated to give the immunity. My last 2 slides. So we also did a overexpression and a complementation study in the Arabidopsis. And as you could see that if you actually have this kind of study in a mutant, and in that case, it is completely dead on Fusarium challenge. But if you overexpress this IRF1, you see a reversal. And if you also see the mutant is complemented, there are some bit of reversal. So with that, it conclusively says that it is giving the immunity. It may be the master regulator. So it's unlike to the worm where it is a DAF16 forkhead, but here it is basically a BHLH family, which is giving this whole, which rescue the phenotype basically. So we have also done a Rhizogene's different method. This is old data, but now we already have the CRISPR lines of all these different if proteins, and we are trying to see what happens with the overexpression line to understand the downstream genes, both from the DAF16 in worm and also in the IRF1 in the plant, to see through the chip sequencing and also to validate our protein interaction data. So with that, the next questions that we are currently trying to address: how does ECM and nuclear crosstalk regulate the immunity through PCD? How does IRF1 regulate immunity through PCD, particularly, and dissecting the role of DAF16 and SAR2.1 interconnection. So with that, I would stop here and would like to will be happy to take question. But before that, this is my current lab group, this is my old lab group, and these are all the people who have contributed earlier in this work. Whomsoever name you are seeing in the green, they all at something or other they have done. It started with Nagaraju and Nashiman, very early days of my lab. And then right now it is being done by Shobha and Archana, who is actually working in this whole area of the transcriptional network analysis and more of the functional analysis. So this is Archana. Shobha just joined, so I miss Shobha's picture, and this all work is supported by NIPGR and also by the DBT, and now I have a program from SCRB. So with that, I would stop here. Thank you very much.
Thank you, Subra, for such a elaborated talk, and it really is very nice to have a comparison between the plant immunity and the animal-based immunity. So I do have one question. I think we are running short of time, so we can take 1 or 2 questions at this particular time, and then probably we will be discussing more in the case at the time of the panel discussion. So yeah. I do have one, the small question. The how you infect the C ligands. Actually, but then
it's very easy. No, it's very easy, Praveen.
Okay.
So it's very easy. Actually, when you actually grow C ligands, you grow it on its natural food, that is OP50, which is an E. coli. So when you make the plate, you actually mix OP50 in that. And when we do the fusarium, we don't put OP50, we put fusarium. So that was the first thing that we wanted to know, whether it will take fusarium as its food and it can grow. So can it grow as a natural, take fusarium as a natural host actually. C. elegans can be taken as natural. So it engulfs the fusarium or OP50, wherever you are doing. So you have to grow spore, you make kind of a spore suspension that all of us do, you also do in your lab, and you just mix it with the plate when you are pouring the plate.
And does the fusarium overgrown on the plates, or it remains in the just like?
No. So it depends on that which are the mutants you are actually working on. So in some cases you would see that fusarium would overgrow, but where there is nothing, but it's so rare case, so many mutants that we have done, but it's very rare case, 1 or 2 times we have seen that. Otherwise it engulfs. So what happens before fusarium actually overgrows, it is taken up by the C. elegans. So it has already ingested it.
does it have,
So it will overgrow. If it is there, you will see the fungal hyphae within the intestine of the C. elegans.
So why I'm asking this question, because at the time of the entire the transcriptome and the metabolome analysis,
there are fusarium genes. There are fusarium genes.
That's why I was asking. And also like sometimes they will try to deplete the media also whenever you are so the nutrition.
No, it's very fast. It's very yeah, it can happen, and in that case you would see a avoidance phenomena. But where it is very much infecting, you don't see anything, because very fast it makes the worm die. It's within 24 to 48 hours, the worm is dead.
I think there is one question from Prabhu. Prabhu, you can ask question.
Yeah, good morning, Dr. Subra.
Yeah, good morning.
So my question is similar to Subhendra, like, so can chitosan in feed prevent fusarium infection in C. elegans?
Again, repeat.
So can feeding chitosan containing feed prevent fusarium infection in C. elegans?
Nice question. We are now trying to do this. I can't tell you right now.
Okay.
Professor Dr. Mukherjee, have any question?
Yeah. Dr. Subra.
Yeah.
Can you hear me? Yeah. See,
I can hear you.
Wanted to know, like, see, the bioactivity of chitosan depends on the chain length. So what was the
what was the chain length? I don't remember it. The student would remember. I don't really remember. I can tell you later.
Okay, no problem. And second thing is that, see, it's so effective.
Because most of the case, whatever chitosan you get, it is a mixed chain length, most of the case. So I have to find out. So it depends on which company you are buying this chitosan. So I forgot that. So I'll let you know later.
It's fine.
I think Divya is...
Why it is still a problem? Like, if chitosan is so effective against fusarium,
it's very effective. So a nanoparticle based formulation might work. So we are trying to see that. So maybe it will work, and we don't know, because chitosan has been used, as I have shown you, even for other animals and many other animals, as well as many other species. So it's not a new thing. It's an old molecule, but we tried to understand the mechanism. But it's an old molecule.
Yeah. Okay.
Divya wants to ask.
Yeah. Hi, Shubhra. Good morning. Excellent talk. I just had a general question. What about the effect of chitosan-induced immunity on beneficial fungi, such as mycorrhiza? What happens in that case?
Divya, I haven't done that, so I can't answer your question. And I'm sorry, because I always kept my mind in the fungus which infects. So I haven't actually read the literature. I won't be able to tell you, but definitely this is an interesting point. I'll read it and then discuss with you.
Thank you.
Yeah. Praveen, a quick question.
Yeah, please.
Yeah, Subra, nice talk. Hello, Subra.
Yeah, I'm hearing, please.
So IRF1, you're saying, is acting as a repressor. So acting as a repressor, is it repressing repressors of innate immunity? I mean,
absolutely. And that is another transcription factor which we have, we think it is repressing that, but we haven't yet done the exact work. And that transcription factor is known to control the PCD. So basically it is a repressor of PCD, which possibly BHLH is repressing. That is our hypothesis right at this point. Maybe another 1 year.
Sure. One last question, and that this time I think Subhendra wanted to ask.
Yes, madam.
Yeah.
Very so several exciting questions coming to mind. So chitosan is toxic to fusarium because at higher concentration it inhibited, and chitin is a part of the cell wall. Some kind of what is the signal for stopping its growth?
So So the chitin is a PAM or the MAM, whatever you call. So it evokes the PTI pathway, but the chitosan is the deacetylated form of the chitin. So it is only the deacetylation which makes the chitosan. And the moment you deacetylate chitin, this chitosan then gives the protection through the chitosan pathway. So signal that way here, I would say chitosan is evoking, but exact signal, if you, I don't know what you are thinking, whether you are thinking about hormone downstream or any other thing. I'm not very clear what you are asking.
No, madam. Chitosan is inhibiting the fusarium growth.
Right.
Is it because of some kind of...
It is inhibiting the fusarium growth, but at the same time, I have shown you the whole data coming from the proteome. In that case, it also actually is going through a particular signaling pathway, right? So therefore, it is evoking a signaling pathway, and this transcription factor is a chitosan-induced transcription factor. So we still don't know what connects, and so we have a couple of the receptors. Only thing we haven't yet tested them, whether they are also falling in the same pathway. Right now, we are trying to connect this, the cell wall receptors with this nuclear localized transcription factor, that IRF1, and what we already have a clue downstream. But this part, we do not have a clue right at this point of time. I think from the interaction data that we are going to validate, we would be having much better clue, maybe for the MIPAM next time.
What is the, madam, duration of
Suvendra, yeah, we are running late. So, the panel discussion, maybe, we will have another opportunity to interact with Subra. Okay.
Yeah, so only thing I'm just telling, because I have some other meeting, so I'll be there in the panel discussion. Just tell me which time.
We are 20 minutes late already, so you can just see. Yeah,
because we started also late, Praveen. No problem. I'll adjust that, but my video cannot.
If you want, we can finish your questions right now.
No. Okay, you can do that. That will help me a little bit, but I will join. My video will not be on. I'll join from home.
It's all right. I can see a few questions in the chat. The Ankita Sri has asked that chitosan masks the chitin molecules to avoid its recognition to the plant, so that the fungus can infect its host. How chitosan application will provide the resistance to plant?
So Gitashri, chitosan basically, if you apply, it will stop the growth of the pathogen on the surface itself, because even if people try to do chitosan nanoparticle, so if you do that, it will be a spray, right? So if you spray it, in the beginning, the fungus cannot grow basically on the surface. So in this case, fusarium is a root-borne pathogen. I don't know how do we can't spray it. So we need to find out something if this can be applied in the root, because the pathogen is going through the root. So I still don't have the complete answer. So I won't be able to tell you exactly.
Dr. Sonti also wanted to ask question. You can ask and interact right now.
I've asked already, but one more question I have, Subra.
Yeah.
You said there is a change in the pectin. I mean, I couldn't really tell how you had actually pinpointed it to pectin.
Yeah, that is basically, we did a Raman spectroscopy. So that Raman spectroscopy basically gives you all the carbohydrates very nicely. So you can actually find them out. And from Raman spectroscopy data, we found out it is a pectin.
So the pectin, I mean, for the others, the levels go down, and for pectin, the level doesn't go up, or
no,
for pectin, the level goes up?
Pectin, the level goes up,
okay.
are same.
Okay, yeah, sure. Very interesting, Subra.
Yeah, so this Raman spectroscopy from JNU was very helpful to understand this whole thing actually. And the metabolite data overlapped it. So that way it's very nice. And cell wall, since you also have a lot of interest in cell wall. So
right.
Very, very important organelle actually. So we still haven't yet looked into so much of huge data. No, so it takes a lot of time. I haven't yet got that much of time to look into the whole data of metabolite, connect it to the whole transcriptome and proteome data. We are now trying to develop that with phosphoproteome also by merging all these networks. So once that merging will be done, it will be more pinpointed.
The other query I had was your EM pictures that you showed, where there is a lot of degradation of the cell. So those are actually part of the parenchyma or part of the xylem?
So it's basically a scanning. So it's basically a scanning.
So the surface, is it?
It is a surface.
I see. Okay, so even in the surface, there is a lot of
So the whole thing is dissoluted actually. So what happens, it is there long back when we started working long years back. There are, I forgot whose paper was it. It says that there are genotypes, genetics data if you see, or from the agriculture data, this pathogen cannot go inside. So first experiment that my first student did, Dipali, where we took all these genotypes and we tried to see how the infection goes in by cross section. And in that case, this WR315, if you see, the fungus will go up to the first layer in the xylem. So in the just it bypass and goes to the parenchyma. It doesn't go inside at all. If there are other genotypes where the pathogen even cannot go up to the parenchyma, it will be totally restricted outside. But since we had a collaboration to find out this whole R gene, now we have already got it now. So in that case, what happens is that we did not do that very immune plant, but now we have actually got the genome sequence done of all these plants. And that will be more interesting in next another 1 or 2 years. But what happens in JJ62, this pathogen goes and it's totally ramified up to the pith and it's totally blocked. So that way, it's a very nice mechanism that each of the individual genotypes have. So this mechanism is operating at the level of genotype.
Right, sure.
Yeah.
Sure. Yeah, quite interesting. Look forward to hearing about the resistance gene also.
Yes, so I can talk to you later, but we are in a very advanced stage. Sure. Another 3 months will make the paper possibly. We are writing that. Little bit of the experiment is pending where we want to also, we started with this long back, but it took long time. 2 students left and I could not follow that. So in that case, this resistance gene is basically coming from the study of 2 real population, as well as now we have the genome sequence data of 6 or 7 genotypes of chickpea. Just we got it. We know even the variants which are there, and it's very tightly linked. And we also have the the knockout using the paper that we made with rhizogenes, not in the full plant, but with rhizogenes. The knockout is giving very nice data and overexpression. So right now we are doing a kind protoplast based analysis to find out what are the target of this gene. So once that target we find out from the interaction study, then we have the whole story.
Very good, very nice.
Thank you, Subra. I think you without video also you can join.
Yeah. I'll try to definitely.
So yeah. So I think you can just calculate the time and then you can join.
Yeah. I'll do that. Yeah. I'll do that.
So
Thank
you. Thank you, Subra. So next speaker is Dr. Ranjan Timuli. He is from IIT Guwahati and going to speak on calcineurin CRG1. Signaling pathway. So looking forward, Dr. Timuli. So,
yeah, good morning to everyone. Hello.
Yeah, we can hear you. We can hear you.
Okay. All right. So I thank Subodip for his kind invitation, and let me tell you that I share a special relationship with his parental lab. So when I got selected at CCMB, I first approached Ramesh sir for doing a PhD under him. But that time sir didn't have any project, so I had to settle with something else, and that's how I landed on a Rossborough lab and till now I am continuing work in Neurospora. So today I'm going to talk something about say calcineurin. In over a couple of years. So work done by various lab members could reveal like many interesting aspects of the calcineurin Neurospora crassa. And this, before going into that, I must thank the previous speaker to make my job easier. She could nicely, elaborately describe like importance of fungi as a pathogen. So among all the organisms, many people are good at, say, human, we eat a lot of food, right? And also different varieties, right? Too much as compared to many others. This basically helps like to maintain like very high body temperatures, right? And that's how we are so lucky, the mammalians, and that dramatically help us to evade the fungal pathogen. During the course of like global warming and various other reasons, so day by day, many of the fungi also has become tolerant to high temperature, and that's how they are right now causing a serious threat to not only to human animals but also the lot of different plants as well. In the So I hope my slides are moving. Is it moving?
Yeah, it's moving. It's moving.
Okay. All right, good. So in our laboratory, we started our work with many different types of calcium signaling genes. In Neurospora crassa, genome is like completely sequenced. So it has about 10,082 different types of genes. And out of this, we have about say 50 of them are belongs to calcium signaling categories. Out of all these calcium signaling genes, one of the very important calcium signaling protein is calcineurin. So what is like very basic mechanism of calcium signaling is that inside a cell, the resting level of calcium is very low. So which is about say 100 nanomolar. So calcium is mainly stored in various type of intracellular stores. For example, say mitochondria, endoplasmic reticulum, and the vacuoles in case of the fungi. So when there is some kind of signal, whether it could be like external signal or the internal signal, this calcium from the intracellular stores release, and because of this, or calcium may also enter from outside cell, and this both of these event can raise the calcium level temporarily slightly above to 100 nanomolar. So if the calcium level increases, then there are some sensors that can detect the calcium level, because calcium at high level is also toxic to the cell. And then once the calcium level increases, then there are some kind of calcium sensors, and these sensors have like a classical EF type calcium binding hand, and they can bind to the calcium molecule. And then once they bind, once calcium binds to its target protein, it can change its shape and structure, because it's a very charged molecule. And once the conformation gets changed, it can trigger the downstream signaling cascade. And among all these sensors, we work in some of the sensors, for example, say calmodulin and neuronal calcium sensors 1, etc. And this calmodulin, once it binds to calcium, it can activate another crucial calcium signaling protein called calcineurin. So calcineurin is like is also only known serine theorin say diphosphatase that requires both calcium as well as calmodulin for its activation. So once it get activated, it can remove the phosphate group, or it can cause dephosphorylation of a important transcription factor called CRZ1, And so once the phosphate, once the CRZ become dephosphorylated, it can go to the nucleus, and inside the nucleus, it can bind hundreds of different kinds of target gene, and those gene can target genes and expression of those genes have provide various kind of cell function or required for various type of cell functions. So this is like a very general mechanism. So like so it is basically driven by the temporary or transient increase in the intracellular free calcium level, and then this level activates some of the calcium sensors, which can eventually cause dephosphorylation of a transcription factor called CRZ1, and that goes to the inside the nucleus, and that can activate the target transcription factors. Right. So this calcineurin has like various type of different structural domains. The preliminary and calcineurin also is consisting of two different type of proteins. So one is like we have a catalytic subunit of calcineurin. Another subunit is called calcineurin regulatory subunit, and physical interaction of the both the subunit is necessary in order to have the active form of the calcineurin. And because what happened is that the fungi, as I said, in order to maintain pathogenicity, fungi must be able to adopt the high temperatures inside the host, particularly in the human. Right? There are many, or human or animal. So usually like mammalian body temperature is much higher than the optimum temperature required for the fungal growth. And so therefore, when fungi, such type of fungi infect the mammals, and they must get adapted to the high body temperatures. So therefore, like many people, they try to target like novel component or novel signaling machinery of the pathogenic fungi in order to prevent or in order to treat the fungal diseases. And people long time back came out some immunosuppressants, for example, say FK506, and this drug can eventually inhibit the function of calcineurin and their function of calcineurin, and that's how this can suppress the immune system. So the equivalent part or the homologous part of CRZ1 in mammal or human, it is called NFAT. NFAT. So these are both like homologous protein, and they they share the structural similarity as well as the functional similarity. Both. And so because this calcineurin is very much crucial for in order to establishment of the fungal pathogenicity in the animal as well as in human. So many people try to identify, or many people try to say target calcineurin for say for different drugs development, but it turns out that many other components that were previously identified for drug targeting having like high sequence similarity to their human component, but fortunately in calcineurin there is a specific region called serine proline rich region, or SPPR, that is basically found in only filamentous type of fungi, which is comprised of both pathogenic as well as non-pathogenic fungi, but it is completely absent in the human. And this particular region is like could be like a novel. This can be targeted for controlling various type of fungal diseases. So that's how like calcineurin has become both calcineurin as well as CRZ has become like a very very much sought in target by various researchers in order to prevent fungal diseases. Work in our lab, in a previous work in the lab, what we could able to show that in Neurospora crassa, this increased level of the calcium can activate calmodulin, which can further activate calcineurin, and then it activates CRZ1, and the CRZ1 goes to the nucleus and binds the specific calcium sensor called neuronal calcium sensor 1, and this is called neuronal calcium sensor 1 because in human and because in human and Drosophila, this is required for the neuronal development during the early developmental stage. And impairment of the NCS1 can also cause like several types of disorders, say, for example, bipolar disorder, autism, and so on. So this NCS1, once it upregulates, it can, the protein goes to the membrane and it blocks a calcium channel called MID1 so that no further calcium can enter inside the cell, and that's how cell can defend itself against the entry of the additional calcium to the cell. And we could also able to show that when we apply this FK5, this drug FK506, when we inhibit calcineurin, then this in that case the NCS1 expression or calcineurin expression get downregulated in Ross precursors. So this was some of the initial our work. Then the work with direct knockout of calcineurin is difficult in Neurospora crassa. That is, as I said, that Neurospora has 10,082 genes, and once after completion of Neurospora genome sequence, there was like a Neurospora functional genomics project, and the aim of the project was to generate the knockout mutants of all 10,082 genes in the Neurospora so that someone can do the functional analysis. But during that essay, it turns out that there are many genes that we cannot knock out because they are very much essential for viability. So calcineurin is also one of those essential genes. So that means both calcineurin as well as calmodulin are essential genes in Neurospora crassa. That means one cannot simply knock out calcium, either calcium or calmodulin, in order to study them. If you attempt that, that will be inviable. So to study such type of gene, there was one system was developed in our collaborators lab in University of California, Riverside. So they have generated one specific hybrid vector. So I won't go through all these details. I just showed some key point. Essentially, this vector contains a conditional promoter. For example, say promoter for a niche gene or TC1. So TC1 is a copper regulatory gene, and this promoter is sensitive to the level of the copper in the cell. So if I add copper sulfate in the media, then the clone gene expression is repressed. That means the gene will not get expressed if the medium contains copper sulfate. And in contrary to this, if we add, let's say, copper chelate or let's say BCS, then expression of the gene is possible. Then the gene will get expressed. So basically, one can conditionally control the expression of the essential gene that is cloned under this type vector system, and this allows us to study the function of essential gene. So we also use same vector to study calcineurin so that we can understand its function in Neurospora. So when we did that, so we basically clone okay, just one more point. So when we clone the gene of our interest, so this also vector has like one additional important part, that it whatever gene we clone here, that is targeted to phenothenate locus. That means the transformant will become exotropic for the phenothenate, so one can select it based on its pen exotroph. That means we know that where our transgenes goes. So when we when we clone this both subunit, calcineurin catalytic subunit, calcineurin regulatory subunit. So here to tell you that, as I said, calcineurin has two subunits. One is catalytic subunit and another one is regulatory subunit. And both subunits are encoded by two different genes. And so we basically clone both these genes both these genes and transform into a Neurospora recipient, and that's how we could able to generate the Neurospora strain, where the endogenous copy of both calcineurin and calcineurin catalytic subunit as well as the regulatory subunits are deleted, but they have one ectopic copy, that is like an ectopic copy in the pen to locus, and thereby we could control or regulate their expression based on the amount of simply by supplying the media either with BCS or copper sulfate. So it is like a beautiful figure that we can see that when we add the BCS, BCS, the growth of the one say in the middle panel, this is calcineurin catalytic subunit construct. So in this state, the only in this state, when we add copper sulfate, the expression of the calcineurin calcineurin is expressed, and therefore it is like a beautiful, nice colony, similar to the like wild type. But when we add the copper sulfate, you can see the growth of the colony is severely restricted, and it eventually die after a few days. Same is happening to the regulatory subunit. So when this stain is expressing the calcineurin regulatory subunit under the copper regulated promoter, so if you add the BCS, so BCS drives the expression of the calcineurin, and therefore the stain is perfectly fine. And in the presence of copper sulfate, the expression is reduced or repressed, and we can put again like a very much restricted growth, something similar, same like the calcineurin catalytic subunit. So we also could do by using this construct, we do like several similar other experiment, and that essentially we could able to show that that calcineurin is required for both asexual and the sexual developments. So here to remind you is that neurospora is having like a two different developmental stages. So it has it will reproduce asexually when there are a lot of media in a and if there is like a nutrition nutrient limiting, if it goes in the under nutrient limiting condition, specifically if suppose the nitrogen is limited, so it will reproduce to sexual mode. So what we also, so here I have shown one defect in the vegetative phase. Similarly, this calcineurin, when we when we like shut down its expression, so those type of those stain, it cannot form any female structure that is necessary for the fertilization. So that's how we could able to show that shutting down the expression of calcineurin causes defect in both asexual as well as sexual developments. We could also could do some kind of say co-immunoprecipitation as well as the confocal microscopy, and doing the both analysis. What we found that the both catalytic and catalytic as well as both catalytic as well as the regulatory subunit of calcineurin physically interact, physically interact. So this assay this was also done by using the same vector system that I have mentioned before. So if we again go back to this vector, so like in this vector is having like some kind of tag, for example, say V5 and the ZFP. So we had another similar vector that having that was having like STEC and RFP. So using some combinations, we could design our experiment. We could like clone both calcineurin catalytic and so they are both calcineurin catalytic and regulatory subunit physically interact, and this interaction activates the calcineurin holoenzyme this calcineurin, we also able to generate some additional mutants. So what the previous slide I have explained is that we could able to study the cell function of the calcineurin by cloning them in a conditional vector system. And here we are showing like this is like some tube. So this tube, so this kind of tube we use it for to study the aerial hyphae, growth of aerial hyphae in the Neurospora crusher. So in the fungi, when we grow in the liquid media, so this tube contains a small amount of liquid media, for example, about say 2 ml. So this so Neurospora will produce. So when you grow Neurospora in this kind of liquid media, it will produce aerial hyphae because the Neurospora conjugates are hydrophobic and they tend to go away from the water surface, and then here in the panel, so wild type and some other strains. So these strains are basically like some mutants of calcineurin, and these mutants we generated by using a technique called reap-induced mutant. So that we are not discussing here right now because of time constraint. So basically these are some additional strains of calcineurin having some specific mutants in the various domains. So some of them have having a mutation in the catalytic domain, some strains have mutation in the regulatory domain, and so on. And so having a mutations in various parts in the calcineurin is also show severe phenotype and then, as I mentioned, that calcineurin, I mentioned that in Neurospora, in fungi, for fungi, adaptation to high temperature is very much crucial for various functions. In order to adapt themselves in the hot environment, or like a host having the high body temperatures. So we tested. So we therefore we were interested to see the role of calcineurin and some of the heat shock protein that are found in Neurospora crusher in this process. So we did some initial round of preliminary experiment, and then we could see that there are in Neurospora crusher there are several heat shock proteins. So out of all this, there are two say main and primary heat shock protein. These are called say HSP80 and HSP80 as well as the HSP60. They are having some kind of importance cell function. So we tested their expression level under various conditions. So we what we have found is that when we test the expression of cell, let's say here HSP80 here, so in the figure panel B, so as compared to the wild type, there are some stains. Those are, let's say, 590, 600. So these numbers are, so these are some stains of calcineurin B and having their having mutations in the various domains. So this mutant stain we generated in the laboratory by this process called repeat in point mutations, and this mutant of this calcineurin having like some kind of problem in the expression of the HSP80. So so that gives us some kind of clue that there are some kind of link between the expression of the HSP80 and wild type calcineurin, and so we could able to do it further by we also test it by using further additional the calcineurin inhibitor. This is like FK506. So when we so here in the below we can see that when we add the F1 drug FK506, which is a blocker of the calcineurin or inhibitor of the calcineurin. So when we inhibit the calcineurin, and then the expression of the heat shock protein HSP80 has become drastically low, low as compared to the without drug condition. And surprisingly, the expression of the transcription factor, the CRG expression, was also low in presence of the FK506. So we 506. So we have done some other. So we did further molecular analysis, and we would able to show the interaction of this physical interaction of the CRJ1 with like HSP80 and that work has been is right now under review. And so in addition to like HSP 80 regulation, then there was another important function that we found, that the calcineurin CRJ pathway is also very critical for driving the expression level of NCA2, and this is also another very crucial calcium protein which is required for resist tolerance to calcium levels. And so we did like further molecular analysis for both NCS2 as well as the HSP80. So this is our we can ignore this complicated figure. So what we did like some kind of chromatin immunoprecipitation assay, followed by as well as the electrophoretic mobility SIP assay. So by doing all these both SIP as well as the EMSA analysis, we could molecularly identify the CRG1 binding sites in both in the HSP80 as well as the NCA2 under various conditions. So these were like a first report showing that CRG1 molecularly bind to such and such sequences in the HSP80 as well as NCA2 genes in Rossborough crusher. And these are about like a unique sequence, and these sequences are different from the sequences that are described for the Saccharomyces cerevisiae and related fungi by some other people in the previously. And then what we can think of is that basically when we have there is raise of calcium. So as I mentioned previously, the calcium can increase because of various by both either entry of calcium from outside the cell or by the release of calcium from the intracellular stores. And this activates some of the calcium sensor, and one of the main calcium sensor is the calmodulin, and this can activate like calcineurin. And again, the calcineurin has two subunits as shown here, like catalytic subunit and regulatory subunit. And we in our lab we showed like the physical interaction of both this calcineurin A and a B. Also, we could able to show like calmodulin also interact with them in order to activate this enzyme. And here also, and we could also able to see like some interaction with about HSP80. So HSP80. So basically, so this is a kind of complex network, and this is a network, and this network is very much essential for providing the Neurospora cell to provide tolerance against the both heat shock as well as other stresses mediated by either say a high amount of calcium in the outside media. In addition to that, we could also show that this transcription factor is very much necessary for say production of carotenoids. So here I didn't mention. So carotenoid is like characteristic the pigment. So Neurospora crassa, once it give a conidia during the vegetative phase, these are orange in color. So these orange colors come from a specific pigment called like carotenoids, and this carotenoids is required for providing the fungus the protection from the harmful UV radiation in the light. So this carotenoid in Neurospora is called Neurospora xanthine, and this Neurospora xanthine is produced via a long complex biochemical pathway. And there are many genes, like for example albino 2, and then cow2 and the yellow one. So these are the expression of certain genes also regulated by the CRG1 transcription factor, and we could able to identify binding site of CRG1. So essentially CRG1 binds to like multiple genes in Neurospora. So those genes also require and we could able to identify many of such molecularly identify many of the CRG1 binding sites in various genes in Neurospora crassa. So I would explain. So I explain or describe most of the work today here done by Abhishek. So who is doing currently postdoctoral research in University of Texas Southwestern, And so I also mentioned the debuggers work. So they have completed PhD. Some work from the Aussie, very little bit. So these are like some of my current and the past lab members. And I thank Fungal Genetics Stock Center for providing us the strains, particularly the Kevin McCluskey, who was and Eric. So they were very generous for sending us. Then our collaborator like Borkovins from UCR, and our lab is and then Ministry of Human Resource Development for funding the student, and our lab is currently funded by DBT, and also we thank US Science and Technology Forum and DST, IIT Guwahati, and I thank all the lab members. So thank you very much. So if you have any specific question, I will be happy to answer.
Thank you, Dr. Temali. We are going to have a discussion, a panel discussion in the end. Actually, the format of this entire the MIPAM is in such a way that we should have a chance to interact with each other. It's an interactive symposium. So it's not a symposium; it's an interactive meeting as well. So maybe in the end we will be discussing more. I think one or two quick questions. I think Subhendra want to ask a quick question.
Yes, we'll. Ranjan, namaskar. So very wonderful. So
Namaskar,
just
sir.
curious to know that you using neurospora also secret proteins that sense outside calcium, extracellular.
Yes, sir. What we found, so what Onina, so see Onina and also some other people, neurospora basically secretes a particular secretory phospholipase. So that comes out of the cell, and that is required for like degrading cellulase. And that's what we found, like cellulase, if the cellulase is present in the outside. But the calcium sensing proteins have not been identified. That has come outside the cell so far.
Okay.
Okay. Yeah.
Praveen sir, we may.
Yeah.
Fine. We are running late.
Yeah. So this is my opportunity to invite the next speaker, and I think he is new to this MIPAM, and he is Dr. Swarup Roy Chaudhary from ICER Tirupati. So he's going to speak about RLK mediated activation of root nodule symbiotic pathway. So another different that he is going to speak about. Dr. Roy Chaudhary.
Yes, I'm here.
Yeah.
So is it possible that I'm not able to share it because I think Dr. Ranjan, he's already shared.
Okay, Dr. Ranjan, you please.
Great.
Yeah. Now you can share.
Hello.
Yeah, it's coming.
It's coming, right? Yes.
Yeah, you go for presentation mode. Presentation mode from the.
Yes.
Not from there, from the bottom. Yeah. Now it's fine.
Fine, right? Yes.
Yeah. It's absolutely fine.
Okay. So at first, I want to thank, and it's a great privilege for me that I'm now part of this MIPMA group. And I'm really thankful to Shubhadeep and also other organizers to give me an opportunity to present my research work. So I joined ICER Tirupati just 3 years back. And now we are really blessed that Professor Ramesh Shanti joined our institute as head of the department. So today I'm going to talk about one of the research work on the RLK mediated activation of root nodule symbiotic pathway. So currently in my lab, we are working basically root nodule symbiotic pathway to delineate the signaling mechanism. In addition, we are working on the drought space our goal is in future to delineate that how drought state mediated signaling modulate the nodule symbiotic pathway. So anyway, so if you look at this picture, you will see here, there is one component. It's called NOT factor. This is derived from some bacteria. And if you look at this image, this is a plant receptor. You see this receptor has an extracellular part and the intracellular part, and in between there is a plasma membrane binding region. So this NOT factor actually binds this receptor, and therefore some downstream signaling triggers, and ultimately it reflected, it form up some nodules, and these are the root nodules, and it only possible in leguminous plant. So if you think about the molecular communication, it is a relay of new paradigm. And this molecular communication always starts with a signal. And that signal is witnessed in prokaryotic in form of antibiotic, toxin, or exopolysaccharide. In case of plant, these are phytohormone, phytoalexin. And in animal also, it is visible in the form of pheromones and scent. And these molecules, when they release from either bacteria, plant, or animals, they usually bind their receptors. And this ligand receptor binding then initiate a molecular communication in all living organisms. So plus think about in plant. So in Arabidopsis, if you look at that, how many this kind of receptor-like kinases are there? So if you look at this image, there are multiple receptor-like kinases are there in Arabidopsis. And the numbers is near about 600. And all the typical picture, if you look at carefully, it has one extracellular domain that is required for perception of the signal molecules. And there is the intracellular kinase domain that is required for the phosphorylation of downstream signaling. And typically there are several kind of receptors are there. If some are lysine receptors, if you look at here, it has a 3 lysine domain containing extracellular domain. Some are leucine rich receptor, some are some lectin perception, lectin rich receptor. So what the function of this receptor? So actually this receptor, when they function, you see these are the receptor, they actually function either homodimer form or heterodimer form. So this is a receptor. When a ligand bind here, you see it associate with another receptor. It is called co-receptor. It may form homodimer or it may form heterodimer. So after binding up receptor, first it autophosphorylate in the main receptor, and then after binding with the co-receptor, it transphosphorylate the co-receptor. And this phosphorylation allow them to interact with some downstream other kinases. It may be RLCK. And this total signaling then activates sometimes G protein mediated signaling. Sometimes they directly activate some NADPH dehydrogenase to stimulate the ROS production. And this ROS sometimes activates several channel protein that releases calcium. And this calcium again activates calcium dependent kinase, or this ROS directly activates several kind of MAP kinase cascade. And this MAP kinase cascade ultimately triggers several transcription factors. And even there are some other process that this phosphorylation of receptor may directly activate this transcription factor with some other intermediates. So all this transcription factor, finally they stimulate different genes, and this gene related product protein releases, and this protein basically responsible for growth, development, plant immunity, as well as different kind of symbiotic response. Now coming to our point, the commencement of symbiosis, there also one kind of this ligand receptor interaction is essential that trigger the signaling system. The molecular communication between plants and rhizobia during the establishment of a synergistic relationship, now a hotspot in research. And this interaction between the molecules and receptor commences the symbiosis. And this symbiosis signal ultimately modulate a series of signals that help the bacteria to stay inside the plant in a special kind of architecture that is called root nodules. So if you think about the general outline of NOT factor induction, biosynthesis, and transport in legume rhizobia symbiosis, so last 30 to 40 years, there are several groups, they work on different rhizobia and they analyze different mutant of bacteria and they predicted different hypothesis how this NOT factor synthesized in bacteria. So based on their hypothesis, we have mapped and we segregate them in different groups. So we designated that this way. Suppose plant root releases some flavonoids, and these flavonoids then triggers some of the rhizobial cell. So how they triggers? The flavonoid enter the rhizobia and they trigger some of the box protein. It is called NOT box protein. We consider this protein now is as patroller. So when rhizobia releases NOT factor, it activates several patroller protein. And inside this rhizobia, there are some plasmid is present. It is called P-sim plasmid or episome. So it contains some NOT factor binding forming genes. So when this P patroller induced by flavonoid, they bind the promoter region of this kind of NOT gene, and thereby this patroller stimulates transcription of several genes. And these genes actually are NOD, NODF, NODG, NODM, NODM, and they actually responsible for formation of N-acyl glucosamine, that formation of several kind of this ketosane backbone. So therefore, we consider this gene as a constructor of the formation of the NOT factor. So when this backbone constructed, they join together. Sometimes it is a 4 unit, sometimes it is a 8 unit, sometimes it is a more than that. So first patroller initiate the transcription, then constructor, they join all this oligomer together and they synthesize it. After synthesizing, they join it. And then there are some decorated proteins are there. So in this NOT structure, you see there are several R regions are there, and this R region then activated by different kind of genetic machinery is a NOD-U, NOD-L, NOD-Z, NOD-T, NOD-S, NOD-Z. So all this gene modulate this several R position by some of them, some responsible for methylation, some responsible for acylation, some responsible for carbomylation. So different kind of modification is integrated. So it means they decorated all these component and therefore it form a NOT factor. And the final thing is the dispatcher. So in bacteria, there are some dispatcher proteins are there, ATP synthase, and that facilitated the movement of this NOT factor from plants in bacterial cell to plant cell. So thereby this NOT factor can associate with this plant root cells. So these are the different constituent: patroller, constructor, decorator, and dispatcher that ultimately form the NOT factor, and then it binds to the plant receptors. So actually it occurs this way. So plant releases some flavonoid, it favors some their compatible some rhizobia, they activates. But one of the interesting thing is there, sometimes plant releases flavonoid, this flavonoid inhibit some of the other kind of rhizobia. So we consider them as a non-compatible rhizobia. So anyway, if they trigger a compatible rhizobia, you see here, this rhizobia then releases flavonoid, they activate the NOT factor cascade of the bacteria, and thereafter they releases the NOT factors. And this NOT by dispatcher, and this NOT factor come to the activate the receptors, and thereby signaling switching on by molecular communication. And thereafter, what happened? This rhizobia, you can see, it enters the plant cell by 2 ways. So here I am showing only one of the approach. This is called infection thread model, because most of the leguminous plant, near about 75% leguminous plant, they exactly follow this process. So what actually happened? This rhizobia, they enter the, they make a weaken of the some plant cell membrane in the root hair cell, and they enter there from the microcolony, and then it's initiate the another NOT signaling that helps the invagination of the plasma membrane. And through this invagination of the plasma membrane, this rhizobia enters inside the plant cell. And they cross for epidermal cell and then cross the cortical cell. So ultimately they reaches in the inside. And in the meantime, in plant cell, there are several hormonal signaling related to auxin and cytokinin. They stimulate the continuous cell division, and that form a big structure, and this structure accommodate all this rhizobia that release from the infection threats, and they differentiate it in form of bacterioids, and thereby they can fix the atmospheric nitrogen. But the question is, when not factor release, how this whole signaling process started, and that facilitated the infection threat formation as well as this nodal organogenesis. So if you look at the process, the molecular signaling of symbiosis, if you look at here, you see here this NOT factor actually binds some receptor protein. It is called NOT factor receptor 1 and NOT factor receptor 5. One is the major receptor, another is the co-receptor. So NOT factor bind with them, they attach together, phosphorylate the downstream machinery. So one of the evidence we have found, that is the G protein signaling and some other MBA pathway. So all these other signaling associated each other, then they trigger the calcium spiking inside the nucleus, that trigger the CCMK kinase, and thereby this CCMK kinase activates several other protein, and finally several important transcription factor like NEAN, NF-YA, and they transcription of several downstream genes. Although this part, the nuclear part, how these several genes activate the several the nodulation pathway in different time points, that is already proved by different groups. But the major lacking is there, that how this NF1 mediated perception activate this ion channel protein. That means the downstream signaling in the cytosolic part. This is still enigmatic. So therefore, it is very important to delineate the mechanism, how after perception of NFR and NFR5 that modulate the downstream signaling. In my postdoctoral research, I have proved some of the evidence, that is the downstream signaling of the NFR1. There I have shown that this G protein signaling some of the component act as a positive and negative regulator of root nodulation, and we delineate the mechanism where I have shown that NFR1 phosphorylates some of the G protein component, thereby they activate the this nodulation process by changing the expression of the some G protein component. So here suppose G beta and G gamma is a positive regulator and G alpha is a negative regulator. So when CMARK phosphorylate G alpha or NFR1 phosphorylate RGS, they helps to conversion of this active G alpha to inactive G alpha. So therefore they reduce the negative regulator and promoting the root nodulation. But this is not the whole mechanism, because this is a part of the mechanism, because when we complemented the NFR mutant in soybean, we have seen that it's not fully complement the nodulation. So it can able to do only partial complementation. So it suggests that there are some other factors definitely there that can modulate the downstream signaling. So the question I am asking here, how does the host determine the cognate microsymbionts? So in this perception, you can see that if you look at the NOT factor structure carefully, here you will see this is an extracellular domain, this is an intracellular kinase domain, and in the middle there is a transmembrane domain. And you can see here that how these receptors are there. If you delineate here, so you will see there are two kinds of receptor. As I told, one is the main receptor, another is the co-receptor. The receptor with typical kinase domain that undergo autophosphorylation upon binding with NOT factor and activate downstream signaling, therefore they can facilitate the transphosphorylation. They are called LIK class, and example is NFR1. There is another receptor, it is called atypical kinase. It is belongs to the LIR class that lack conserved residue required for the ATP positioning. Therefore, they devoid of both autophosphorylation as well as transphosphorylation abilities, and they are called co-receptor. Example is NFR5. So what actually happened if you look at this image, this normal condition, these are the NFR1, NFR5, these are the lysine receptors. So NOT factor, when bind with this receptor, they interact each other, and then they activate several effector molecules, and that effector triggers some calcium spiking in the nucleus and stimulate the symbiotic signaling in plants. So one of the MS thesis student, she started to identify all the LIR and LIM receptor from Medicago transcapsulata, and we have identified all the receptor, and we have found from other evidences that basically in Medicago, empty LIK3 is act as an NFR1 and empty NFP act as a NFR5. Similarly, another model legum, it is called Lotus japonicus. Here also we have identified all the homologs, and we have seen here also, this is one of the LIK is LJNFR1, it acts as a main receptor, and LJNFR5, it acts as a co-receptor. So if you assemble all of them together, we have seen that here is if you look at this tree, that LJNFR1 and MTLIK3, they belongs to same group and act as a main receptor during the root nodulation, and MTNFP of in Medicago and Lotus japonicus in NFR5 belongs to the same group. They act as a co-receptor in the root nodule symbiosis. So, but one of the interesting thing is here that this NFP mutant, if you study in Medicago, you can see this is the NFP mutant. So in wild type plants, you see after rhizobia treatment, all the root hairs they form some branch and they form some crozier structure. So that means they're able to perceive the knot factor. But in NFP root mutant, you see all the root hairs are straight. There's no crozier structure. So it suggests that NFP mutant, there's no root hair branching. And even that this NFP receptor, we have found that there is no calcium signaling and no expression of downstream gene. So it suggests that NFP is a signaling receptor. So several evidences in Lotus japonicus, this NFR1 and NFR5, they have also shown exactly similar kind of phenotype. So therefore, we can consider NFP is a signaling receptor, and in Lotus, both NFR1 and NFR5, both are signaling receptor. But the amazing thing we have observed is another scenario in Medicago LIK3 that is considered as a NFR1 in Medicago. So here you have seen this wild type plant, this when rhizobia enter, they form a microcolony and they then form a infection threads. This is a proper infection threads. But in LIK3 mutant, when there's a mutant, LIK3 mutant, we have seen there is no proper infection threads. Their rhizobias are growing haphazardly, and even they form an infection threads, they just form a vesicle-like structure and end up there. So that means they are not able to enter the cortical cell, or they are not able to form nodules. But it means that this LIK3 mutant, they act as the only entry receptor. The reason is, in this LIK3 receptor, there is a proper calcium signaling, and also in this receptor, there is a proper downstream gene expression. So it suggests that LIK3 is an entry receptor, is not a signaling receptor. So this is a dilemma between the two model plants. So where all the receptors are, both co-receptor and receptor act as a signaling receptor in Lotus, wherever in Medicago, one acts as an entry receptor, another acts as a co-receptor. So it means that more dissection is required to identify what are the putative co-receptor of this MTLIK3. So it means that this receptor binding is not the simple, the way we think, that just NFR1 and NFR5. So it means that as there are evidence from multiple copies are there in both Lotus and Medicago, probably they have some contribution to perception of the knot factor signaling. So if you look at here we have started my work in my lab in the chickpea. You know that chickpea is a very important plant in India. And we have identified all these LIK and LIR sequences, means active kinases and atypical and typical kinases from chickpea. And by blasting with the Medicago NFR1 and NFR5, we have found their compatible gene exists there in chickpea. And we have cloned this gene. And if you talk about the chickpea, you know that in India is the highest producer of chickpea. And previously, our previous speaker also mentioned that why chickpea is so important in India. But the problem in chickpea, in production of the yield of chickpea, if you see in India, it is just 922 kilogram per hectare. But in the other country like Canada, it is a 2077 kilogram per hectare. So we need to increase the yield of the chickpea in India. So therefore we need some strategy. And I think this one of the strategy may be if we able to fix the nitrogen fixing ability in chickpea, because chickpea nodules only form when the soil is suitable. And there's because we try to grow this chickpea in Tirupati, it's not able to produce nodules. This is the reason is the soil pH is one of the important marker for production of the chickpea nodules. So therefore, it is very important that if we able to increase the nodules numbers in chickpea. So and nitrogen is the major things. That's why we are interested of the nodule formation, and nitrogen is the integral part of the protein, nucleic acid, pigments, and enzyme. And if you able to increase the nodule number of chickpea, that will provide a lot of nitrogen in plants, and by uptaking several photosynthetic products that is necessary for bacterial survival. So this way they can form a symbiotic relationship. So we first isolated all this NFR protein from others legumes like groundnut, soybean, and some medicago, lotus, and also NFR5 protein, and we make a phylogenetic tree. If you look at carefully this phylogenetic tree, see this NFR1 very close to located to medicago LIK3 as well as lotus NFR1. And another way, this chickpea also NFR5, you can see it is very close to medicago NFR5 and lotus NFR5. So that means in other groups, that soybean groups are present in the different lineage. And if you see here, if you segregate all this NFR5 and NFR5, they are segregated into two distinct groups. So am I audible?
Yes, yes.
Okay, so then we have studied first the localization of NFR1 and NFR5, and we have seen that both chickpea NFR1 and NFR5, they are localized in the plasma membrane, and this is obvious because this is the membrane bound protein. And then we have studied the expression level of chickpea NFR1 in different tissues, and we have seen that it is significantly expressed in the root tissue as well as nodule tissue compared to other tissues, vegetative tissue. And even in NFR5, we have seen it expressed in the root tissue and nodules tissue compared to other vegetative tissue. So now we are asking a question: what contribute to the ligand specificity within the ectodomain of NFR1? Because it contains extracellular domain. So this is the extracellular domain. And here are three lysine modules that I have explained before: lysine 1, lysine 2, and lysine 3. So we identify this lysine module from others plant along with the chickpea NFR1 and chickpea NFR5. And you see that this lysine module are distinctly variable. So it means that this variability is required for the perception of knot factor. So that therefore one particular rhizobia can able to infect only a particular host plant. So there it is very important to delineate that what is the proper mechanism that a particular rhizobia can able to infect only particular host. So there's a stringency of specificity. So this is a variable, but one of the important things is there, this lysine module basically complex by 2 alpha helix and 2 beta sheet. And all three attach together and form a clover leaf-like structure. And in between of both of them, there are some cysteine residues there. So first we make a model of this NFR1 and NFR5, and we have seen that based on the template of LI6, we have made this model, and we have seen that 3 domains are present in the ectodomain of NFR1 and NFR5, and we have studied then the lysine motif, and in between the lysine motif there is a cysteine residue, as I told, that connect all these lysine modules. So you see here this lysine 2 and lysine 3 motif, they are connected with this CXC motif and CXC. This is the cysteine rich module. They help them to connect lysine 2 and lysine 3, and this is lysine 3 to lysine 1. And these regions are highly conserved between all the NFR1 and NFR5 protein. But if you look at the between 3 and 1, there are some cysteine rich modules are there that can help to accommodate the disulfide bridge, that also highly conserved between the all NFR1 and NFR5. So that means this cysteine residues are highly conserved, but the architecture of lysine modules are different. That probably responsible for rhizobial specificity. So how they attach each other, you see the cysteine from one with another cysteine, they attach with a disulfide bond. There are 3 pairs of cysteine. So this way it can form a clover leaf like structure that can facilitate the function binding of knot factor within the NFR receptor. So there are some recent report, Bosugi 2020 Science Group, some Aarhus University, and Geisel in 2021 PNS in Cambridge University. So they did the crystal structure of ectodomain of NFR1 in lotus, and they did the crystal structure of NFR5 in Medicago. So it's just they make the only partial structure. They're unable to form the crystal structure because it is very difficult to form the crystal structure for this lysine module. But according to their basis, it seems like in Medicago, basically lysine 2 motif is responsible for binding of NFP. Based on the specificity, they bind with all of them, but the specificity is determined by lysine 2 motif, and LIK3 in Lotus japonicas, NFR1, that is the active receptor, their binding specificity is determined by lysine 1 modules. So based on that, what we did, as chickpea, the rhizobia name of the chickpea is the Mesorhizobium caesari, and the structure of Mesorhizobium caesari is not available now. So therefore, we use the Sinorhizobium knot factor and Rhizobium leguminous knot factor. This is the knot factor of Medicago and knot factor of Pisum sativum. We took them and we did make a docking analysis with our chickpea NFR1 and chickpea NFR5. And we have found that some compatible residue that are basically responsible for the binding. And therefore, we did the whole docking analysis. You can look at this docking analysis. You see this Medicago, this Sinorhizobium melati, their binding docking score is very high compared to others because this is the same species. And we have seen is the Rhizobium leguminosum. They are actual there's a rhizobia of the Pisum sativum. So this knot factor, their binding affinity is more compared to others. But in chickpea, you see, they can have the similar kind of efficiency of Medicago Sinorhizobium meloti. And also in NFR5, we have found some binding efficiency. And similarly, we have detected the residue that are probable interactor of the knot factor. So we want to delineate the mechanism if we by side-directed mutagenesis to figure out how the whole process is working. Now, the next question we are asking about the intracellular domain, that what contributes to the ATP binding specificity within the endodomain of NFR1. For that perspective, we studied the multiple alignment with the C-terminal or kinase domain of NFR1, and we have found some distinct difference and some homologous region also. Suppose NFR1 chickpea, we have compared with other chickpea in the legume NFR1, and we have seen that some of the regions, suppose glycine-rich loop, XK motif, DFG, that belongs to the catalysis loop of the ATP, binding of the ATP, and that facilitated the catalysis of gamma phosphate of ATP, that is highly conserved in most of the NFR1. But NFR5, we have found some distinct difference that these glycine-rich loop regions are mostly absent. And also there is another motif that is required for the activation loop that is also mostly missing here. And also in addition, we have found some of the regions are truncated in NFR5, in all NFR5, but that is exist in the LIK3 and facilitate the substrate binding. So now we have, based on that, we have made the structure of the kinase domain of NFR1 as well as NFR5. You see here that ATP binding site and this PPAQ motif that is present there that facilitated the binding of the ATP, but this G-root loop is absent in NFR5. Therefore, ATP unable to bind this region. And also you can see this PPAQ motif is basically facilitated the substrate binding, and that is also absent in NFR5. So suggests that for this existence of this domain, ATP binding site, as well as PPQ motif, facilitate the NFR1 for the ATP binding, and that means autophosphorylation, as well as they have the capacity to do transphosphorylation. But NFR5, due to lack of this ATP binding site, they are not able to bind ATP, as well as lack of this PPAQ motif, they are unable to bind any substrate. So it has no autophosphorylation activity as well as transphosphorylation activity. So it acts as a pseudokinase in chickpea. So we have docking with the ATP molecules with NFR1 and ATP with the NFR5, and we have identified some of the probable residue that is basically involved for the interaction with the NFR1 as well as interaction with the NFR5. But this interaction, you see, this is strike compact structure, but as ATP unable to bind NFR5, so this is a dispersed structure. That means they're not able to properly bind, and this hydrogen bond binding efficiency is significantly less. So it suggests that ATP is unable to bind NFR5, but it's strongly able to bind with NFR1 for promote the autophosphorylation. So we measure the some glide energy score after docking. You see here, NFR1, it's the score is 110. It is significantly high compared to others. We dock also Medicago, Lotus, PISM with ATP, but you see in chickpea, it is really strong, this ATP binding affinity. So that means the chickpea kinase domain distinctly different from others. So there are some other functional role maybe is there in the chickpea that is not exist in the model legumes like Medicago and Lotus and PISM also. But in NFR5, you see their glide energy score is very and docking score both are significantly less. So it suggests that ATP unable to bind NFR5. And we have detected the probable residue that form a hydrogen bond and make the stable structure with NFR1 with the knot factor. In addition, we did the MD simulation to see the stabilization of the structure, and we have detected the residue that probably responsible for the proper hydrogen bond and which have proper hydrogen occupancy. So now we are interested that what are the phosphorylation site, because when ATP bind with the NFR1, so therefore they probably precede the autophosphorylation. So we did the probable software. We have detected after ATP, this is a green color is the ATP, and it binds with the NFR1, and we have detected some probable phosphorylation site. So in the kinase, you see there are basically two lobes. One is N lobe, another is the C lobe. So N lobe basically involved in the active binding of the ATP, and C lobe basically facilitated the substrate binding. And we have detected the probable autophosphorylation site in NFR1. And one of the important residue is T476, but in NFR5 there is no autophosphorylation residue, but we have detected some transphosphorylation residue. As NFR1 and NFR5 interact each other, so therefore NFR1 able to phosphorylate the NFR5, and probably these are the transphosphorylation residue that is exist in the NFR5. So to delineate this structural thing in the biological level, to that what actually happened in the functionally. So we make some overexpression construct. We overexpress the chickpea NFR1 and NFR5 and also one of the mutant, and we have performed the hairy root transformation to see the nodulation phenotype. And after hairy root transformation, we have seen that after overexpression of NFR5, nodule number significantly increased in the root. And also you have seen the similar thing in case of NFR5. But whenever we mutated one of the phosphorylated residue, that is T476, that actually present in the catalysis domain, that is basically required for the ATP binding, as well as it is present in the activation loop that facilitated the substrate binding. So that regions, if you mutate, you see the nodule number significantly reduced in this overexpression of this mutant version. So now we have identified most of the residue. So now in the phosphomimic approach, we will do some phosphodeficient approaches, and phosphomimic approaches will overexpress, and also RNA approaches. We want to delineate the mechanism how NFR mediated signaling modulate the downstream signaling in chickpea. So this is the image, empty vector. You see there are nodules are there. This is small size, but NFR mutant, you see the nodules are basically large size and numbers are more. And NFR also nodules are bigger and large size. But in this NFR1 over the mutant line, you see nodule number drastically reduced. And if there are some nodules also there, these are very small nodules. So based on that, I conclude it here that several conserved and variable residue crucial for knot factor binding and phosphorylation remain in both NFR1 and NFR5. CA-NFR ectodomains show high conservation in the domain architecture with other model ectodomains. And NFR have strong binding efficacy towards their native. That's why the Sinorhizobium minority, we have seen their knot factor significantly bind in Medicago, whereas the Pisum sativum knot factor, they have shown significant binding with their receptors. So that means that specificity and binding specificity is basically species specific. So it's very important to delineate the mechanism that what kind of binding affinity in there in case of chickpea between knot factor and knot factor receptor. So CNFR is an active kinase, whereas CNFR is a pseudokinase, and it is not functional for catalysis. And we have some future plan, generate overexpression and mutual lines of NFR to understand their effect on nodulation efficiency by Hairy root transformation. In vitro binding assay, we have purified already NFR1 and NFR5 protein. So we are trying to do some in vitro binding assay to delineate the binding between the knot factors with the knot factor receptor. So we are trying to synthesize knot factor by in vivo and in vitro approaches. And site-directed mutagenesis study we are doing to delineate the in vitro phosphorylation assay to delineate the downstream mechanism and identify and characterize downstream interactor is one of the major approach to identify the downstream signaling cascade. So I'd like to thank our director, Professor Ganesh, and our head of the department, Ramesh Shanti, and also biology department. And also these are my lab students. And this work mostly done my postdoc, Bhagat Kumar Palaka. And these are other students. They're involved in other root nodules in biosis process. Some of them route stress project they're working now. And I'm really thankful to ISAT project, ISAT Tirupati for funding, as well as my Ramalinga Swamy re-entry fellowship and startup grant and MHRD star grant. And finally, thank you all of you for listening my talk. And I'd like to prefer to some question as well as some suggestion.
Yeah, Sarup, very nice and elaborated talk. It's a nice beginning, and I think first time I am also listening to your talk. Although I had read your plant cell paper long back and other papers also, but no, few more questions I can see in the chat box. So maybe since we are running sort of time, so maybe we will be discussing after the Hiten's talk. So just I think just one or two questions if you can answer in between. You can just just stop sharing so that another person will start. Meanwhile, we can take a question of Subhendra.
So I was just thinking, Namaskar. So the question is the evolutionary selection for these different rhizobium. Is it because the amount of nitrogen they can make, that is the host dependent? There is a binding of nod factor and the other the mechanism, but otherwise, what is the why not a rhizobium can fix nitrogen in many different hosts?
Because rhizobium need a place where they can survive. Because regular rhizobia, they are unable to fix the nitrogen. So they enter the plant and stay within a structure. It is called root nodules, and they differentiated in another form of bacteria. It is called bacterioid. And when they transform into bacterioid, there are some another genetic machinery that is turned on in this bacteria. And that is basically nitrogen fixing gene. And then they can able to fix nitrogen. So therefore, compared to free living form, it's able to fix more nitrogen in when they accommodate and stay inside the plant and within the nodules.
I think let's call Hiten now. Hiten, then Dr. Rai Chaudhary, please stay with us, because we do have a very good system in this particular meeting, and a previous meeting also, the panel discussion that I really loved that type of meeting. So Hitendra, you can start sharing. Hitendra is from CCMB Hyderabad, and he is going to discuss about the genomics assisted molecular mapping of rice loci associated with tolerance to biotic stresses. Ethan.
Yeah, so good morning to everyone. And today I will present a progress on our work on genomics assisted molecular mapping of rice loci associated with tolerance to biotic stresses. And this work was initiated by Dr. Sonti sir, and now I am taking them forward. And in this presentation, in initial few slides, I will speak about development of improved Samba Masuri rice. Then I'll show progress on further improvement in improved Samba Masuri rice that we are doing. And then I'll come to this topic. So you can see this picture. About 20 years ago, this variety improved Samba Masuri rice was developed by the team of Dr. Sonti and Dr. Sundaram sir. And this improved Samba Masuri rice is a bacterial blight disease resistant derivative of Samba Masuri. So let me briefly tell you the development of this variety. So Samba Masuri is a very popular rice variety which is grown in about 5 to 10% of total rice cultivation area, and it is very popular because of excellent grain and cooking quality, and it is a high yielding rice variety, but it is highly susceptible to a disease called bacterial blight. So this is a serious rice. Bacterial blight is a serious disease of rice. It is caused by this bacteria called Xanthomonas oryzae, Pathover oryzae. And when we inoculate rice leaf, we see with this bacteria, we see this yellow disease lesion. And in severe infection condition, the whole fields looks like dried out. So it could cause severe yield loss, and there are no effective chemicals to control this disease. So what Dr. Sonti and Sundaram's team did, through marker assisted selection, they introgressed these three bacterial blight resistance genes into Samba Masuri rice, which is which has fine quality and high yielding traits. And finally, we obtained a variety which we named as improved Samba Masuri. So it has all the good quality traits of Samba Masuri, and also it has these three bacterial blight resistant genes, and it is a bacterial blight resistant. As you can see in the this picture, Samba Masuri leaf is susceptible for bacterial blight, whereas improved Samba Masuri is resistant. And this variety was released in 2008. And as you can see, representative field picture. So this field has improved Samba Masuri rice, and it's a free from bacterial blight disease. And this adjacent field is Samba Masuri rice, and it is severely affected by bacterial blight disease. So this variety is now being cultivated in about 2 to 3 lakh hectares in several states of India. And in 2017, we tested this rice for glycemic index value. And National Institute of Nutrition, they did a clinical study, and they reported that improved Samba Masuri has low GI value. The GI value is 50.999. And so far, we have licensed this rice variety to 3 companies. So what next after improved Samba Masuri? Improved Samba Masuri is resistant for bacterial blight disease, but it is susceptible to many other diseases, pests, and also abiotic stresses. And we need to fortify these additional, to address these additional stresses, and also to increase its yield so that farmers can adopt this variety. So with this idea, now we are adding additional traits into improved Samba Masuri rice. So we are adding traits like resistance for blast disease, tolerance for brown plant hopper, and also tolerance for abiotic stresses like drought, lodging, submergence, salinity, and also higher yield. So one by one, we are adding these well characterized gene circuitals for these traits into improved Samba Masuri. So individual and also in combination will have these traits down the line in Samba Masuri rice, in improved Samba Masuri rice. And this we are doing in collaboration with Indian Institute of Rice Research. And first success in this effort is we have now improved Samba Masuri rice with salinity tolerance. So sorbitol QTL have been introgressed into improved Samba Masuri rice. As you can see here, this is a donor line for sorbitol QTL, and these are 4 introgressed lines in the ISM background. And in NaCl stress, we can see these lines perform better compared to improved Samba Masuri rice. And also they have a better root architecture compared to improved Samba Masuri rice. And this variety has already passed national field trials, and recently it has been approved for release for coastal salinity zones. So here I'll show representative pictures for high yielding traits. This panicle is for improved Samba Masuri. Here we have introgressed OSSPL14 gene, and you can see the higher number of grains. And here we have introduced OSSPL14 and SCM2. So here we can see higher number of grains as well as stronger gulp. And based on yield improvement, this variety is now, it has been promoted to advanced varietal trial 2, and we are hopeful that this variety also get released. So in this effort, I think many varieties will reach to farmer. So now we have seen that marker assisted selection works, but this we can use only if the variation or the trait of interest is available in the rice germplasm. So can we extend the scope of marker assisted selection and identify novel genes or QTLs important for agronomic traits that are not available in the rice germplasm through mutagenesis and genomics assisted breeding? With this idea, we did EMS mutagenesis in the Samba Masuri rice background, and we generated 10,000 mutant population. And in collaboration with Indian Institute of Rice Research, we screened this mutant population for important agronomic traits, including 3 biotic stress related traits, which I'll show you. So one of the biotic stress related trait was yellow stem borer. This is an insect pest of rice, and there are no known source of tolerance available in the rice germplasm for this insect. And in the screening, we obtained few mutant lines that show tolerance to yellow stem borer. The second biotic stress related trait that we screened was sheath blight, and again, in this case, there are very few rice lines that show moderate level of tolerance, and we obtain mutant lines that show a good level of tolerance. And bacterial blight is third biotic stress related traits that we screen, thinking that suppose down the line, if resistance in improved Samba Masuri breaks down, we may need a newer source of resistance. And again, here in this case, we obtain mutant lines that show a good level of tolerance to bacterial blight disease. And these are few additional agronomic traits for which we have obtained mutant lines like early maturation, stronger cull, and also for higher yield. So at the moment, our focus in these 3 biotic stress related traits, and what we are doing is we are trying to identify the causal mutations through NGS and bioinformatic analysis. And also we are trying to develop molecular markers so that we can use these traits, we can introgress these traits into other rice varieties. And also we are trying to characterize the mutations. We are trying to understand how the tolerance is happening in these mutants. So In order to identify the causal mutation, we are following delta SNP plot analysis or QTL-seq analysis. So how we are doing this? First, we are generating a F2 mapping population. So we cross a mutant line which has trait of interest with wild type. We get F1 plants. Then we self this F1 plant to get F2 mapping population. And here we screen the population for trait of interest. Then we identify the plants which show either wild type look-like plant or mutant look-like plant. And then we isolate genomic DNA individually from mutant look-like plant or also wild type look-like plant. And then we make a pool. And this pool is then used for NGS analysis. We subject for sequencing. And the sequence reads are then aligned with the wild type reference genome. We do SNP calling, and then we calculate SNP index. So in a particular position, if all the reads show SNP, then it is considered as SNP index as 1, because SNP index is calculated like number of SNPs divided by total number of reads. So all 7 reads has same SNP, so it will be SNP index 1. In this case, only 3 reads have SNP, so SNP index will be 3/7. So like that, then we get SNP index and we plot SNP index. And wherever we see a peak like this, that is considered as possible candidate reason responsible for the phenotype. So this is a mute map approach. For a similar way, we do SNP index plotting for wild type also, wild type bulk also. And in delta SNP index, what we do, we subtract wild type SNP bulk to mutant type bulk. And wherever we see SNP index this reaching to 1, that is considered as putative candidate reason responsible for the phenotype. So with this strategy, coming to yellow stem borer characterization of yellow stem borer tolerant mutant. So yellow stem borer is a serious insect pest of rice, and it causes dead heart symptom in vegetative stage, and also in reproductive stage it causes white ear symptoms. And here you can see the panicles are white in color and they are empty, actually they are not filled. So this is a specific pest of rice, and because of this insect pest, there could be yield loss between 10 to 50%. And as I mentioned earlier, there are no known source of tolerance available for this pest in rice germplasm. So in screening, we obtained this line called SM92 that shows high level of tolerance. This is a wild type Samba Masuri rice line. You can see dead heart symptom. And some SM92 is a mutant line that show high level of tolerance to YSB infestation. So then we use this line for QTL-seq analysis and further characterization. When we did QTL-seq analysis, we observed these 7 peaks or 7 locations, 7 regions that we predicted might be responsible for YSB tolerance. And then we further validated these regions using CASP assays. So in this assay, a fluorochrome labeled allele specific primers are used to identify the genotype of interest in the samples in question. So we designed about 50 markers covering these 7 regions, also some additional regions which are say from here or here, which are not associated with the tolerance. And then we did this genotyping. And what we observed, we could associate these 3 regions with the tolerance phenotype. And a total of 6 markers we could validate that are associated with YSB tolerance. And so these are the positions. And the percent association that show level of tolerance we could also assign to these markers. And these are the genes. And we are now thinking to further characterize these genes. Just 1 minute. Yeah, so now to further gain knowledge in the mechanism by which this line is showing tolerance, we have done RNA-seq analysis. And from this analysis, what we observed that secondary metabolism related pathways might be responsible for tolerance in this mutant line. And we have observed phenylpropanoid and lignin related pathways genes are upregulated. So these genes might be providing physical strengthening of cell wall, or either or it could be toxic effects of these secondary metabolites. Because of that SN92 line is tolerant to YSB. So to summarize, the yellow stem borer tolerant characterization of yellow stem borer tolerant line using bulk segregant analysis and next generation sequencing, we have identified 5 putative QTL regions, 7 putative QTL regions in the chromosome 1, 5, 7, 10, and 12. And out of these 7 QTL regions in these 5 chromosomes, regions in chromosome 1, 10, and 12, we found they are associated with YSB tolerance with varying levels of contributions. And we have developed 6 SNP markers. They were those that were found to be significantly associated with the trait. And from RNA sequencing analysis, the RNA sequencing analysis indicated that the tolerance could be because of phenylpropanoid pathway and secondary metabolism related pathway. So coming to sheath blight disease in rice. So sheath blight is a disease caused by Rhizoctonia solani, which is a necrotropic fungus. And the symptoms include grayish-green lesions, mostly on lower leaf sheaths and also occasionally on the leaves. And because of this disease, there could be yield losses ranging from 5 to 50%. And as I mentioned earlier, there are only a few reports of tolerance to sheath blight in rice germplasm that show moderate level of tolerance, like there's a variety called Tetup. And we have obtained a mutant line, this phenotype you can see here for SB6 plant, and that show good level of tolerance. So we did molecular mapping of in this gene, in this mutant line, as well as in another tolerant line SB87. And in both of them, we have identified 2 QTL intervals each for these 2 lines. For SB6 in chromosome 1 and chromosome 7, for SB87 in chromosome 1 and chromosome 4. And what was interesting is in both of them, there is this a region in chromosome 1, which are like adjacent. So we are hopeful that this reason might be interesting for or important for tolerance to sheath blight disease. And we are further developing marker, and also the characterization through RNA-seq is ongoing for sheath blight tolerant lines. So to summarize sheath blight part, 2 rice lines, SB6 and SB87, with significant tolerance to sheath blight was identified. And we have used these 2 lines for detailed characterization. And using QTL-seq approach, we could identify 2 candidate intervals each in these 2 lines. and marker validation and RNA-seq analysis is ongoing for sheath blight. So coming to third biotic stress related trait, that is bacterial blight disease. And we have identified mutant lines for bacterial blight. So as you all know, that Xanthomonas oryzae, Pathware oryzae is a causal agent of bacterial blight, which is a rapidly evolving rice pathogen. And we know that exostrains have evolved to break down existing major resistance genes like XA21. So this is one of the major resistance gene for bacterial blight. And in our screen, we have identified mutant lines that show resistance against the exostrain that breaks down XA21. And one particular mutant line that I'll show data is BB42. So this line, as well as the other lines, can serve as potential source of additional resistance genes to further improve rice varieties. So this line BB42, it shows resistance against IXO 1221. This strain is able to break down XA21 resistance genes. And you can see the disease lesion, it's a severely virulence deficient. But then we try to map the mutation in this line through classical mapping approach, which I mentioned earlier. We could not identify the candidate region responsible for the phenotype. And we are trying to figure it out this. We used two different populations, but then also we could not reach to the target region. And now we have further characterizing this mutant line BB42, and based on phenotype, as you can see here, the BB42 show reduced growth compared to wild type. So then we hypothesize that there might be the expression of defense genes or a constitutive active defense might be there in this line. And with this hypothesis, we check for expression of key defense genes, and we observe that in BB42, the key defense genes like PR1, PR4, PR10, they are induced. Also, we perform our RNA-seq analysis, and here again, we confirm that the key defense genes are upregulated in BB42 compared to Samba-Masuri line. So this further strengthens the previous observation that an active basal defense gene in BB42 is present. So to summarize the characterization of bacterial blight tolerant line, BB42 was identified to be a promising mutant line to study bacterial blight tolerance. And conventional mapping could not identify responsible locus for BB tolerance in BB42 mutant line. And the study revealed an enhanced basal immunity in the rice line BB42, which could be the reason for its tolerance to Xanthomonas oryzae, Pathware oryzae. And RNA-seq analysis of BB42 further confirmed the presence of active basal defense gene in this line. So to conclude the overall work, we have identified pre-breeding mutant rice lines with enhanced tolerance to an insect pest called yellow stem borer, a fungal disease called sheath blight, and a bacterial disease called bacterial blight. Yeah. So, and then we have identified QTL intervals for YSB tolerance in this line SM92, and 6 linked SNP markers we have developed. And also we have identified QTL intervals that that are showing possible association with sheath blight tolerance in 2 sheath blight tolerant lines that are SB6 and SB87, and marker validation and RNA-seq analysis is ongoing. Conventional mapping approach could not help to identify the locus responsible for BB tolerance in BB42 mutant line, and we are trying to figure it out this. And also we have done RNA-seq analysis for SM92 and BB42 lines to know the possible mechanism of tolerance in these two lines. Yeah, so thank you. And I would like to acknowledge Dr. Ramesh Sonti, who initiated this work and also guiding whenever necessary. And the characterization of all three biotic stress-related traits are done by Gokulan. He will present tomorrow some other work that he is doing. And Kamal is also helping in characterization of sheath blight tolerance mutant. Zamaluddin and his team is helping in generating mapping population for molecular mapping. And I also thank Dr. Raju, Kranti and other lab members for support. And we have a very good collaboration with the Indian Institute of Rice Research, particularly Dr. Sundaram Group, who is helping in development of these marker-assisted selection lines. And Dr. Seshu Group helped in screening of all these mutant lines, and all other team members are involved in field related activities. And I would like to thank directors of CCMB and Indian Institute of Rice Research, and we have funding from CSIR and ICR. Yeah, thank you.
Thank you, Hiten. I always appreciate and love to hear your talk. Every time, last talk you talked about the pyramiding of the different bacterial blight genes and other things. It's really great, and it's very, very applied. So now what I can do is that I can invite all the speakers of this session, and I think Dr. Subra has already joined, and other also can join, and people can ask question. Say
so like, while Praveen, while others are joining, maybe I can start discussion.
Yeah. So any question for Dr. Subra? If anybody has, you can ask question.
Otherwise, I can possibly ask Hitendra, actually.
Yeah.
So,
So basically, panel discussion is for that only, that which we can interact. Even the speakers of the session can interact with each other.
Yeah. So Hitendra, very nice talk. So first of all, I missed out very important two talks. Actually, I had another meeting I was attending, so I missed Ranjan and Swarup's talk. I'm sure I'll be able to catch up with them later. But therefore, right now, for both of you, I do not have any questions because I did not hear. Now, for Hitendra, it's very nice talk. So, two or three things. One is that, Hitendra, you are focusing on three important rice disease. But, although we know that there is a blast resistance being developed by IARI long back, that I do not see, but that is breaking down. That's what I understand. So that PI gene that has been put into the background of the Pusa means basmati background. So whether the parent that you are using to actually put all these three other disease, does that parent have already the blast resistance into it? If not, then why are you leaving that disease? That is one thing, because it's a very important disease, and breaking is reported now from India. Now, this is one. Second is that you have a lot of nice data from your QTL-seq, and also you have a very nice collaboration with Sundaram's group at IIAR. So what I was thinking, and you are sitting in CCMB, right? So yes. I'm putting this question, means not question, actually, I'm just suggesting you that, all these, whether it is the different disease or it is saltol or any other thing, QTLs are known, but fine mapping and actually getting the gene would open up a very new direction in the mechanism. And since you have already done the QTL-seq and you need little fine mapping, and in that case, why don't you have some other students to work on from that angle? Maybe not for all disease, but whichever is your top priority. That not only give the mechanistic detail, but also you can possibly come up with some targeted genic marker, which can also be helpful in the, the agriculture. So these are two things that I was thinking that I would discuss with you.
Okay. So can I?
Please answer.
Yeah. So regarding blast, so already multiple genes or QTLs, I think all known for blast disease. So that's why then we thought maybe not to screen for blast disease, our mutant population.
No, what I was thinking, Hitendra, there are so many traits that you are focusing. Now the magic kind of a breeding is available. What I was thinking is that when you are trying to stack all of them, if you try to use a magic kind of a breeding approach, you can have one of the parent, which is a high yielding mega variety of rice, having the blast resistance. And then you can introgress these all the QTLs. So it's not that from the beginning you do, but maybe at the end you can actually try to introgress.
Yeah, so that is also possible. But then since improved Samba Masuri is our base material, because it is a low GI rice, okay?
Right.
And it is a fine grain rice variety, high yielding rice variety. So we thought maybe all these traits we include one by one in improved Samba Masuri so that farmers can use. So that's why we have used in improved Samba Masuri. We haven't taken any other rice variety to.
So what is the situation of Samba Masuri in the south or middle of this India? Whether it is having effect of the blast or it is already a blast tolerant?
No. So blast resistance now we have added in our program. It is not resistance to blast.
Okay, fine.
Yeah, so we are also adding blast resistance related genes.
Fine. And about going to the gene and doing
right. No, that is
basic science will be really interesting. I'm sure students sitting at CCMB will enjoy that kind of work. Will understand the mechanism much better.
I completely agree with you. Yeah, that we are in doing now.
Yeah, nice talk, Hitendra.
Yeah, first I will read a few of the questions because they have already written in the chat box. So Anu Singh I think it's for hello sir, it's in addition to the knot factor, rhizobial surfaces also have polysaccharides like EPS and LPS. Do these have any active role in the promoting bacteria infection and nodulation? Swaroop, I think is for you. Yeah, you have to unmute. You have to unmute, Swaroop.
I want to say this is a nice question, but several studies already established the theory that knot factor is the predominant molecule compared to other molecules. Maybe there's other molecules like exopolysaccharide, EPS, they have some role, but their role act as an accessory molecule. Without presence of knot factor, they are not able to induce the NFR receptors, or maybe not able to induce the downstream signaling cascade. So therefore, without these molecules maybe act as accessory molecules, not the major molecules, that I can say.
And second question is that how plant differentiate between beneficial and pathogenic rhizobium strain during signaling by Shraddha Dahali?
Yeah, that's a really nice question actually, but how plant will differentiate. So this is a very difficult question to answer, because as I have shown, there are multiple these lysine receptors are there, right? So there is only two receptors, NFR1 and NFR5. They are involved for nodulation, but some of the function already established. Suppose one of the receptor I'm talking about in Arabidopsis, CERK. So that receptor is highly homologous with the NFR1. So in the leguminous plant, this CERK is not there, but there is NFR1 is there. So recent one of the study that is published in the Science, they established one of the things that I have shown the three modules, lysine 1, lysine 2, and lysine 3. So they have shown in the lysine 1 region, so symbiosis knot factor binds. In lysine 1 region, this is determined some of the specific residue, but there are some other residue that is responsible for binding of chitin. after perception of fungal. So this is a competition for binding of chitin molecule and NOT factor. If NOT factor bind properly, then it promotes the nodulation. But if there's a chitin molecules are there, binds with that particular receptor, they will promote the plant immunity. But even there are some other things also in the downstream. When the rhizobia invade inside the plant, within the nodules, there are some NCR peptide released from the plant. And this NCR peptide is one kind of specific peptide. It has antimicrobial activity. They only inhibit all pathogenic bacteria. But this is some amazing mechanism that need to be established in future. This NCR peptide only allow the rhizobia to enter. But they are not inhibiting, they are showing antibacterial activity against other pathogens.
Yeah, Dr. Sinjyoti from NIPJ, she is working on such aspect. It's very nice.
It's very nice.
The other question is by Jawahar Singh to you. Is that out of CANFR1 and CANFR5, which is most likely to be transphosphorylated by the downstream signaling molecules, and which one is one NFR1 or NFR5 is going to have a serious impact on early signaling?
Yeah. So NFR1, NFR5, as I told, that when this NOT factor signaling initiate, it require one major receptor or typical receptor, another is atypical receptor or pseudokinase. So here NFR1 is the major receptor and NFR5 is the co-receptor. So both are required. So both have the extracellular lysine module, and lysine and both extracellular domain of NFR1 and NFR5 also interact. That we have also shown. And when NOT factor bind with that region, there's a clover-leaf-like structure of lysine of NFR1 and NFR5 when the NOT factor bind. And then what actually happened here, NFR1, due to its kinase activity, it phosphorylates the downstream region, and that is autophosphorylate its kinase domain, and therefore it can transphosphorylates the CNFR. So here CNFR actually acts as a scaffold protein. But this is also necessary. Still, now a lot of research is going on. So one of the recent report they shown that first probably NFR1 activates, and thereby they interact with NFR1, and then NFR1 able to transphosphorylate NFR5, and thereby downstream other can interact. So this is them different groups. They're establishing different hypotheses, but still it is enigmatic.
And any since you have worked in chickpea and anything which you can correlate with the Medicago truncatula and the Lotus japonicus, which has been already some part of this type of things has already been. So any correlation that you have got, or chickpea is totally different?
The chickpea is not totally different, because I have shown my research results that there are a lot of homologous regions are there in terms of extracellular domain and in terms of kinase domain. But there is a variation in the extracellular domain that is determined the lysine module. Probably that is responsible for the binding of NOT factors. So supposing the chickpea there, rhizobia is the Mesorhizobium loti. But in Lotus japonicus, this is Mesorhizobium, chickpea is a Mesorhizobium ciceri, and Lotus, it is a Mesorhizobium loti. And Medicago, it is Caenorhizobium meloti. Yeah. So this is a direction is there based on the lysine, but their kinase domain is highly homologous. But we are trying to identify the interacting partner, and we have found that some of the other receptor-like kinases can interact with them. But the molecular mechanism, how it is going on, it need to be delineated in future.
Subhadra, you wanted to ask some question to Hitin?
Yes. Namaskar, Hitanji.
Namaskar. Yeah, I saw your question.
This resistance, the resistance is recessive if we cross with the sensitive one or the original one. If you take the mutant, and what is the phenotype?
Yeah, so far the lines which we have studied, they show kind of recessive phenotype. We don't know for because we have multiple lines and we have studied only one or two for all of these traits.
And no intermediate phenotype, codominance.
No. So in case of bacterial blight, we see 3:1 ratio. Okay. And for all for sheath blight and YSB, we see intermediate phenotype.
Okay.
Amit want to ask some question?
Yeah.
Yeah. So actually, I have questions for both Hitendra and Saru. So I will start then with Hitendra. He is just talking. So Hiten, so it's very nice talk. So I have a questions and also one information for you. So with respect to Dr. Shubhra Chakraborty's, I mean, like suggestions for the blast. So in West Bengal, 3 districts, we have tried ISM cultivations last year, Kharif season.
Okay.
And so these are all Gangetic plain areas, and we have not observed any blast disease.
Okay.
So this is for your informations. I think I have communicated, but this is the things. And
But
for beer,
what about bacterial blight in
yeah,
other
so
variety?
yeah, so basically these 3 districts are known to have a lot of rice cultivations. We have not observed any serious disease for the ISM or the batch you have shared with us. So, and the yield was very good, and it was so good that the farmers they kept few seeds for this season.
Okay, good.
Done. And another thing I would like to say that you have mentioned already 3 companies. So this is sometimes happens what scientists feel and the market demands. So they're actually thinking it is for the diabetic. They are not considering it from the higher yield perspective.
Right.
So market propositions has to be like that. It is a very higher yield and disease resistance from the farmer's perspective.
Okay.
But from the consumer's perspective, then it will be a diabetic things.
Yes.
So marketing of the seeds would be for the farmer,
Okay.
but the marketing of the grain would be for the consumers.
Yes.
So because as a scientist and the R&D, we really don't discriminate these two. Right. For you, the product means is the
Amit,
seed that is
actually,
for farmer.
yeah, so we are targeting both way, and at the moment focus is for grain side as a low GI rice. We are actually in discussion with Adani Wilmer also.
Great.
If they
So
can
they
take,
are in the food part, basically, not
yeah.
on the seed part.
Yes.
So these companies whom you have licensed, so basically their products is the grain, not the seed for farmers, right?
So no, so 2 of them was for seed business earlier. Okay, and one recently is for grain for as a low GI rice.
Okay, great. So and so that part is over. Another point is you have mentioned a new ISM with the salinity tolerance.
Yes.
We are looking for rice varieties for the mangrove areas in Sundarban areas.
Okay.
So one thing is that because Samba Masuri is a Kharif crop, and in the Kharif season there would be regular cyclonic storms in Sundarban.
Okay.
So, and it is associated with the movement of the saline water to the mangrove forests. For few days it will be there. So do you recommend to do a trial of that particular variety ISM with salinity tolerance that you have released for the coastal part, also for the mangrove areas?
I think, yeah, it can be tried. So what we can do, we can send some seeds and then you can try there.
So have you tried it already in some other mangrove areas?
No, no.
Hiten, you have a strong culm mutant also.
Yes.
That
So
maybe you can send it. That you can send it.
No, Gopal, can you just tell which mutant?
No.
A strong club
Samba
mutant.
Masuri itself, we have this stronger culm trait also.
So that the stem will not lodge during
Okay,
flooding.
so this is a
So that will
lodging
be very
tolerant
good.
variety.
Yes. So
So
besides
this
this,
is
he is
a
working on
line
that
of the
also.
original
What
ISM.
we are doing, we are making combination of these traits. So a combination that has salinity tolerance and also stronger culm, then that can go to your area what you are asking.
Okay, so that would be really great. And one last point to add it. So when you are incorporating or changing the multiple traits, I think 2 important traits should not be changed. that is the maturity date. That is for, I mean, very example, because even if you you know that for QTL and other things, whenever we change one particular agronomic trait, it affects the other traits in some ways. So enhancing the lodging tolerance should not reduce the yield, and enhancing the yield should not increase the maturity time.
Right, I agree with you. So this will
Right. So
by doing
yeah.
backcrossing.
Yeah, so when we are giving it to farmers, and when we are telling, say, these one will have higher yield, the first thing they will ask that whether the maturity time would be same or it will be 1 month more.
But Amit, I think maturity time early may be better.
No, Gopal, so maturity time, ideally, we do not want to change because this is associated in the crop calendar.
No, but actually,
I mean,
if you
from the
can mature
science point of
early, you
view,
can have more crops per field actually. Per
no,
Gopalji, it depends on farmer preference. Some area they prefer early, some area they prefer even longer
Yeah,
duration.
that's what I'm saying. That even early can be advantageous.
No, I know what Gopal is telling. It is from science perspective, but from the field perspective, the best way not to disturb the cropping cycles. Even if a change in 7 days early or late, it may be good for the science, but not for the farmers. So what Hiten mentioned, that is right. In say Chhattisgarh, you will find they are actually looking for 7 days early. Right. In some cases, it would be 7 days late. But on an average, we would like to stick to the same duration. If it's 120 days seed to seed, it should be like that.
Okay.
I mean, always there are sub-segments. So that is with the Hiten. And so can I ask the next questions to
Yeah. Sarup, you ask quick questions so that
one moment. Meeting has started in other computer. So just thing to Hitendra or anybody who is hearing one point. So actually, you must be knowing that you are doing a lot of CASP and other genotyping work. So with all difficulty, NGGF is now in place. So from March, possibly it will start giving thing for everybody in the country. So any future thing, come to NGGF, and Gopal, Senthil, all are there. Everybody can tell you about it. So this is not for Hitendra, but for anybody in this audience who would like to use this facility, most welcome. So with that, I will take a leave, please. If anybody else have any other question for me, I can answer and then leave for the other meeting.
Any question is for Dr. Subra? I think I can't see anybody, raising hand.
Fine.
So,
fine.
Yeah.
So I will just take.
Swaroop wants to ask. Swaroop wants to
I want to ask that what kind of facility we can get it from NCGS, if you just briefly say. What kind of facility we can get from NCGS, that is NGS, that facility in NIPJ that you are talking. You are talking about NCGS.
He is talking about the facility of NGGF.
So facility NGGF has genotype, also it has CAS, next gen sequencing. So anything genotyping or next gen sequencing anybody wants to do, most welcome. Okay, Swaroop, and anyway, I know that you are already talking to Sanjuti, so Sanjuti can give you the exact update. Otherwise, the person concerned who would be doing is Dr. Sabbata Bhatia and also Dr. Swaroop Parida and Dr. Ashish Ranjan. All of them are taking care. In this gathering, Gopal and Senthil knows about it.
Okay.
Yeah. Next, I think let's move to the discussion part. So I think Senthil wants to ask some question to somebody, and Amit also having one question to Swaroop, I think. So Amit, you can start first to Dr. Swaroop.
Okay, yeah, thanks. So, hi, Dr. Swaroop, and very nice because I think I know you for a long time, and for the rest, you as my junior to my university days, and we're just looking maybe after 20 years or so, because during his Bose Institute, I heard about him, but I have not met. So that's great. So one very quick thing, because as you know that I'm totally in the applied aspect right now. So I was wondering for the nodulation part, So how will you differentiate the efficacy of the nodulations based on the soil type and based on the seasonal variations from the field perspective? So the efficacy of the nodulations based on the seasonal variations and based on the soil variations.
Soil variation is one of the major determinant of root nodulation because the soil pH is one of the important marker. So that's why I'm telling that the chickpea that is going in the North India, there, I don't know, some of region they produce a lot of nodules. But here in the South India, some of the places, they are not able to produce a lot of nodules. The soil pH probably activates some of the, specifically the iron and some other things. Maybe they form some kind of compound that inhibit the perception of knot factor by knot factor receptor.
So Swarup, maybe there are different rhizobial strains, like Sanjoti had isolated few of them. So maybe you can try using that and see whether they can nodulate there or not.
Yeah. Absolutely.
So that is one thing. Another thing, maybe beside pH, the microbiome, rhizobiome structures may also be depicting the success of your rhizobial strain. So it's not like that the strain may not give you nodule. I think due to some reason they are not surviving, colonizing the plant. That's why they are not giving you. So the efficiency of the colonization. So associate, it's important.
The microbiome also. And in addition, you are talking about the seasonal thing. Definitely the seasonal variation that, you know, that water content in the soil and the dryness, the drought is one of the important marker that we try to delineate in future, because in the presence of drought, the AVA signaling on. And this AVA signaling component, they inhibit the
after lunch session and also for tomorrow. So I'll catch up, but I'm going to another meeting. Maybe I will miss 1 or 2 lecture again in the evening. So all the best.
Santhil, you can ask question.
No, so actually I was following up with Gopal's point about the, the strain variations and the soil variations does matter, but actually it's difficult to conclude that in Tirupati you don't get, nodules. So for sure, as Gopal suggested, Sanjiti has a lot of collections, so maybe you can try one. Maybe there is some suitability that it is not actually surviving in that environment and you are not able to get it.
Yeah, because until I heard Sanjoti also initially telling that initially she had difficulty in nodulation. Then when she started getting newer strains, they are efficient colonizer and giving good nodules to her. So maybe you have to find out your own rhizobia strain there. That
no, actually, artificially we are getting nodules. Supposing that we are using soil right. So what we are doing, we are using the soil right and we are autoclaving, and then we are washing with multiple time water, checking the pH of the soil right, and then
that's what I'm saying. That maybe the naturally occurring rhizobial strain in those soils may help you.
Yeah, and even that's also on point. And also, I think the nitrogen amount in the soil also may be one of the determinant. Because if nitrogen amount is more, then probably plant has some negative regulators are on. They are not able to access the rhizobia. That is also another point. So if any soil in the nitrogen amount of more, specifically for chickpea, I have noticed that it inhibits the nodulation.
That is very much correct. And also age of the plant. I think after flowering, no nodules are formed. It all happens before flowering and it probably stops after once the flowering is initiated. Even when the transition of basic vegetative to reproductive phase starts physiologically, not even a visible flowering comes, then the nodulation stops for chickpea.
Dr. Prasoon Mukherjee is
I don't know really, but will it be cultivar specific also? Like, is there any report of cultivar specificity?
He's asking that whether is any cultivar specific things are there or in the case of rhizobium.
So we are now working on the groundnut. We are checking of different varieties of groundnut from here, and we have found some differences. Because some
from the what I suppose that he's asking about the chickpea cultivar itself.
No, chickpea cultivar we have used only 2 cultivar yet, and we have seen the more or less similar kind of things. Maybe in future we'll try to use some other cultivar.
What he is mentioning that may be correct also. Like sometimes it happens, like the different chickpea cultivars do have a different, so that you can think over it. So Prabhu also wanted to ask. I think he has started opening his, and Subhadeep also he wanted to ask some question. Subhadeep, you can go ahead. Subhadeep, yeah. Subhadeep, you can ask question.
Swarup, the knot factor interaction and other, it is a bimodal interaction. So both bacteria and plants are exchanging a lot of signals. So I heard like in rhizobium also quorum sensing plays a very important role. In this thing. So do you have any idea like about in the plant aspect, like whether interkingdom signaling occurs in legume and
okay.
Yeah, there is a difference in the determinant and indeterminate also, the type of colonization.
Actually, and some nodules are determinate, some nodules are indeterminate based on their meristematic activity. But in case of you are asking about the quorum sensing, so I'm not aware of properly that how the rhizobia quorum sensing work. But I know that when rhizobia infect the plant, they first form a microcolony. And this microcolony, when they form the microcolony, then there's a knot factor. This interaction are two types. On the first time when rhizobia is outside, they release this knot factor, then they bind. And then they form a, they invaginate the membrane and they enter the membrane and they form a microcolony first. And then inside this infection threads, again this knot factor signaling on. Again, there microcolony releases some knot factor and they make a invagination in the plasma membrane, and therefore they form a infection threads and then gradually they move. So I don't know about this the quorum sensing work here, but they form a colony and then they
for triggering the colony formation from a planktonic to that stage, there may be some endogenous bacterial signaling working and interaction with the plant.
Absolutely. I think the first, as I shown my this one of the slide, there are multiple different.
So I was thinking in that angle because you told that you are getting difficulties in establishing nodules. So maybe they're looking at data at which stage the defect is, like going through the bacterial process, like in vitro, if you can check at with the cut nodule or whatever the system you have, is it possible to check at what stage? Because if there is some bacterial factor that you can exogenously supply in the soil.
Yes.
I think it's a good point that he has, that this is how, this symposium is very, very important. I think it's a great thing that Dr. Sonti has given to us so that we can interact with each other and we can, exchange our knowledge other than other symposium where people are asking questions and just like that. You get lots of suggestions from this particular interactive meeting. It's great.
Praveen, very interestingly in agrobacterium, the TI plasmid transfer and lot of other processes are also influenced by quorum sensing because it has a strong AHL.
So agrobacterium, I think a few papers has been published also recently. Recently I could see some of the papers. So any other question to any other?
Subhadipda, in future, definitely I contact you.
Please.
This point about the quorum sensing of rhizobia. That we can able to think about something more about that.
Yeah, that's what I was saying.
Yeah, it's a great time. We can.
I have one.
Yeah, Dr. Prasanna Mukherjee wants to ask one question.
Yeah, I mean, based on the knowledge base, do you have like way to genetically engineer Rajobacteria to improve the host range? Like based on whatever existing knowledge we have, the understanding. So where are we now, basically?
You are asking to whom? Sarup.
Sorry.
So
Okay.
yeah, so actually there are bacterial perspective, there are several genes involved in the knot factor formation as well as LPS and exopolysaccharide formation. So people have tried several mutant of this bacteria and they have seen their role. But if you are thinking about engineering with the bacteria to increase the nodule number, maybe we can increase some of the knot factor production and that can facilitate it. But I think the important part is not only the bacterial side, basically the plant side is most of the important. Because after
no, I'm talking about improving. I mean, increasing the host range. Like from peanut will infect chickpea, something like that.
So you are talking about that peanut rhizobia can infect the chickpea.
Yes. Can we engineer? Are we in a position to do that with our existing knowledge?
So that's why we are trying to do some analysis.
So, Prasanna, there is a very interesting work which now people have started, and some people like one lady called a song in I think in Seattle or some. I'm forgetting the university, but she has been working for rhizobium interaction for a long time. So they are looking for mutants, rhizobium mutants, which are hypernodulating or which are little less responsive, means constitutively responsive to like not the chemicals which are released from the plant. So so that people are looking for mutants or variants which have a broad host range. Well, this is a good aspect. So finding natural isolates. First, the best thing is to find some natural isolates which has a broad host range. And for side by side, people are doing forward genetics approach, like large scale EMS mutagenesis or transposon mutagenesis, to look for mutants which can be used for broad host range infection as well as hypernodulation phenotype that can benefit the interaction between the bacterium and the host. And also they are also looking for mutants which are now less dependent on the plant phenolics, which are the compounds which are released, meaning to say that constitutively not factor producing mutants.
Correct. So I think Gopalji wanted to ask some question.
No, sir, I'm okay.
Is okay. And Prabhu, once you opened your video, so I think you also have some question.
Yes, please. Yeah.
Yeah. You want to ask some question?
No.
Okay.
So maybe, sir, maybe I can add on a little bit. Actually, I have been discussing with Sanjyoti because we work on bacteria and we love bacterial genetics. So I've been always encouraging her, why not she start working on bacterial factors for symbiosis. So I don't know, maybe Swarup can correct me. She feels that there are many efforts and till now people have not really come up to major master regulators from bacterial side. So that's why she was afraid of touching the bacteria.
Yeah, that's the point. There is a if you increase the amount of knot factor, how plant perceived by receptor and how the signaling occur.
No, so don't touch knot factor, no. So let's say touch codon sensing, touch something EPS, touch something so that the survivability of the rhizobium increases in the soil.
But
So those factors I was more interested because it's really a very good factors which can promote symbiosis.
So then we need to figure out, because basically, as I told, that lipopolysaccharide and other the exopolysaccharide or EPS, mostly they are accessory molecule. Not factor is the major molecule. Till now, it is irrelevant. Maybe in future.
Because not factor is helping during plant colonization. What the first thing is, the bacteria has to establish in the soil. If it is not able to establish in the soil, then obviously no matter what it does in plants, it's not going to help. So I think the first people have not really looked the bacterial ability to survive in the soil.
Subhendra, you have something to say? Yeah, you are mute.
Sorry, I have. So one thing that nitrogen fixation in happening in the root, where the nitrogen content is lesser than the aerial part. If we see the exposure to shoot, so why nitrogen fixation is not occurring in the other part of the plants?
No, nitrogen fixation is occurring basically root, and then it transported to whole plants. That's why if there's a nodules, you will get plants are relatively healthy compared to the non-nodulated plant.
Yeah, those nodules we don't see information in the stem. We don't
there are
see.
some, that is not formed, but some of the example is there. Some bradyrhizobia, they from one of the Sesbania rostrata, they from nodules in stem. This is one example. But basically, you are right. It's only they form in the roots, because there is a root hair region that only secrete, that only probably contain those kind of receptor there. Because the receptor that is actually required for this perception, you see that I have some expression data that basically this receptor expressed in the root region and within nodules, not in the other regions. That may be one of the reason. And in the stem, there is one of the receptors are there. It is control the auto regulation of nodulation. That is different scenario. So suppose plant produce some nodules and they realize this is enough. Then some peptide from the root, they transport it to the leaf and they activated some signaling and they send some microRNA based signaling to tell root, okay, you have to stop, no nodulation now. It is called auto regulation of nodulation. So some receptors still there in the root, in the stem, but they perceive some peptides and act and they just control the nodulation. But you are right, there is we never see any nodule formation in other parts of the plant. Only it is possible on the root.
Very nice.
Yeah, I think we have already exceeded 1 hour and 10 minutes. This is the beauty of MIPAM. So now the people who know about MIPAM, we continue up to now 9 o'clock sometimes, 10 o'clock in the night. So this is the beauty of MIPAM. So, but last question I have to ask to Hitendra. So Hitendra, have you got any non-synonymous type of the mutation in any of the gene of those 3 QTLs that you have pinpointed in your QTL set analysis?
Oh, we have few of them. We are going in more detail to study them.
Okay. Senthil, you want to conclude or it's okay?
No, I'm fine, sir. I was just floating on. It's already time. We can discuss some.
You can.
I was just thinking.
Last comment. Yeah.
There is a. It's been known for a long time, but even if you see APS journal also, there is a new journal for phytobiome, wherein you try to study the dynamics of this population. It's not just segregated as a pathogen alone or rhizobium alone or some other thing alone. So that field is actually kind of emerging, and there is a new journal also they have started by American Phytopathology Society. So I think the group now we have included rhizobium people also. Earlier we used to be only the pathogen guys. Now I think we should
beneficial pathogens also, or beneficial bacteria or microbes. Yeah. Thank you very much, all of you, and I thank all the speakers. of this session. And thank you very much. We will be, I think, what is the time of next session?
I think 4:30.
Starting maybe from 5 or so.
5, okay.
Just I will check.
5 नहीं है शुभदीप भाई, 5 नहीं है, शायद 4:30 कुछ है.
4, so let's meet again at 4:30. Thank you very much. No, really nice.
Can I ask just one question?
Yeah, please.
Yeah. So in IPG, Gopalji and Senthil, I want to ask one question. So that is it possible to generate transgenic chickpea in IPG?
Yeah. Is there? There is a, already we do have the in IPG there is.
But only problem will be, Swaroop, that actually we have the facility, so we can generate it. But there is IBSC biosafety rules are there, na? So basically, you cannot get the seed. So you may have to your IBSC approval, NIPGR IBSC approval, and RCGM approval to take the seed back. Otherwise, transgenic is easy at NIPGR.
So is it possible I can send some construct and you can generate the plant there, and then whenever the plants are ready, then we can go for the IBSC meeting from here and NIPGR?
No. Before that only you need approval.
Before sending the construct. Okay.
Yeah.
Okay.
Because the rule is like that. We cannot touch any construct unless you have the approval.
Okay. Thank you.
But Swaroop, we can collaborate also with some people, that where the transgenic facilities already established in their own lab, the individual labs, so that you can collaborate and they individually you can work on.
But still he needs.
Definitely. Anything that he want to touch, in this regard, that he has to be,
because we are suffering. Like we made some transgenic which we have to ship it to CCMB. So that's the approval.
Yeah. Thank you all.
Thank you.
Okay, thank you all. Okay, thank you.
क्या भाई प्रभु?
हाँ शुभदीप भाई, और बताइए कैसा आप? बहुत अच्छा गया सेशन.
हाँ, सब सारे सेशन बहुत अच्छा जा रहा है.
That all plenary talks are really great, ना? बहुत अच्छा.
सब बहुत अच्छा हो रहा है सब.
और आप भी पूरा अभी कल पूरा पैक दे रहे हैं.
कल तो पैक्ड है, थोड़ा बीच में आऊंगा मैं, क्योंकि अब कल तो पूरा स्टूडेंट्स का ही है, तो
हम लोगों को नहीं मिलेगा पैक कुछ मिल रहा है सर?
नहीं पैक्ड वाला हम सोच रहे थे कि जोमेटो में टाइप करके सर वो करना मुश्किल हो जाएगा.
मैं शुभदीप बोल रहा था एटलीस्ट स्पीकर को तो कुछ स्विगी से ऑर्डर करके.
सर हम लोग भरपाई कर देंगे नेक्स्ट टाइम.
विद इंटरेस्ट वी विल इंटरेस्ट के साथ हो जाएगा, है ना?
जितेन प्लान करेगा नेक्स्ट MIPM.
आप रियली you as a host would have been wonderful actually. बढ़िया रहता है.
मैं तो पूरा प्लान कर लिया
था. अभी
हम लोग का बाहर का टूर एंड देन CCMB में गेस्ट हाउस वगैरह, फिर CDFD का.
नहीं, आप लोगों ने बहुत मेहनत किया था. हितेन, यू, सुवेंद्र, पर्टिकुलर आप दोनों ने काफी बैकग्राउंड वर्क कर लिया था. वैसे आई थिंक थिंग्स नॉर्मल कुछ हो सकता था लगता है.
नहीं अभी भी many places है. Like अभी right now I am holding a position in JNU administrative position. So yesterday one the thing has come for the symposium. So it was in hybrid mode. So then the rule says that you can't right now. So because if somebody is coming from outside India, then you can't. So it's very difficult. तो वो तो आप नहीं अभी कर सकते हो. मे बी लेटर बिकॉज बाहर से आने वाले के लिए वो चाहिए होता है ना, वो वीजा क्लियरेंस के लिए वो चाहिए होता है, वो सम ऑफ द थिंग्स दैट यू विल नॉट गेट राइट नाउ. तो प्रॉब्लम है मतलब अभी भी, बट आई थिंक इट विल बी ओके आफ्टर सम टाइम्स, मुझे तो ये लगता है। और मुझे ये लगता है कि ये एक हम लोगों ने ये मीटिंग एक करा लिया ना, अभी एक ऑनलाइन टाइप का मीटिंग करा लिया, हम लोग जल्दी ही करा सकते हैं एक ऑफलाइन टाइप का मीटिंग, अगर इफ एवरीथिंग इज
वही हम सोच रहे हैं
कि एक ऑफलाइन मीटिंग,
मई जून में
वो काफी अच्छा टाइम है, बट मई जून में आप देख लीजिएगा हैदराबाद में बारिश बहुत होती है।
नहीं, मई जून में नहीं, मई जून में हम थोड़ा बात करके, विंटर में अच्छा होता है।
प्रभु कैन आल्सो थिंक ओवर इट, वहां पे
विंटर में अच्छा रहता है।
समर में चंडीगढ़ वगैरह अच्छा है, मतलब काफी बढ़िया होता है।
समर टाइम इज,
हाँ, तो दैट इज आल्सो गुड प्लेस।
अभी आप थोड़ा मंडी में भी यू कैन प्लान सम लोकल ट्रिप आल्सो। वहां पे भी एक वन गाय आई थिंक सुरेंद्र कोलैबोरेटर है, वो वर्क फ्रॉम
मंडी में, वो ये क्या नाम है,
IIT मंडी,
IIT मंडी में है वो।
तो
IIT मंडी में कर लेते हैं, IIT मंडी अगर तैयार हो तो वहां कर लेते हैं, दैट विल बी ग्रेट फॉर ऑल ऑफ अस, है ना सुभदीप?
हाँ मंडी
अच्छा है, ये इनसे बात करते हैं, रुकिए, मतलब ही इज अ वेरी नाइस पर्सन।
नाम मैं ढूंढ रहा हूँ, मैंने मेल किया था एक बार उसको, तो कुछ मेटाबिलोमिक्स करता है।
हाँ, बहुत बहुत बढ़िया, मतलब लास्ट टाइम ही केम टू NIPJR for the MIPAM last वाला में, तो he has delivered a very good lecture.
Subhendra knows him.
I also know him.
तो बात करके थोड़ा देखिए आप लोग अगर उधर हो जाए तो।
नहीं, बहुत इंटरेस्टिंग काम कर रहे हैं एक्चुअली,
बहुत इंटरेस्टिंग।
वो भी वहीं से, उड़ीसा से लगता है ना?
नहीं, this is from I think Andhra।
हाँ रहे, ओके। चलिए, वो भी कनेक्ट हो जाता है ओडिशा से कहीं कॉर्नर से दोनों स्टेट कहीं पे जुड़ जाते हैं वैसे।
ओके,
चलो फिर मिलेंगे फिर इवनिंग में 4:30, राइट? 4:30 बजे।
हाँ, 4:30 बजे, या राइट। ओके, थैंक यू। बाय डॉक्टर प्रवीण और हितेन, बाय
बाय।
हाँ, आज 14th डे है। रेखा आर यू ऑनलाइन? आई थिंक शी कैन नॉट हियर, आई थिंक। तो अभी आज 14th डे है, कुछ डेटा कुछ विरलेस डेटा का कुछ पता चला?
मंडे विल सी प्रभु।
मंडे, हाँ मंडे हो जाएगा ना। हाँ, क्योंकि वो आप लोग वो डेटा ऐड कर सकते हैं, देन वी कैन गो अहेड। हाँ, और बाकी सब ठीक है? यू आर इन आई थिंक ऑफिस?
हाँ राइट।
हाँ, ठीक है, नाइस। तो ठीक है, देन विल टॉक टुमारो।
Good afternoon, Alok.
Yes,
do you want
yes.
to try? Do you want to try your slide? Meanwhile, others can join.
Okay, okay, yeah.
Just see everything is working. Will you?
Yes. Yeah. My voice is coming.
Yeah, yeah.
Okay, fine.
Hello, Alok.
Hello. Yes, yes, sir.
Yes, yes, sir. Hello, Alok. Good
yeah.
afternoon.
Good
Yeah.
afternoon.
Good afternoon.
Good.
Gopalji, slides are coming now.
Yeah, yeah. Slides are coming. Just try to make it full screen.
Slides are coming. Just try to make it full screen.
So, Gopal, you are sharing now?
Yes, yes. But I think some echo is coming. Subdeep bhai, some echo is coming from somewhere.
Okay, I will tell this CDFD host to.
Gopal,
Echo maybe from your end, Gopal, because Subhadip's voice was not echoing.
No, no, now.
Yeah.
Now it's okay.
No, no, now nothing coming.
Yeah,
So some other problem. I
Some other
hopefully
problem.
sorted out. Yeah.
Right, right.
So, Professor Pandey.
Yes, sir.
Professor,
Hello.
Professor Pandey, make it full screen.
Yeah, yeah. Full screen. Yeah.
And ये वाला
गोपाल जी कहाँ वो YouTube और Zoom दोनों ओपन तो नहीं किया ना?
नहीं मेरे तरफ से एको नहीं हो रहा है प्रभु।
अच्छा मुझे भी कोई एको नहीं लग रहा है।
अब ठीक हो गया, अब ठीक हो गया, कहीं पे कुछ था वो ठीक हो गया।
अब आ रहा ठीक है।
नहीं शुभदीप को बहुत ऑप्शंस दे दिया ना, YouTube, फेसबुक, Zoom।
YouTube में पूरा लाइव रिकॉर्डिंग देख सकते हो।
हाँ, आपने पूरा रिकॉर्डिंग भी लगा, वो रिकॉर्डिंग भी आ रहा है, बहुत अच्छा अरेंजमेंट।
So Subhadeep, how long that recording will stay in YouTube? Forever or?
Forever. CDFD channel,
Okay.
This will be in the CDFD YouTube channel.
Okay.
CDFD DBT YouTube channel.
Shubhadeep, from CDFD website, can you go to that YouTube channel?
Yes, yes.
Oh, actually, I looked at, but I did not find the link. Maybe I will try.
Oh, you from the CDFD website, you can go to Facebook.
Okay.
From the Facebook, you can open the YouTube.
Subdeep bhai, actually, I just tried MIPAM. I wrote MIPAM in YouTube, and then it link came up, so it's visible.
Okay.
It's coming. It's coming. Alok, your slide is still not visible yet.
Still not visible?
No.
अरे आलोक उसको पहले खोल लो, वो जो स्लाइड है ना।
ओके ओके।
हाँ बस यहाँ तक दिखता है, फुल स्क्रीन में नहीं दिख रहा है।
फुल स्क्रीन में वो जो एंड, वो जो शो बटन में क्लिक करो।
नहीं मैं अभी कर रहा हूँ जस्ट
आलोक कुछ और प्रॉब्लम, और एक बार प्रॉब्लम हो गया ना, क्या ऐप यूज कर रहे हो या ब्राउजर से कर रहे हो?
नहीं ब्राउजर से कर रहा हूँ, ब्राउजर से।
ओ ब्राउजर से प्रॉब्लम आता है, तो ऐसा कर लो तुम, वो जो पीछे लेफ्ट साइड में जो वो है ना, वो क्या बोलते हैं उसको यार,
स्लाइड को इंडिविजुअली क्लिक करना,
स्लाइड को इनविजिबल कर दो, क्लोज कर दो, और राइट साइड वाले को भी क्लोज कर दो, फिर बड़ा दिखेगा। ठीक है?
अच्छा अच्छा अच्छा, राइट राइट राइट। अब ठीक है? अब ठीक है?
हाँ ठीक है, अभी चलेगा।
हाँ ठीक।
नहीं तो आलोक तुम ऐसा करो, जो स्लाइड शो कर रहे हो ना, कंट्रोल F करो। कंट्रोल F5।
कंट्रोल F5
हुआ कुछ बड़ा?
ऐसा हो गया थोड़ा सा।
हाँ बड़ा कर दो, खींच दो। अच्छा
ठीक है यार, चलेगा।
स्लाइड्स भेज सकते हो उनके टीम को CDFD में।
नहीं तो यार तुम
हो जाएगा,
बंद
हो
करके
जाएगा।
फिर दोबारा शेयर करो, बंद करके दोबारा शेयर करो।
ओके ओके, ठीक है।
Even PDF also there is an option. You can present in PDF also.
हाँ, PDF also you can.
Usually, if you share your entire screen, that works better.
Yeah, that's better.
Instead of just the selected window.
Yes.
आलोक कहाँ से दे रहे हो प्रेजेंटेशन? सीमैप से या कानपुर से, लखनऊ?
नहीं, कानपुर से, कानपुर से।
ओके
ओके, कानपुर
वहाँ से निकल ले एक्चुअली, मतलब अभी सीमैप में खत्म हो गया ना।
राइट, ये ऐसे में खत्म।
आशीष दिव्यांशी भी ज्वाइन कर लिया है?
हाँ, सिविल भी ज्वाइन।
ओके, तो सुखदीप भाई, shall we start?
Yeah, start, no problem. पहले उसको लोड तो कर लो, कर दो।
आलोक फिर से
नहीं, कुछ प्रॉब्लम हो रहा है दैट।
तुम स्लाइड मेल करो भाई, मैं चला।
नहीं तो यार वो जस्ट उसको विंडो बड़ा करके दे चालू कर दो।
अभी आ रहा है?
आ जाएगा। हाँ, परफेक्ट।
आ गया, full आ गया।
Perfect. It's working now. Okay. So good afternoon, everyone. I hope we had a nice morning session. So in this session, we are going to have 4 talks. The first one is Alok Pandey. He was working at CMAP and recently moved to Kanpur University. So over to Alok.
Yeah. Thank you, Dr. Gopalji. And first of all, I would like to thank organizing committee of MIPAM 2022 to give me opportunity for the presentation of my research work in this renowned seminar series. And as he already introduced me, Dr. Gopalji, earlier I was inspired faculty at CSIR CMAP. Right now I moved to Department of Biotechnology. That was formerly known as Kanpur University, and right now its name is Chhatrapati Sahuji Maharaj University at Kanpur. So today I will talk about the role of an orphan response regulator, that's called the BEMR, and that governs multiple virulence functions of Xanthomonas oryzae pathorae. Yeah. So earlier I was at CSIR CMAP and I was involved in several projects, and I have worked on like a downy mildew disease of opium poppy, the Papaver somniferum, and it's a source of many medicinal compounds. And also I have worked on an endophyte isolated from several medicinal plants that can enhance the secondary metabolite and that can also protect the medicinal plant, particularly opium poppy plant, against a very devastating disease of this plant that's called the downy mildew, which is caused by a oomycete pathogen by the Carnospora mechanopsides. But today, in today's talk, I have taken the research part from my inspired faculty ground, and the focus was on the Xanthomonas oryzae pathorae rice infection. So let's begin the talk. The subject of this talk. So bacterial cell is equivalent to an information processing device, and the ability to make decisions at the cellular level is critical for survival and long-term proliferation. But how cells accomplish this feat without the luxury of nervous system or brain? So the answer lies with a diverse and important set of molecules found inside all cells called signal transduction proteins. And in bacteria, 2-component signal transduction protein represent the major family of proteins regulating sense response machinery. Right? So 2-component signal transduction systems are one of the most prevalent ways by which bacteria sense, respond, and adapt to various environmental changes. It has 2 components. One is called the histidine sensor kinase that's located into inner membrane, and response regulator is localized to cytoplasm. So after receiving an environmental stimulus, the one histidine conserved histidine residue in sensor kinase gets phosphorylated, which is then transferred to response regulator, and where it will be transferred to an aspartate conserved spotted residue. And after phosphorylation, now by means of there will be output domain. So they will affect the output domain, and by means of these output domain, they will affect or regulate or modulate the downstream factor that facilitate the adaptation and disease development. And output domain can be divided into can be classified into 4 groups, like by having these properties like DNA binding properties, or RNA binding properties, or protein binding properties, or having domain that participate in protein-protein interaction, or having the enzymatic activity, like they regulate the cyclic di-GMP intercellular concentration, particularly in Xanthomonas oryzae, or in the Xanthomonas genus in general. So that's why this is the way they regulate the adaptation and help in disease development. So what was my project? That was directed towards understanding this following fundamental questions, like how does 2-component system help in bacterial pathogenesis, and how they make a response that makes the bacterial plant pathogen to survive and prosper in host or ex planta condition. And for this, I took this pathosystem, rice and Xanthomonas oryzae, Pathoraoryzae, in short XO we can call it. That's the causal agent of bacterial blight of rice. So after analysis, bioinformatic analysis, we found that this XO is equipped with large repertoire of 2-component system. You can see, I mean, it's a very intelligent kind of bacteria, because it has number of sensor kinase and response regulators. And the more number of 2-component system partners are there, it indicates that IQ level of that bacteria is very high. And also, in other sense, you can say that they can survive in many different kind of environmental condition, and also inside their host, right? So here you can see that I have given the data for like Korean strain, Japanese strain, and Philippines strain. And there is that's called one is the BXO1, that's an Indian strain, and it has the 38 sensor kinase and 53 response regulator. And I targeted to mutagenize all the response regulator, like 53. Okay, so a systemic mutagenesis of all response regulator was performed to establish their role in exopathogenesis on rice. And this how I got the mutant for all the 53 genes, like by using the insertion inequation by using the suicide vector, and the mutation was achieved in such a way that there will be no polar effect on the downstream gene. And then I started we started, in fact, the phenotypic analysis of each mutant descripted that can code for a response regulator gene. And today I will focus on one gene, that's a VEMR, and this gene is very important as for pathogenesis, as we found that this you can see the lesion lengths. The rice leaf inoculated with this mutant is severely virulence deficient, and after complementation like VEMR XO plus, they regain, that strain regain the ability to cause the disease on rice. So on the data has been given for collected on the 7 days after infection, 10 days after infection, and 14 days infection, and it was severely virulence deficient, this mutant, and that's why we were very interested in taking for the detailed characterization of this mutant and what is the role of this gene in the pathogenesis, exopathogenesis. And also we performed the epiphytic inoculation, and we found that there will be no leaf actually. We found nothing, no infection when we infected the rice leaves with VEMR mutant, but 70% and 69% respectively, these rice leaves were infected when inoculated with wild type or the complemented VEMR mutant. So these two kind of pathogenicity tests establish that this gene is very much required for the pathogenesis, and this gene is also essential for the in-planta growth and in-planta migration. In-planta growth was performed by looking at the the in-plantar growth kinetics of the mutant and wild type and the complemented mutant. And we found that the WEMR mutant doesn't grow like a wild type. I mean, it doesn't achieve the maximum saturation point in plantar. And also in such a way also we found that they were also deficient for the in-plantar migration, right? Now I come to the another part that this WEMR, which is encoded by this gene. Actually, here I have written the gene number. It's a WEMR actually. So this is also is required for the induction of hypersensitive response on the non-host plant. I took two plant non-host. One is tomato here, and another is the tobacco, where the data that will come in the later slides. And also we found that this mutant causes less electrolyte leakage. We suggest that the hypersensitive reaction is very less in the case of the tomato leaves, which is infiltrated with a mutant. So next is this also we use this non-host tobacco plant, Nicotiana benthamiana, and here you can see the HR associated HR in the spot where we infiltrated the wild type, complemented mutant, but we observed little chlorosis when we infiltrated the tobacco leaf with WEMR exomutant. And there is also we used buffer and just a control strain, that is a type 3 secretion system mutant strain. We found no HR associated with this, right? And then we also looked at the growth of exo strains in the non-host tobacco plant, and you can see the wild type. Wild type is a 0 time point. We used wild type WEMR mutant and complemented WEMR mutant and the type 3 SS mutant. At the 0 point, around 10⁻⁷ cells were infiltrated. And after 1 day and 2 days, and at the 3-day time point, we calculated the cell number at the infiltration site, and we found that wild type become 0 actually because of the HR which is happening, and the complemented WEMR mutant behaves like a wild type. But what happened in case of mutant, that it decreased, but it is not completely dead actually. That is happening because of HR reaction. So they survive somehow. The WEMR mutant, they survive inside the non-host tobacco plant. Whereas you can see the type 3 sequence system mutant, its number is absolutely not going on. Somehow they are there, and I think I should play it out like this is a very interesting thing actually. The cell CFU value for the type 3 secretion system is not going down. So they are surviving there, but I mean they are not showing any HR kind of reaction. Okay, and also we found that we detect the hydrogen peroxide in the Nicotiana benthamiana leaf using the DAG staining method, and we found that the wild type is producing the wild type and complemented WEMR. These two strains are producing the large amount of hydrogen peroxide, whereas that WEMR mutant is deficient in the, I mean, is failed to induce the accumulation of hydrogen peroxide, and which is associated with HR. Generally, commonly it is wherever HR is there, and the hydrogen peroxide production of hydrogen peroxide perform a very important role. So reduced HR, reduced electrolyte leakage, reduced hydrogen peroxide production, reduced expression of HR marker gene also we took for this assay, and reduced the rate of bacterial cell death in the leaves of non-host plants infiltrated with mutants, suggesting that the response regulator encoded by WEMR exogene is required for optimal expression of HARP, hypersensitive response, and pathogenicity genes, operon. That's a HARP operon, and that's very important for the encoding component of type 3 secretion system. And as all of you know that the type 3 secretion system is implied by pathogenic bacteria to inject a number of effector protein directly into the plant cytosol in order to suppress the plant defense response that helps the colonization of plant pathogen. So whether this BEMR exo mutant is now has become the inducer of plant defense response or not, we tested. We performed the next experiment. So, and also before going into those data, whether that is inducer of plant defense, we also we checked the expression of harcoperon in this background. We found that the certain genes, that is a certain genes that encoding component of type 3 secretion system is required for the optimal expression of HRP or the operon, the harcoperon, right? Like we tested the expression of the HRCC gene and from HRCT, HRCU, and HRCR, and HRPE and HRPF, respectively from the ABCDEF operons, right? And then we infiltrated this mutant into the into the that rice leaf at the very young rice leaf. And we found that in those leaves, wherever infiltrated with BEMR mutant, we found the deposition of callose. And what is callose? Callose is a 1,3-beta glucan polymer, and its deposition in leaf cells is induced by bacterial pathogen. And what is the role of that? It forms a physical barrier to prevent ingress of pathogens. And enhanced callose deposition is considered as an induction of defense response. So, and you can see there is no callose deposition in the leaves infiltrated with either wild type or complemented BEMR mutant. So increased callose deposits, as well as also we performed the QRT, the RT-qPCR that assisted observation of upregulation of defenses such as PR4, PR10, and WRP13 and FLS2 in rice leaves infiltrated with BEMR mutant, suggests that BEMR encoded response mutation is essential for the suppression of induction of rice defense response. And that's why we use this thing, whether now they are actually enhancing the defense response of rice, whether they will provide the protection to rice plant against subsequent exo infection. We performed this assay, and we found that the pre-treated leaf with pre-treated rice leaf with a BEMR mutant display the enhanced resistance against subsequent exo infection. So you can see that these red arrows represent the site of pre-treatment, either with the first, that's a wild type, and or with BEMR mutant. And when the set of these sets of rice leaves were infected with wild type after 24 hours, then we found that the leaf which were pre-treated with only wild type, you can see the lesion there. They cannot stop the disease, but in case of rice leaves which were pre-treated with BEMR, they protect the leaf, and there is a very less lesion length you can observe, and that sort of lesion only you observe in around like 40% of the leaf. And here, and this the black lines, the vertical black lines is representing the visible lesion in the rice leaf, right? And one interesting thing also I observed, and that is you can see here this thing what happened actually. This thing I observed, and this is the site of inoculation after pre-treatment. This is site of inoculation for wild type, and here I saw a kind of a HR type of reaction. So this is very interesting, and what is going on here? Still, I'm looking for that. Okay. And then also so that was the previous slide. I will go back into the previous slide. So here I would like to say that this strain can be utilized as a biocontrol agent, and also for the future work I will also look for whether this mutant can protect the rice plant to biotrophic or necrotrophic pathogens. so or other pathogen, and what would be the mechanism, whether they are following the standard method like whether salicylic acid or jasmonic acid ethylene pathway, both are, which pathway is working here. So accordingly, we can elucidate the mechanism in WEMR mediated protection of rice plant. So that would be that I will plan in the future what is going on here. And this mutant also display reduced swarming mortality, that you can see in swarming assay. So mortality is reduced for this mutant. And also we observed, we just infiltrated with, we tagged this mutant with the GFP, and also with wild type with the GFP, plasmid encoding GFP. And then we performed the biofilm assay using the confocal, and we found that wild type is making the normal biofilm, where WEMR mutant was deficient in the forming the normal biofilm. Okay, and this and also this mutant is deficient in the production of the EPS. EPS is also one of the very important virulence factor. So they are this one single that regulator is regulating multiple virulence functions. So I think in addition to the type 3 sequencing system, they are deficient in several virulence functions. That's why they have become the very potent inducer of rice defense response. Okay. So to summarize this thing, so this WEMR XO response regulator is required for virulence and hypersensitive response on non-host plant, and they are also required for separation of host rice immunity. And this feature is that's why the WEMR XO mutant is a biocontrol agent, and they are also required for the EPS production, motility, and biofilm formation. So altogether, it seems that the signal transaction governed by WEMR XO response regulator control expression of XO factors required in multiple virulence functions. So I would like to thank the team who they have worked for this project, the Parul, Kritika, and Rusali, and I would like to also acknowledge Dr. Ramesh V. Sonti for sharing the strain, the original strain, Dr. Prabhu Patil for sharing all the exogenome information when it was not in public, and Dr. Gopalji Jha for giving me several research related materials which was very much required for this project. So I am very thankful to all of them, and Inspire Faculty Grant for this work, and CSIR CMAF for hosting me as an Inspire faculty. Thank you.
Thank you, Alok, for such a nice, clear, very clear presentation, and also timely presentation. So I'm very happy. So we can have few quick questions for Alok, and then rest we'll keep it during panel discussion.
Gopal, maybe can I ask one?
Yeah, please go ahead.
No, actually, this is very interesting topic to me, so I'm interested to interact. So basically, I will reserve a lot of my points to the discussion section, but quickly. So there are 2 points you said in your presentation. One is that it is responsible for hypersensitive response. At the same time, it dampens the innate immunity. Am I understanding correct, or?
I mean, the mutant is inducer of defense response because it is a deficient in producing the type 3 sequence system efficiently. So that's why they have become the inducer of that rice defense response. So that is one thing. So that's why the rice plant is now showing the enhanced resistance against exo infection. It means I think the PTI is on. So,
okay, no, I was just facing some because I was thinking like a typical defense response is HR, while the dampening of other responses need not happen. So it cannot act in both ways. One that enables disease, another one that provokes the defense response. Anyway, so that clarification. The second thing I wanted to ask is about the the plant targets where you be interested in some plant targets because you established a nice system. Now, if you are able to, knock out certain plants and then look for same Benthamina, same tomato that you have used, and then look for, you can actually find a typical pathway that operates, that provokes the HR. Because it's a non-host plant, you'll be able to come up with that. Have you done any of those assays?
No, I haven't done.
So you entirely focused on the bacterial side.
Yes, bacterial side. Yeah.
Okay.
But I am interested in finding what is going on actually. What kind of plant pathway is targeted by this pathway?
Yes,
so,
that is very easy to do, especially in Benthamina and tomato, whichever the host one, non-host one. But yeah, it's a very good typical type 2 non-host resistance pathway. That is more robust because HR is one of the typical responses that can almost kill the entire pathogen and cause complete resistance if you can exploit that pathway. I think I will reserve rest of my discussion to the panel time.
Yes. Thank you.
Alok?
Yes, sir.
I have a question. Prabhu here. Very nice talk, Alok.
Thank you.
So this is regarding your gene expression studies.
Yeah.
So they were carried out in planta or using some in vitro, like using
which gene expression study? Like one type 3?
The type 3, right?
A type 3, that's from the bacterial side. Bacterial side that grown in the GOM2 medium. Yeah.
GOM2. Okay.
Yeah.
So okay,
not from the in-planta, but from the GOM2 medium. It's similar to plant environment.
So how about this rice mimicking media?
Rice?
Is it that there is another media, no? This rice mimicking media, where they use rice.
I mean, GOM2 is equivalent. GOM2 is equivalent to that one. Yeah.
Not much difference.
So the expression of these operons were observed only after growing cells in the GOM2 medium.
Yeah. Okay. Yeah. Fine.
So maybe we'll take one more question and then we'll shift it to next talk. So Rahul is interested to know how BAM-R is modulating T3SS, any direct binding to the T3SS operon.
So it might be somehow, there will be unknown regulator in between, and either they are affecting directly promoter of those genes. That is still not known how they are affecting the type 3 16 system, but might be directly affecting or indirectly by using some kind of mediator.
So Alok, there are some known T3SS regulators actually well characterized. So maybe you can first look at the expression of those regulators in your protein background. So maybe we'll discuss in the panel discussion. So let's thank Alok and move on. So next speaker is Ashish Ranjan. So over to you, Ashish.
Thank you, Gopalji. Can you see my screen?
Yes. Make it full screen. Yeah, perfect.
Good afternoon, everyone. First of all, I would like to thank MIPM organizing committee for inviting me to give a talk. And today I'm going to talk about soybean sclerotinia stem rot interaction. So one of the important pathosystem my lab is interested is to understand the molecular interaction of the host plant, soybean, and the necrotrophic fungal pathogen called Sclerotinia sclerotium. So these Sclerotinia sclerotium, they make this black melanized structure called a sclerotia. Here I'm showing you on a PDA plate. When we grow this Sclerotinia, they make this sclerotia, but in natural condition, they are found in the soil. This structure can survive in the soil for more than a decade, making it difficult to control.
Ashish, 1 minute. Prabhu, I think your mic is unmuted. Please mute it. Some background noise is coming, I think. Maybe CDFD post or... Yeah. Ashish, go ahead, please.
Okay, thank you. Yeah, so these melanized structure called as sclerotia can survive in soil for more than a decade, making controlling this pathogen with conventional control strategies like crop rotation is difficult, because you don't want the growers or farmers to grow their plants after 10 years for better control strategies. And this pathogen has a broad spectrum of hosts, which includes more than 400 different species of plants, mostly dicots, and which includes many economically important. So under favorable condition, this sclerotia grows and make this mushroom-like structure called apothecia. And these apothecia, each of them can have millions of spores, and under favorable condition, they can fall on the plant flower tissues and then grow and then infect the plant. So unfortunately, we do not have any of these hostages which are completely resistant against this sclerotinia stem rot. Here, I'm just showing you a global distribution of this sclerotinia stem rot, and you can see several of these red dots all over the world map, except Antarctica. You will find that every part of the world has reported this sclerotinia stem rot. And according to an estimate in US and Canada, like in 2017, sclerotinia stem rot of soybean led to estimated loss of 1.2 billion just in one host crop. If you add other crop plants, maybe that will be multi-billion losses because of sclerotinia stem rot. So one of the important way to control plant diseases is to breed resistant line. And for breeding resistant line, it's important to understand the resistance mechanism of plant and also the virulence strategy of pathogen. So keeping that in mind, at University of Wisconsin-Madison, when I was a postdoc, I started this work, and there I screened a number of lines for resistance where we can see some enhanced resistance against sclerotinia sclerosum. So here I'm showing you two panels of plant. The above one is susceptible interaction, where we challenge this sclerotinia grown on media plate directly on the soybean plant, and resistant interaction. So what we see here is that within 96 hours post-infection, the lesion length in case of susceptible variety is much bigger compared to the resistant interaction. And if we follow for a few more days, what we are finding after 7 days of infection, the susceptible.
जब वो लाइव चलेगा तो ये था क्या मॉलिक्यूलर इंटीग्रेशन प्लांट एसोसिएटेड माइक्रो हेलो
susceptible.
ये तो क्वेश्चन है
So there is some kind of noise.
एक बार हम एक चीज चेक करना चाहते हैं टी ब्रेक बहुत लंबा है
आलोक तुम साउंड बंद करो
ओके
So after 7 days of infection, we can see in susceptible variety, the plant is dead, but in case of resistant interaction, we see a restricted growth of this pathogen. So using these two recombinant inbreed line, we ask this question that what are soybean genes and metabolites involved in resistance response to sclerotinia sclerosum infection? So we used transcriptomics, which you can use to understand how the expression of RNA changes, and also metabolomics to understand what are the different metabolites involved in this interaction. Our ultimate goal was to understand the molecular mechanism which governs resistance to a sclerotinia sclerotium in soybean. I'm not going to tell you about transcriptomics or metabolomics, but I'm going to show you what were our key findings from the analysis of transcriptomics and metabolomics. And our study suggested that in case of resistance response, reactive oxygen species scavenging and antioxidant processes play important role. Why they are playing important role? Because these pathogens are necrotroph and their aim is to kill the plant. And one of the strategy the pathogen uses is to increase the expression of this reactive oxygen species. So the resistant variety of the plant, what that does is they increases the scavenging capacity of these ROS and antioxidant processes so that increased ROS do not kill the plant cells. So this is different than what we are hearing about biotrophic interaction. The other key finding was that we found that jasmonic acid derivative, jasmonic acid isoleucine, which were early induced in case of resistance response compared to susceptible response, and which might be important for early defense signal construction. The third key finding, which I'm going to talk in detail in today's talk, is a reprogramming of phenylpropanoid pathway leading to increased antifungal activity. And I will give you a glimpse about what phenylpropanoid pathway is and how that can act as an antifungal pathway. So the phenylpropanoid pathway starts with aromatic amino acid, phenylalanine, which is acted upon by different enzymes to convert into different molecule. And one of the important constituent of plant is lignin. So L-phenylalanine get converted into lignin. As you know, lignin is very important for defense of the plant. At the same time, they are deposited in the secondary wall of cell wall. Don't worry about these all names. We are not going to discuss these names, but just I wanted you to have an overview of what this L-phenylalanine can lead to. The other pathway which also originate from Coumarel OA is anthocyanin and phytolexins. So whatever you are seeing in the red are upregulated in resistance response compared to susceptible response, and whatever you are seeing in blue is downregulated in resistance response compared to susceptible response. And from that, it's clear that anthocyanin and phytolexins are upregulated in resistance response compared to susceptible response. And we see some of these metabolites which are upregulated in resistance response compared to susceptible response. Here I'm showing you metabolite profile of few of those compounds. And here in the x-axis, what you are seeing in blue bar is susceptible interaction, but what you are seeing in the orange bar is resistance response. And these are different time point of interaction. So 48-hour post inoculation, these are the constant like peak height of metabolite, and the y-axis is showing you the peak height. And we see that in case of resistance response, the precursor molecule phenylalanine is highly upregulated in resistance response compared to susceptible response. We also found the same thing with one of the molecule called benzoic acid, other metabolite caffeic acid and ferulic acid to be upregulated like this orange bar in case of resistance response. So this gave us clue that phenylpropanoid pathway, especially these metabolites, are important for resistance response. So our next step was to just check if few of these metabolites behaves like antimicrobial compound. So we did a plate inhibition assay where this is a solvent molecule, DMSO. Excuse me. And then these are the different concentration of these three different metabolites. ferulic acid, benzoic acid, and cinnamic acid. And you can see that as there is more concentration of this compound, they are inhibiting the growth of sclerotinia sclerosum. You can see decreased hyphae at different concentration of these antimicrobial. So it suggested that this might be important for survival of plant, like resistance response. So in my lab, a doctoral student, Nick Talmlo, he is working on this different important gene to understand the role of the genes and how manipulating these genes can impact resistance response in plant. So we have identified few of important genes which are highly induced, and currently we are doing weeks to understand their importance during this pathogenesis. And now I would like to switch gear to another observation which I shown you before, that in case of resistance response, we are finding that the pathogen is not able to grow in the resistant variety, and we see some red coloration in this stem. So here I'm showing you two plants, one plant, the same variety of plant, highly resistant plant. One is mock inoculated, another one is after 10 days of pathogen inoculation. And our hypothesis was after this observation that maybe there is some molecule which is antimicrobial, and maybe that is giving its coloration. And we know that some of the gene in lignin production and anthocyanin productions are involved in this kind of coloration. So we went ahead and did ethanol extraction of this induced plant and non-induced plant. So they are same variety, only thing here the induction of defense response has happened. So we extracted these plant extract, and obvious thing was to challenge the sclerotinia sclerosum with this extract. And so here DMSO is controlled, the solvent, but you can see in when these sclerotinia were challenged with green stem, there was good growth of sclerotinia with green stem extract. But in case of red stem extract challenge, there was reduced amount of sclerotinia sclerosum, which told us that this may be because of having antimicrobial activity of this extract. So next thing we wanted to know if this kind of defense response is specific against fungus, or it's like plant sees anything and it start using all its defense mechanism to take care of the disease. So we used omics method, which is called chemical genomics profiling. So I will give you a little glimpse about what is chemical genomic profiling. So this chemical genomic profiling uses a library of yeast, Saccharomyces cerevisiae, where each gene has been mutated with a kanamycin cassette, and each gene has a 20 base pair barcode, which can tell you that if this gene has been mutated. And so this library of Saccharomyces cerevisiae is treated with the control and your metabolite of interest. Once you treat them, you grow for some time, extract DNA from them, amplify this mutant code, which will indirectly tells you what strains are sensitive against this metabolite and what are resistant. So this was done with the collaboration with Jeff Petrosky. And what we came out with this chemical genomics analysis, that the strain, so here in x-axis you see the fold chain versus control, like DMSO control, and here there is just a p-value of how much significant is the result. So whatever you are seeing in the blue color are those which are sensitive strain, and whatever you are seeing in the yellow color are resistant strain. So in case of resistant strain, we found that one strain, which was the most resistant, is a mutant which produces lot of ergostrol. And what are these ergostrol? these ergosterol are like cholesterol in our plasma membrane. So this gives strength to the fungal pathogen. And we found that few of top strains which were sensitive are deficient in ergosterol production. So this gave us understanding that maybe this red stem extract is impacting ergosterol biosynthesis in the pathogen. So next obvious step was to measure this ergosterol in control, just solvent, green stem extract, and with red stem extract. So you can see in the x-axis these different extracts and solvent, while y-axis tells you ergosterol amount in those sclerotinia. And we can see there is significantly reduced amount of ergosterol getting produced in a sclerotinia challenged with red stem extract. So to just summarize what we learned from this free genomics, omics, like transcriptomics, metabolomics, and chemical genomics about the resistance mechanism of the plant. So what happens, our model suggests that when there is a sclerotinia sclerotium, it may apocyria, and some of these molecules on outside of the pathogen, like PAMs, might be getting recognized, and it evolves different type of strategy to control the pathogenesis of this pathogen. Plant uses inhibition of ROS as one of the way to control the disease, and another one is activation of jasmonic acid isoleucine, and then production of different kinds of phenolics compounds which might be inhibiting this pathogen by acting on the ergosterol biosynthesis. So far I have talked about what plant does to resist itself. But another important part is to also understand the virulence strategy of this pathogen. So for this study, we chose to do a transcriptomics, same samples, but here we divided our study for susceptible infection, because there we will know more about how the pathogen strategizes to cause disease. So this is early infection step, like 24-hour post-infection and 48-hour post-infection. And then we decided this as a late infection because there is a big lesion compared to this early infection. And I'm just showing you a heat map of this transcriptomic study, which also relates to the phenotype which we are seeing at early induction of this sclerotinia gene. So this is 24-hour sample, 3 biological replicate, 48-hour sample, and this is 96-hour sample. And whatever you're seeing in the red is upregulated gene, whatever you are seeing in blue is downregulated. So I will just conclude with the strategy which sclerotinia sclerotium uses to cause disease. So what we are suggesting from this transcriptomic study that when sclerotinia sclerotium attacks the plant, it uses different kind of cell wall degrading enzyme, chitinase or chitin binding proteins to bind to chitin, such that chitin do not become a PAMP molecule for plant recognition. And there are several different kind of proteases to take care of different kind of protein molecules secreted from the plant, like we heard about cysteine rich compounds and other type of things which plant produces. It also produces botrytinic acid pathway gene, which is important to take care of antimicrobial compounds secreted from the plant. It also, the pathogen also overexpresses this transporter, which is important for detoxification purposes. And in the next step of the infection, which is late stage, this pathogen produces the different kinds of polyketides, detox enzymes, and effector, which might be inhibiting a different pathway in the plant, which includes programmed cell death and jasmonic acid isolation activation. So with this, I'll conclude my talk, and I just want to acknowledge my lab members. Nick Talmo is a doctoral student, Grace and Kay, and Kaba's lab members, where I started my postdoctoral study, and my collaborator, Bob Stupel, who is helping me with bioengineering the soybean plants, Mehdi, Jeff, and Damon, and these are the current funding bodies. Thank you, and I'm ready to take any questions.
So again, thanks, Ashish. Excellent talk, and we'll take few questions again, and then we'll shift to the next speaker. Any question for Ashish? So maybe I can take ask the question. Okay. You, Divya, you go ahead.
Quick question for you, Ashish. A very nice talk, very interesting work. I just wanted to ask, did you follow up beyond 72 hours post inoculation when it came to the phenylpropanoid pathway genes, and whether any of those metabolites were also induced in the susceptible interaction, but a delayed kind of response?
Yes, we did follow till 10 days. So we sent also sample for 10 days inoculation. We wanted to know if there is more of these antimicrobial compounds getting accumulated. And we do see there is like consistent maintenance of this high antimicrobial compound in case of resistance response. But in case of susceptible response, even after 10 days, most of the plant anyway is dead. But whatever, if we take care of the amount of sample, those antimicrobial were still less. It was not as good as resistances.
Okay, thank you. Yeah.
So Ashish, I was really interested to know about this yeast chemical screening, library screening. So my question is, why do you think, what led you think that yeast may behave similarly to that of Scrotinia? So do you have some preliminary observation, or you just went ahead and tested?
No, so this chemical genomics profiling is used to understand mode of action for any antimicrobial compound. And I was already working on different antimicrobial compound, which we published in PNS and other journal, where we shown that using this as like not direct, yeast is not a fungus, but in a way, indirect way of eukaryotic system to understand where it can inhibit. So, and we use the same system for red stem extract, and now we have just published one paper with the Japanese collaboration where we have shown another antimicrobial compound which inhibits a different part of cell wall for antifungal function. So it's like transcriptomics and metabolomics method to know mode of action.
So do you think this library is public commercially available, or we need to collaborate with them to commercially available?
It's commercially available. Only thing is analysis, maybe you have to collaborate.
Okay. Thank you. So maybe one more question. Senthil, do you want to ask?
I just want to have a quick question, Ashish. I think I asked similar question when you were here also last time. But my point is the sclerotonia. You showed an extensive pathway of the pathway that also can shuffle into lignin biosynthesis and lignin related stuff. So is sclerotonia, when it is crossing stem rot, is lignin a barrier, or the sclerotonia uses this as a carbon source? Because we are finding slowly for our pathogen, which is an necrotrope, that lignin is becoming a carbon source rather than a barrier.
Yeah, very good question. Actually, in case of sclerotinia interaction also, less lignin means people have hypothesized that leads to resistance. So, but my hypothesis is like total lignin. If there is total lignin amount going down because of resistance response, may not be the true thing happening here, because we checked for this solubilized lignin in case of susceptible response and resistance response, and we don't find any significant difference. what we are hypothesizing that the metabolites in this pathway, like ferulic acid, caffeic acid, and other metabolites, different aldehydes, they are upregulated in resistance response. So not the total amount of lignin, which is important here, but what that lignin is made up of is more important.
Yeah, so you're saying whether it is S by G ratio or whichever the type of lignin and whose ratio decides. Okay. Okay.
Exactly. So that is what we are finding, and we are concentrating on that. Yeah, please.
Namaskar. Assis. So just I have a general query on these fungal pathogens or the pathogen which are, suppose it will be inoculate in the stem. Does the disease happens at a distance, or the disease and the pathogen locations are identical?
So that's a very good question. I'll say that it depends on what kind of pathogen you are following. If you are following a biotrophic pathogen, maybe you will not know where it started and where it is going if you have never inoculated yourself, because they are keeping the plant healthy as long as they are feasting on it. But in case of necrotrophic pathogen, because they want dead cell for their own survival, for nutrition. So mostly there is a small phase where they remain biotroph just to colonize it. But once they colonize a little part of it, they start producing the cell degrading enzyme and other things to cause cell death, because that is their nutrition and they want it right away. They don't want to wait longer and longer. So the infection and the lesion length start at the site of infection, and then it increases as they colonize more and more of the plant.
So, Subin, shall I add little bit with respect to Ralstonia? So that
it will be nice.
Rhizoctonia. Actually, again, Rhizoctonia is a necrotrophic fungal pathogen. And the interesting part of it is, as Ashish says, for most of the necrotrophs, the infection starts at the point of inoculation and it spreads. This Rhizoctonia, what initially it will do, it will move parallel to the rice veins. Okay, so let's say you are infecting here, it is not started causing necrosis there. It will little bit move and then it will cause one lesion there. Then again, that lesion will not increase in size. It will cause another lesion there. And later on, all the lesion will coalesce. So something is very interesting going on there. That it is like necrotroph, it wants to kill. But I think initially it's like a very naughty pathogen, which will first go spread everywhere and then will make a bigger lesion.
Very nice observation. Very nice.
That is something which we have.
Yes, I think there is a biotrophic phase involved before this necrotrophism. Actually,
that's what I've been claiming.
Yeah, actually, by typical textbook definition, now if you go and start thinking and telling your student that okay, this is a necrotrophic pathogen, they completely go in a different direction where this is not truly necrotrophic.
Agree.
Whatever the given the
right. So maybe we'll keep discussion in the panel. So let's move on and let's invite Devyanshi. So I think she is new to our group, but she has been doing excellent work, particularly with respect to Ralstonia pathogenesis. So it's my pleasure to invite her for the talk. Devyanshi?
Yeah, thank you, sir, for introducing me. As Gopalji said, this is my first time interacting with all of you, and I want to thank the MIPM committee for organizing this and giving me this opportunity to present my work. I'm going to share my screen and can you see my slides?
Yes.
Okay, so I work at Department of Plant Pathology at Minnesota, and I want to start with acknowledging that we all understand that plants are autonomous organisms. However, plants are colonized by different kinds of microbes, bacteria, fungi, archaea, and other stuff. And these microbes have the potential to influence plant health and productivity. And there is enough evidence to convince us that these interactions are important for the same reason. Looking at the spectrum, these relationships between the microbes and the plant can span from beneficial to pathogenic relationships. And these interactions are dependent on environmental conditions, also on nutrient status and plant species. In my lab, we focus on pathogens, particularly, and we also focus on beneficial microbes. In terms of pathogens, I study gram-positive bacteria, Clavibacter, that causes wilt in corn, and also continuing to work with Ralstonia solanacearum. And in terms of beneficial microbes, I look at mycorrhizal fungi and its interactions with diazotrophic bacteria and how it can help in efficient uptake of nutrients. So today I'm going to focus on my work during my postdoc at UW-Madison. This is the pseudo-colored scanning electron micrograph of Ralstonia solanacearum cells colonizing xylem of a tomato plant. So you can see that it's forming, started to form some aggregates, and eventually it grows as biofilm to clog these xylem channels. So Ralstonia solanacearum is a very well-known bacterial wilt pathogen, and it has an extensive host range of about 200 plant species in 50 families. On the top left corner, this is geranium, an ornamental plant. You can see the wilting symptoms. And depending on the disease that it causes in particular host, the names of the disease are slightly different. For example, in banana, it's called blood disease. So you can see this red discoloration, which gives the name blood disease. Also, it causes bacterial wilt in many solanaceous plants like tomato and potato. But other than that, it also causes bacterial wilt in many weeds, shrubs, and trees. That includes monocots and dicots. And here you can see potato, it's called southern bacterial wilt or brown rot. And on the right corner, you can see if an infected plant, you cut the stem and put it in the clear water, you can see the slime exuding from the cut stem, which is an indication that of heavy infection, and that the bacterial EPS or the ooze is exuding from the cut vascular bundles. So Ralstonia solanacearum is a widely distributed pathogen, and because of the extensive diversity of this pathogen, it's popularly known as Ralstonia solanacearum species complex. So here I want to draw your attention to the top clade, top group. This is our Ralstonia pseudosolanacearum. Earlier, it was grouped as phylotype 1 and 3. So phylotype 1 is found in Asia. Phylotype 3 is found in Africa. But the genomic data or the genomic comparisons of over more than 50 strains now convince us that phylotype 1 and 3 have the average nucleotide identity as above 96%. So now they're grouped as one clade. So Ralstonia solanacearum phylotype 2 comes from American continent, and Ralstonia syzygi phylotype 4 is found in Indonesia, and there are other subspecies. So here they cause disease in cloves and banana, and as I mentioned, they also cause the disease name are Sumatra disease or of clove or blood disease bacterium. So today I'm going to present my work on phylotype 1 strain, which is GMI 1000. One common thing, despite coming from different geographical locations and their biology is a bit different, one common theme is that they all colonize xylem and they all cause bacterial wilt. So let's take a look at the general life cycle of Ralstonia solanicerum. So it's a soil-borne pathogen, and it's understood that in soil, it's living as a saprophyte, and particularly at low cell density. And once it finds its preferred food, which is found in the root exudates of the plants, it can colonize the roots and use its chemotactic ability and flagellar motility to colonize the roots. And once it makes its way through wounds or emergence point where the root growth is happening, those are the hot spots for Ralstonia to enter. What happens is it also has a chance to colonize the developing xylem vessels. So it makes its way through the xylem vessels, and eventually, once the conditions are favorable, it starts forming biofilms. And once it reaches a particular concentration, it also secretes the slimy material, which is the exopolysaccharide. And at about 10 to the power 10 CFU per gram, the xylem vessels are clogged, and that's when it becomes a parasite. So it's using plant resources for its benefit. Now, when it has used everything up and the plant dies, it makes its way back to the soil. And now it's again at a low cell density in soil, starting its journey as a saprophyte. So it's an interesting lifestyle, two different lifestyles, as a saprophyte and as a parasite. So the question we asked is, does population density help this pathogen in transitioning between saprophytic and parasitic lifestyle? So taking a step back, most of you are probably aware of this density-dependent behavior, which is called quorum sensing. So I was showing you soil where the pathogen is present in low cell density. So we call it as quorum sensing inactive state. Whereas in planta, once it builds up its population, the concentration of these triangles or the signaling molecules that help the pathogen to gauge the cell density increase and hit the threshold concentrations, it activates this quorum sensing state, which helps in activating several virulence factors in this pathogen. So a lot of studies have been already done, what's upregulated and downregulated at wild type when it's in the quorum sensing active state. But what we don't know is what is happening in quorum sensing inactive state. And what's driving this interest is that Ralstonia can survive in soil and water for longer periods of time. So we've got interested, it's like, what is happening at the inactive state? How is it surviving? So one approach to get a glimpse of the saprophytic lifestyle is to make quorum sensing mutant, or we call it quorum sensing inactive strain. So here we mutated this transcriptional regulator, PHCA, which is known to sense this quorum sensing molecule and activate the virulence factors. So right away, you can see when we made this mutant, the colonies change its appearance when you compare it to the wild type. So it loses its EPS production ability, and you can probably see some brown pigment that is also an indication of this mutant strain. So before we began this study, we were already aware that some of the genes are positively regulated by this master regulator. So it's a LYSAR type transcriptional regulator, and EPS and plant cell wall degrading enzymes were already known to be positively regulated by this protein. And we also knew some negative traits, negatively regulated traits by PSA, such as type 3 secretion system or flea genes that express the motility, and siderophore genes. However, many of these studies were done in vitro, and every time we made comparison between in vitro and in vivo studies, we find some inconsistencies. So for our purpose, we decided to compare the global gene expression of the GMI 1000, which is the wild type strain, and a PHCA strain, which is the quorum sensing inactive strain. Now, the natural way, as I showed you earlier, that they're soil-borne pathogen, and they colonize the roots and make their way into the plants. However, PHCA mutant cannot colonize the roots and make its way to the xylem vessels by itself. So we had to come up with a different method, which is also known as a petiole inoculation, so directly inoculating the stem. So what we did is put 2,000 cells of Ralstonia serum, either wild type or PHCA. And first, before we begin performing the actual experiment of transcriptomic analysis, we wanted to compare the population kinetics of both the strain, the wild type and the PHCA strain. And over time, when we were monitoring, we found that at 72 hours post-inoculation, the population was quite similar. This was an important step because we were worried that we'll have a lot of wild type cells, but probably not enough PHCA cells because it's already compromised in its virulence abilities. So that was a sweet spot for us to do the transcriptomic analysis. So collected the RNA, sent it for RNA-seq, and when it came out, this is what we got. So the gray dots over here indicate non-differentially expressed genes, and the black ones are the differentially expressed genes. So I want to draw your attention that the false discovery rate was set quite high, so it was less than 0.005, so it was quite stringent. So whatever was positively regulated at low cell density, that means in PHCA strain, are observed here, and whatever all the downregulated ones, we consider that it's expressed at high cell density. So while we confirmed a lot of previous findings, what stood out from this study is two things. One is the pink dots here representing the genes that map the metabolic pathways that utilize the carbon, nitrogen, sulfur, phosphorus, and iron. And another interesting thing we found was the attachment proteins. Some were upregulated and some were downregulated. So we began characterizing first with the metabolic pathway. So it looks like it has the ability to use diverse kind of nutrients. So what we did is did a growth curve with both the PHCA mutant and the wild type in healthy xylem sap of tomato and infected xylem sap. So here you can see that both the red lines, they pick up fast. And in case of infected sap, PHCA grew very well as compared to other treatments. So that was a surprise that this mutant, we were hoping that it would be compromised in so many ways, but it turned out that it has increased fitness. So we started looking at different transporters in our transcriptomic study, and we found that several transporters were upregulated in PHCA mutant in planta. And at that time, when I was doing my postdoc, there was a PhD student in the same lab Tiffany Love Power, and she was doing the metabolomics. So it was kind of good collaboration because she was doing metabolomics, collecting xylem sap from diseased and healthy tomato plants. I would always reach out to her, say, do you find this substrate in your xylem sap? Because I'm finding this transporter is upregulated. And we both sat one afternoon and we found that a lot of the substrates that she's finding, the corresponding transporters are upregulated. So we were both pretty excited about this coherence in metabolomics and transcriptomics data. So that gave us an impetus to further test the ability of PHCA mutant to use broader or diverse nutrients. So we used biolog plates. These are 96 well plates, each well with different carbon substrate or nitrogen or sulfur substrate, whatever you want to test. So here, the green curves indicate that PHCA is growing better than wild type. Yellow indicates the wild type growth. And if the wild type takes over the PHCA, you would have seen red curves. So you can see many green curves indicating that PHCA is versatile in using these nutrients. So for you to summarize, what we found is the red ones in this graph indicate that only PHCA can consume this and not wild type. Blue ones, only wild type can consume, but not PHCA. And the gray, the light gray one is they both use equally well, and the gray ones are neither of them are able to use. So you can see a lot of red bars over here indicating that PHCA is really doing well in using these substrates, be it sugars or amino acids or carboxylic acids or other nitrogenous compounds. So we wanted to map the transcriptomic and metabolomics data. Sorry, this is a complicated figure, but I will walk you through it. So all the metabolites that are in blue fonts indicate that these were found in the metabolomics experiment. And all the purple arrows indicating different metabolic pathways indicate that the genes that make up that pathway were induced in the PHCA mutant. And the green arrows indicate that the genes were induced at high cell density only in wild type, as or higher as compared to the PHCA mutant. So you can see that all the sugars like galactose, sucrose, trehalose, all of these were being utilized by the wild type, whereas the PHCA used this intermediate metabolites, indicating that it upregulates many core metabolic pathways and has the ability to use diverse nutrients, be it fatty acid degradation compounds or nitrogen metabolism and others. So next, another thing we found is the nitrogen metabolism. So several nitrate transporters and other glutamate transporters were also upregulated. And they all indicated that probably they feed into this making of nitrite. So nitrite concentration should have been up if this is true. So what we did is we did some experiment where we could detect the nitrite per cell. So in vitro, we found that PHCA was making more nitrite. In planta also, we found that it was making more nitrite. So this confirmed our transcriptomic analysis. The same goes true for siderophores that we found. So when we inoculate the growth medium with low iron, we found that PHCA was able to grow better than wild type or the complement strains. So all of these functional experiments, along with the transcriptomic data and the metabolomics data, indicated that the PHCA has the ability to de-repress diverse transporters and catabolic pathways when the pathogen is at low cell density. And this also suggests that it's acting as a generalist metabolic strategy, which allows the pathogen to survive longer in soil, water, or it could be early phase during infection. Next, we focused on our functional characterization of attachment proteins. So we have not characterized the individual protein, but the behavior of the pathogen was looked at. So first we did the biofilm experiment, the classical crystal violet experiment, and we found that it poorly attaches to the PVC substrate and to other Ralstonia solanacearum based on this confocal microscopy. So here you can see aggregates or mature biofilms happening in wild type and the complement strain, whereas PHCA mutant was not able to form that 3D biofilm structures. So after that, we were interested in looking the cells in planta. So we did scanning electron microscopy of infected tomato xylem vessels. And this is the wild type. You can see nice rod-shaped cells. Whereas the PHCA ones, you can see that they're highly aggregated, and also the cell size is smaller as compared to wild type. So this is the closer look, and you can see this fibrils. So generally this would have been EPS in case of wild type, but because the way SCM samples are prepared, this is what we are seeing when they are desiccated. And we also saw a lot more of these fibrils when we looked at the PHCA treated samples. So what does all of this mean? So we started looking at other xylem vessels. So what we found is xylem vessels in case of wild type were colonizing pretty much all the xylem vessels in that particular section. Whereas in case of PHCA treated cells, we found that one or probably two would be heavily infected, but other xylem vessels would not be colonized at all. So this behavior indicates that it shows increased attachment for quorum sensing inactive strain and reduced spreading, which means that the quorum sensing inactive strain is less dispersive than quorum sensing active strain. To confirm that, we did some root attachment studies on tomato seedlings, and we confirmed the same, that PHCA was attaching more than the wild type or the complement strain. Another experiment that we did is we did the petiole inoculation and sampled after having first symptom of wilt. In wild type, we sampled +2 centimeter, so above the point of inoculation and below the point of inoculation. And here you can clearly see that PHCA has problems spreading above the point of infection. So overall, it indicates that it loses its ability to disperse and it's highly aggregated. And we hypothesize that probably these attachment proteins, or also known as adhesins, help Ralstonia to attach to the host or to each other during the early stage of infection. So to summarize this talk, what we think is PHCA is allowing the pathogen to do this trade-off between saprophytic lifestyle and parasitic lifestyle. And in saprophytic lifestyle, it's more focused on scavenging nutrients and attachment to the plant surfaces. Whereas later in the disease, you can see in this SCM, it's heavily colonized, it stops moving and attaches to each other and enjoys the conditioning of the xylem vessels that the PHCA has done, and then produce or launch their virulence factors like EPS production or cell wall degrading enzymes. So with that, I want to thank Allen Lab members during my postdoc. It was a great collaboration with other PhD students. She's now faculty at UC Davis, and also people in microscopy facilities helping me to take images and prepare my samples, and Department of Plant Pathology for giving me opportunity to continue my work. And also want to thank once again the MIPM organizing committee to give me this opportunity and connect with scientists in India. And with that, I'm happy to take any questions.
Thank you, Devyanshi, for such a nice and elaborate explanation of how the saprophytic and the pathogenic mode of Ralstonia differs. So we'll take a couple of questions and then we'll shift to next. Any question for her? Suben, you have to unmute.
Yeah. Namaskar.
Namaskar.
So very nice to learn. So why does the bacterium motility is very high? What is the PSCA mutant than normal? It spreads very fast.
Yes. So PHCA mutant, as you saw in scanning electron microscopy, and it doesn't spread. So at low cell density, it looks like it can spread, but at high cell density, the motility genes are shut off because it's a waste of energy to have motility genes produced when they're already building up high in the xylem vessels. They don't need that because already the xylem flow in that xylem vessel is assisting them to do. But 5% of that population which builds up in the xylem vessel, they do express some motility, and that is to disperse from the biofilm and colonize new places in the plants.
And this was nice. The growth rate is high. The xylem sap of PHCA.
Yes.
Is very interesting. Okay,
yeah.
Great.
Thank
so
you.
Simple sir will have question. Beautiful.
If we have time, one more question, or we can.
Yeah. Or maybe we can.
Okay,
we'll take one. We'll take one question, then we'll shift to Sanjay.
No, I was just trying to know the same follow-up. Like this, this mutant is metabolically equal in its capacity, like a wild type. Like, for example, if you provide some, already xylem is a nutrient-limited environment, but if you subdue the nutrition, will it still have the same motility, same level of spread, same level of? So, how metabolically are they active in utilizing this mutant points? I'm asking.
So I showed you the biolog plates, and it has those nutrients which are also not found in xylem vessels. So what we found is traditionally xylem is considered nutrient limited, but still we found from metabolomics there were 110 compounds, and out of that 110, there were 76 compounds that this mutant was able to use. And when we did biolog, other than 76, like something that is not found in xylem vessel, it was also able to use those compounds.
Okay, so it's metabolically equally active as that of a wild type. Okay, thank you.
Yeah.
So let's once again thank you, Devyanshi. So we'll have, hope you have time to attend the panel discussion at the end. Yeah, sure. So let's now invite Sanjyoti to make her presentation.
Yeah, thanks Gopalji, and thank you also for the organizers. If I keep my video shut, is it okay? Actually, I didn't realize it's cost of computer under the MIT.
Yeah, I was going to tell you, you have shifted your banner from MIT to MIT.
No, I didn't bring my computer. So is it okay if I keep my video off?
You can keep it on also, it's up to you. We don't mind it. So it's okay, you go at least.
Yeah,
Share your slide, yeah.
Yeah, just give me a minute. So am I visible? Like my screen is visible?
Yeah, perfect. Sanjyoti, you go ahead.
Yeah. So first of all, thank you all the organizer and also putting me in the slot where I wanted. And my sincere apology for not presenting in the entire symposium due to some of my personal commitment. So I will switch my gear from pathogenesis to symbiosis. So my lab is focusing on symbiotic nitrogen fixation and understanding why, like what are the predisposition or the factor which are responsible, which gave the plant blessings for nitrogen fixation, as well as we are also trying to understand that why among the different legume, they are so different. So this is the leguminous clade. So in legume, majority of the plants, they can fix nodule, and among them papillinoids, the subclade, they can fix nitrogen in more efficiently, and 90% of the plant in this subclade, they can actually fix nitrogen, which is like more sparsely dispersed along the other clades. So in our lab, we are working with Medicago truncatula, which is a model legume. Then chickpea, another crop legume, and peanut, another crop legume. So choosing these three legume, there are two logic. Definitely that the crop versus model, but apart from that, they are they are evolutionary present in the different phylogenetic position. And additionally, the nodules are either determinant or indeterminate, means they do have meristem or they loses their meristem along their development. And the bacteria, the symbiotic unit. So after the endocytosis, it's called bacterioid. Their structures are different. So as you can see, these are the single bacterials, single plant cell, and these green dots are single bacteria. So the rhizobia, they looks pretty different when they enter inside the plant cell, and it's largely controlled by the plant, not the bacteria. So this is for chickpea, the bacterioids are small. In case of peanut, they're again spherical but large. And in case of Medicago, they're very gigantic, endoreduplicated, elongated, and sometimes Y-shaped. Evolutionary, there are around 25 to 30 million year difference. So there is a diversification, and that also give rise to this different characteristic that plant evolved. And with respect to peanut and this clade, there are around 50 to 55 million years. So we are trying to understand that what are the basic mechanism which is conserved among these legumes, and what are the advanced mechanism that gives peanut very high nitrogen fixation capacity versus Medicago moderate and chickpea low among the three. So today I will talk about mainly the work that Vikas did during her PhD, and there was enormous support from Vendrila, and also Isha contributed significantly in last 1 year. And in the model legume, Divita and Akanksha are working, so they will present their work tomorrow. So we started with RNA sequencing of peanut, which is an allotetraploid legume. And when we started this work, it's actually around 2015, the genome sequencing was not available. So we took different time point from 0, 6, 10, 15, and 21. So as you can see, the nodule is developing, their size and shapes are changing, as well as their nitrogen fixing capacity are also with starting from nil to increasing. So we do have all the couple data for nitrogen fixation. So for lack of time, I'm not showing those data, but the point is 0 to 10, there is no nitrogen fixation. 15, they started, and 21, the nitrogen fixation, boom. So then we analyze the RNA sequencing data using allotetraploid peanut progenitor genome that was available that time. And later, after the publication of the genome, we also capitalized on the tetraploid genome to analyze the data. So we find many surprising factors, including the absence of legumoglobin in peanut. But for today's presentation, I will focus, and which is the focus of our lab, that how the symbiosome developed. Symbiosome is the bacterial unit inside the plant. So they are actually enveloped with the plant membrane. So all the time the bacteria is residing inside an organelle-like structure. So these are peanut semi-thin section, one single cell. Then this is the nucleus. And after the endocytosis, you can see the bacteria, they are elongated and later they transform to this spherical shape. This is a single electron microscopic picture showing that this is the spherical bacterioid. So inside the bacteria, and this is the membrane, that is the plant membrane. So the phase when the bacteria is actually this elongated shape, at that phase, they don't fix any nitrogen for peanut. And then they changes to this spherical shape. So for Medicago, this elongated bacterioid keep on elongating more and then they become Y-shaped. So I will explain it later in the next slide. And this transition from this elongated to spherical is a very unique feature for peanut. And we ask the question that what are the factors that actually helping peanut, or what are the factors that actually a plant factor that guiding this bacterial transformation. So we ask, as you can see, the shape change is usually associated with endoreduplication, as also this is case for Medicago. And there was an old publication, they were mentioning that there are endoreduplication. So we ask the question, what are the factors that guiding this endoreduplication? And along with that, we also ask the question, what are the factors that helping to shape that spherical symbiosome formation? So before going about peanut, I will give you a brief knowledge what we know from Medicago. So from Medicago, we know that the bacteria after endocytosis, so this is the plant membrane and bacterial membrane, and this green thing are bacterial genome. They keep on endoreduplicating up to 64-fold. So now the bacteria is having many more nitrogenase enzyme, so they can fix more nitrogen. So that is a simplistic explanation. There are many more complicated biochemical things are happening inside. And for this elongation, plant-derived molecule need to be delivered from the ER to the symbiosome. And this delivery is guided through vesicular trafficking pathway. And in ER, there is a particular enzyme complex, which is called DNF1 complex. The name came from a reverse genetic screen, does not fix mutant. So if you have the mutation, it will not cleave the signal sequence, and then the protein will reside in ER, and then the bacteria will not elongate. So they will not able to fix, the nodule will not able to fix nitrogen. So this we had in our mind in the background. And what are the factors that actually plant giving to the bacteria? These are small peptide called nodule specific cysteine rich peptide. So as from the name, you can understand that there is only single unique factor about this peptide. It's presence of cysteine and a signal peptide. So how they are manipulating the genome endoreduplication, it's interacting with the bacterial cell cycle, but beyond that, the knowledge is limited. So the first thing that we look for in peanut transcriptome data, that whether these nodule specific cysteine rich peptides are there or not. So I will just introduce you this structure. So this is the elongated Medicago like bacterioid. So they take unique elongated or Y-shaped structure. And in case of peanut, these are their spherical symbiosome. So for this elongation, there are over 750 NCR peptide. They only express during nodulation. They do have signal peptide, and they usually have 6 conserved cysteine residue. So at that time, we were unable to detect any NCR peptide. One of the reasons is they're very short, they're 20 to 24, and they're very dissimilar. They don't have very high sequence similarity. The other reason is that in the genome, they were not annotated. So later, when we did our annotation, we were able to find around 55 defensine-like NCRs. So these NCRs, why I'm mentioning defensine-like? Because defensine are the pathogenic signature. So defensine are the molecule usually plant secrete to kill some pathogen. And they have this 8 cysteine residue. So it was actually proposed by the symbiotic community that from this 8 to 6 conversion of the cysteine was necessary for this peptide to do its symbiotic function. So what we get in peanut, we get only defensine like NCR, and that too only 55. Among them, 30 are not yet annotated in the genome. So we annotated by annotating this gene from our transcriptome data. So we thought the probable answer for this endoduplication is defensin. But along with the defensin, we got upregulation of a lot of cysteine-rich secretory protein, antigen 5, and pathogenesis related 1 or CAP protein. So what are CAPs? They are nothing but PR1 protein. They are homologs. And in peanut nodule, there are around 59 CAPs. They are expressing only exclusively in nodule, not any other part of the plant. So what this heat map are showing? This heat map are showing that this cap, as you can see, that this is cap and the top designation is NCR. So like NCR, they're expressing later during nodulation. And if you can recall the microscopic image from 10 to 15 dpi, the conversion of this elongated to spherical symbiosome is happening. And their expression is also coinciding, both NCRs and cap, at around 15 dpi, indicating that this cap is doing something, or this symbiotic PR1 is doing something for during symbiosis. Another interesting fact that we noticed that during this conversation, as we go through from 10 to 15, the shapes are changing from intermediate elliptical shape, and then probably this elliptical shape can fix some nitrogen, and then they're booming at 15 dpa. So why I'm repeating this slide? Just to emphasize that this conversation and this expression profile, they are coinciding. That give us a hint that this is doing something during nitrogen fixation. So we look for this cap protein, and so to give one small introduction, as the PR1 protein is a pathogenesis related one, and which usually get induced during pathogenesis. So we look for peanut pathogenesis related protein, and we were able to see that those proteins are there. But apart from that, there are 50, and that's why in peanut genome, there are much more PR1 protein. They're doing symbiotic role. And they do have this cap domain. The cap domain, I will describe about cap domain in my upcoming slides. Apart from that, majority of them, they do have signal peptide. And then this signal peptide do have significant sequence homology, especially with respect to aromatic amino acids with NCRs. So this gives us the hunch that this PR1 protein are probably going to symbiosome and they are doing symbiotic function. Then we generated a phylogenetic tree, taking PR1 protein from Arabidopsis, from other legumes like soybean, medicago, lotus, and also from human and yeast, tomato. So we try to take as much as protein that we can take to see what is the diverse evolution that is happening in this protein family. So this black letter is denoting all the other caps which we are taking from other plants and also from peanut. And when the peanut gene is expressing during nodulation, they either demarcated with green or red. So it depending on whether we are taking the gene from a progenitor genotype or a tetraploid genotype. So what we are seeing that the symbiotic PR1, they're clustering separately, and this is their diverse evolution. And there is a small clade. In this clade, there is only PR1 protein from peanut, but among them, there are few PR1 genes which are expressing during nodulation. Even we can see that there are human PR1s which are clustering with medicago and soybean genes, but not with like nodule-induced peanut PR1s or caps. And here I will show there are two canonical PR1 of yeast. So they are coming here and they can show sequence homologies with Arabidopsis. So we go ahead. So because we'll present the detailed analysis of these genes, but we go ahead and we purified the protein from symbiosome. So we first did Parkol gradient, isolated the symbiosomes, and then did this MSMS analysis on this symbiosome isolated protein. We got bacterial protein, peanut protein, and along with that, as expected, we got some defensins, that is NCRs, and also the caps. So these caps belong to group 4, 3, and only we got 1 from 2. So I will go back to my previous slide, and now I will describe this group again. So as these PR1s, they are actually only present with the other PR1 protein. We thought that they actually evolved and they give this diverse PR1. So we name them 1, 2, 3, and 4. So what we are now seeing that majority of 3 and 4 proteins, they're going to symbiosome, only 1 from group 2. So we go ahead and design RNA construct. So these PR1 proteins, they're very homologous. Again, it's difficult to design RNA construct specifically against them, as well as it's very difficult to design qRT-PCR primer also for each gene. So we decided to design RNA construct, 1 against all the PR1 protein, which we designated as RNA A, and 2, 1 against group 2 and 1 against group 4. It was impossible to design anything against group 3 specifically. So in every cases, we do see a significant reduction, significant deformation in nodulation, and significant reduction in this pink nodulation. So this A construct and C construct. So basically what you have to understand that A is going to downregulate all the PR1 protein probably, because we don't have a still efficient means. We are working on them to do qRT-PCR and do specifically identify which gene is getting downregulated. So A is probably downregulating all the PR1s and C is downregulating mostly group 4 PR1. So in every cases, we are seeing this, instead of this pink nodule, white nodule, then clusters of nodules and nodule with deformed structure. So if we cut this nodule, for lack of time, I'm not showing the picture. So if we cut up the nodule, it looks like more lateral root. It's like spiral vasculature, but the bacteria is still there and they are making symbiosomes. So it's a very unique combination of structure. So this data is telling us that this PR1s is helping in nodule development, because otherwise we won't have gotten this clusters or this spiral vasculature containing nodule, and as well as it also inhibiting nodule initiation. That's why instead of pink nodule, not inhibiting nodule initiation, it inhibiting this symbiotic nitrogen fixation. That's why we are getting a lot of white nodule bumps and less pink nodule. So now the question is that what this PR1 can do. So we look at the literature, and there is a Switzerland group, and this is from Roger's group. So what they are doing, they have a very unique assay. So if you remember, I mentioned there are 2 PR1 protein in yeast. So these are PRYA and PRY2, PRY1 and PRY2. So these 2 protein are helping in transporting cholesterol acetate out of the cell. So they generated 2 mutant, one which will create the environment. So there will be more cholesterol acetate accumulation, and the SY1 mutant, which will inhibit acetate to normal cholesterol conversion. So there will be accumulation of cholesterol acetate. Now in this PR1 and PR2 mutant, there will be no export of cholesterol acetate. So in yeast, PRY1 and PRY2, they actually transport cholesterol acetate from the cell to out of the cell. That's why you can see that in case of this 2 mutant, there is no cholesterol acetate transport out of the cell. And in case of when you have this quadruple mutant, they are actually transporting it. Here also the quantification. So when they complement the mutant with plant PR1 protein, like this is Nicotiana P14C or PR1A, so they were able to complement this transport assay. So it summarizes that in this case, there will be this PR1 protein, plant PR1 protein, pathogenesis related one, they are doing this function. So we collaborated with this group, And they did this assay for us, and what we can see, at least 4 of the PR1 protein that we have chosen from this 49, they are not able to transport any cholesterol acetate out of the cell. So indicating they are not cholesterol transporter. So now the question is what they can actually transport. So we look back, and there are some lower fungus and also yeast PRY1 only, they can also bind to fatty acid. So if they're binding to fatty acid, they're binding it through their pocket inside these 2 alpha helices, and they bind to cholesterol through a motif that is called caveolin-like motif, which is present on like in the surface. So we speculate that, okay, whether they are actually transporting this lipid. So this is still speculation. So we were able to purify the protein, but we have some supporting data that is giving us hint they're probably transporting lipid. So when we generated the PR1 protein with GFP fusion, so this is the whole nodule, and this is inside the infected cell, and you can probably see there are some green dots. So if I zoom in this green dot area, you will see this blue are the nucleus, and they are this green protein are accumulating in some large green dots. So these are probably lipid bodies. So I will explain why I'm saying they're probably lipid bodies. So before that, if we zoom in the infected area, we are seeing that this PR1 is actually accumulating on the symbiosome membrane. Not only just uniformly around the symbiosome membrane, they are actually accumulating in some puncta, indicating they are probably making some lipid rafts. So we further did Nile blue stain, which is a typical neutral lipid stain, and we were able to see similar lipid staining in the uninfected zones. And in the infected cells on the symbiosome membrane, again, blue are the symbiosomes. These are the, like there are lipid granules or lipid dots on symbiosome membrane. So we still need to do this, like the staining and the localization together. So now going back to our transcriptome data, we thought, okay, peanut being an oil legume, so it generate a lot of oil in their seeds. So whether these oil biosynthesis genes, they're actually accumulating in nodules or not. So when we dig up data, we do see there is a lot of oil biosynthesis gene, whether it's a plastidial and then, so the eukaryotic and then that Kennedy pathway, the majority of the gene, they are upregulated. So this like red arrow, if they are fat, that means there are over 100-fold upregulation. And if they are narrow, it could be 2 to between 10-fold. So there are upregulation. And then the hypothesis that we started that CAP is present only in peanut, it's not present nodule specifically in any other legume. So there will be some unique lipid that probably they are transporting, which is also not present in any other legume plant. So likewise, we got this fat B, an enzyme, which is hydrolyzing this ACP from fatty acids. So whether it's C18 or not, until we do lipidomics, we will not know that. So this is just like, again, a hypothetical flowchart. But the point is we identified some fatty acid biosynthesis gene, which is unique to peanut. And we thought, okay, they are transporting this. So I will mention again, I will introduce DNF1, because if you remember that CAP do have that signal peptide, and we do see their localization, whether using proteomics or by using GFP fusion, they're going to symbiosome membrane. So we thought that, okay, in ER, if this lipid biosynthesis is happening, then CAP is also there. So the PR1 is also there. So they can bind to this lipids. And from this ER, if there is this lipid body is forming, they will diffuse out of the cell. So in case of the uninfected cell, if you remember that they are present in all bodies, like they're present in those punctus, probably all bodies. So we also like express this CAPS in tobacco cell, and in tobacco cell, If you express normal PR1, they usually go to apoplast. Here we can see they're around the nucleus, and this rays, they're indicating they're in the ER, and they're also in the apoplastic space. So validating the thing that they're going through ER, and in uninfected cell, DNF1 is not expressing because they are nodule specific genes. So probably that's why they're getting non-signal peptide cleaved PR1. They're getting loading there and staying in the lipid bodies. But in infected cells, this after the processing of the signal peptide, they're taking the lipid and going to the symbiosome. So this is our transcriptome data is supporting the fact, and we got the protein. So we probably will know the answer in next few months. So with that, I would like to first thank the team. So because really did a very good job in this whole work, and Isha did all the protein work. So this is a cysteine-rich protein. It was difficult to purify. So she took this hard path to purify the protein. This project is funded by DBT, which is just going to finish this year, and also NIPGR. And we do have collaboration with Peanut Genome Group, and also this PR1, I'm collaborating with Roger Snyder from University of Freiburg. And with that, I would like to thank you for your time.
Thanks, Sanjati, for such a nice elaborate study on the symbiosis. So maybe we can take one or two questions, and then we'll start the panel discussion where we can discuss about each of the presentation. So any quick question for Sanjati?
Yeah, Sanjati, this oleosomes or whatever the oil bodies, are they readily giving away the fatty acids to the microbe that easily? Because they are destined for certain location, right? Namely seeds or something. So are they being hijacked or?
Yeah, so this enzyme that Senthil I was mentioning, this is an enzyme which cleaves the ACP from fatty acid, and thereby it actually helps to release free fatty acid in the cytosol. And that free
How does it get access to that? Like physical access, like these fat bodies and the bacteria at the synthesis site itself, or during transport, or?
So the transcriptome data, this one, what I'm showing, these are the genes which are actually inducing in nodule. As I mentioned, that if the this arrow are fat, they are over 100-fold. And if these arrows are short, like they're narrow, that means they're from 2 to 10-fold. So what I would like to show here, that the whole fatty acid biosynthesis pathway is getting upregulated in nodule. It's not in seed. So we took the knowledge from peanut seed, the biosynthesis, lipid biosynthesis gene from seed, and then we would like to see the counterpart, whether they are actually expressing during nodulation. And we can see almost the whole pathway. And just to bring into the context, actually the similar pathway also being hijacked or being used by during mycorrhizal symbiosis to give lipid, because mycorrhiza are lipid auxotroph. So plant need to provide lipid, then only mycorrhiza can colonize the plant. So we're thinking similar symbiotic strategies being taken by this peanut also.
Okay. Now I was just thinking this penetration peg and these roots that where nodules are formed are far distant. That's why I was wondering how. Okay, thanks.
Thanks, Cynthia.
Hi, Sanjati. It's a very nice talk. So I have one few questions. So one of the question is that this capped protein able to interact PSP, PL, or PC?
Yeah, so we don't know that answer, Sarup. We just got the lipid strip and we were able to purify the protein. So next 2-3 months, we should know the answer.
Okay, and the second question is that is the CAPS protein. Is it possible to purify in the bacterial system, or you have to use some other system?
Yeah, we were able to purify it from the bacterial system. So there are some strain which can actually make this sulfihydral bond inside the cytosol of the E. coli. So okay. these are those strains. So in that E. coli strain, we are able to purify them.
Okay, so this is like origami or something, this kind of strain.
So this is not origami. This is there is a strain called shuffle. So we were able to purify it in shuffle. So one more thing I can show here, that this particular cap protein, So this is their monomeric actually a molecular weight. So even on SDS page, if you don't add BME or DTT, they actually make dimers. So this is Western blot validation showing that these are the dimers. So they are very strongly making this cysteine-cysteine die bond. So there is a strength shuffle. You can purify this protein.
Okay, so this is nice to know because maybe this is useful for any kind of cysteine-rich protein, right?
Yeah. Yeah. So this make actually this like ER-like environment so that you can fold. So there are many mutation that make like kind of ER-like environment to make this a protein to fold in cytosol.
Okay.
So Swarup, shall I actually, so we can start the panel discussion and you can take up, initiate, means continue the discussion. Okay. So shall I request all the speakers of the session, Alok, Ashish, Devyanshi and Sanjyoti to switch on your video and request all the participants, and maybe you can also have questions for each other. So let's have a great elaborate and descriptive panel discussion. Thank you. Swarup, you can continue with Sanjyoti.
Okay. Hello, Sanjyoti. So CAPS is I know that CAP is a defensin-like protein. That's you were telling, right? So it has any possibility it can inhibit some bacterial or fungal pathogen? Is it tested?
Yeah, so that is another thing we are testing. So usually we are testing on different variety of bacteria and fungi, but still that also we don't know the answer. So probably for some, because we have 49 and we think they can also be developed as a like potential a product against pathogen. We are testing. We don't know the exact answer, Sarup. Maybe in a year we will know.
Maybe Sanjyoti, you can switch up your sharing mode so that
you stop sharing the slides.
Yeah, stop sharing.
I'm sorry.
Also, Sanjyoti, these caps during the evolutionary lineage, it is somehow restricted to only the groundnut and the other legumes does not get it. And is there anything of other oil seed crops or other oil crops that comes in non-leguminous oil crops that these caps are? So how is this caps group of proteins evolutionary lineage goes to? Because you mentioned that it is somewhere unique to peanuts, right? Groundnut.
Yes. So the exact answer we don't know, Senthil, but we did the promoter analysis and probably we are hunching that which transcription factor is actually upregulating, which is a very nodule specific transcription factor. So this is how it is getting induced. So instead of getting, so PR1 get induced under this SAR pathway. So in this case, it is getting induced under the nodulation pathway. So even if we take this same promoter and we put it in Medicago, it will express specifically in Medicago nodule. Though Medicago nodule don't have any cap, nodule specific cap. The pathogen one is constant. So in peanut, it got evolved from pathogen one in symbiosis role. So now to connect to your second question, that oilseed. So our closest oilseed reference is soybean. And when we thoroughly check the soybean data, there is no cap, but this lipid being utilized. So lipid, either it could doing something in that in the spherical bacteria or like spherical membrane organization, or lipid is just a carbon source. So in soybean, there are like recent studies is telling that lipid is biosynthesis gene is getting induced instead of sugar, like not instead of sugar, sugar and lipid are giving the carbon source to the bacteria to make this nitrogen fixation cycle to go on. But we think in peanut that instead of sugar, because they do have more lipid, so they are now changing this from more from sugar to lipid. As we don't see much of the sugar biosynthesis, like the sweet transporters, and they are there, but their level of induction is much lesser from Medicago. So we think this is the switching, because they have that more, so they're giving this more.
Sanjyoti,
yeah. In your RNA,
Okay, Senthil, continue.
No, I was. It's a different question, a quick one. At the end, that RNAi graph, no, you showed those defunct nodules, white nodules. How is the immunity upregulation there? I mean, are they defunct because of what? Like, why they couldn't be a pink nodules?
So, Senthil, when pink means there is a synthesis of leg hemoglobin. Peanut don't have a leg hemoglobin, but now we know there is some like class 1 hemoglobin that get recruited during peanut. So leg hemoglobin will make non-oxygenic environment, and this non-oxygenic environment will sense by plant regulator, and then the transcription factor will upregulate the downstream gene for nitrogen fixation. So until there is this non-oxygenic environment inside the nodule, there will be no upregulation of this nitrogen fixation gene. So in this nodules, this switching is not happening. And like I want to Vikas to have some unique data, that's why I didn't show those data. So some of them, they're expressing only this cap, they're only expressing in vascular bundle. So we think that cap also doing some developmental role. Okay. Otherwise, and some are like transporting, but we are at this moment only focusing on this transport.
Okay. So I was thinking it's not something to do with the hyperimmunity or something because of this PR protein. Okay,
no,
thanks.
Yeah.
So Sanjoti, I was going to ask the similar question. So as you are saying, CAP is phylogenetically related to PR proteins. So we know that PR protein will eventually lead to SA signaling. So CAP also lead to SA kind of signaling, or?
Actually, like Gopalji, when in wild type nodule, all the caps are expressing, we are getting most beautiful pink nodule with highest nitrogen fixation. So clearly, this is not doing pathogenic function.
No, so, pathogenic means I'm not saying maybe SA signaling somehow helping this symbiosis. Or
so that's why when we take this, so we also ask this question. So usually under the SA signaling, this PR1 protein get induced. But in our case, if we take, so we don't know much about peanut nodule, but we know quite a bit about Medicago nodule, and we know that AC signaling is not working in Medicago nodule. So we take peanut promoter gas and we express it in Medicago. We also did protein localization, so it also goes to Medicago symbiosome. So indicating that under the symbiotic condition, it is working. Then when we analyze the promoter, we are getting some symbiosis signature. So because it's testing now that he can show the transactivation that is actually getting induced under a symbiosis specific transcription factor, and there is no connection with respect to this SA signaling.
So that means if you add SA, this CAP genes will not be expressed.
Yeah, so actually he also did that. He took the pathogen specific cap, he cloned those caps, and then he add bradyrhizobia. So the opposite way. So first answer is if you add SA, it will not get activated. And from peanut, he also took that pathogen specific PR1 gene, and he took the promoter and he gave bradyrhizobia to show whether the pathogenic one is getting induced, because this is also bacteria. That answer is also no.
Okay, thank you.
Yeah, thank you, Gopalji.
So any question?
Yeah, can I ask a question to Devangshi?
Please.
Yeah, so like in your bacterial mutant, you were showing the bacteria are smaller in size. So I was just curious whether there is anything, because we do see shape change with endoreduplication. So are there any relation?
Relation between cell size and virulence, you mean?
Like my question will be like when they're like smaller and when they are doing proper codam sensing and they're like larger in size. So between these two phases, are there any ploidy difference in the bacteria?
Oh, no.
Okay. So it's more like exopolysaccharide and the outer layers.
Yes.
And I will
I think there are two kinds of EPS. One is loosely bonded and one is capsular EPS. So we think that in PHCA, all the loosely bonded EPS is lost. And the size difference, we don't know the exact mechanism why that is happening. But yeah, we think that it's actively dividing when it's at low cell density. So it's not elongated cell at that point.
Okay. And I will have one more question. So if I'm like recalling properly, you mentioned that there is high nitrite, right?
Yes.
So usually nitrate is a toxic, toxic molecule we consider like, so still your bacteria are growing more. So can you
Yeah, I think PHCA just wants to survive and it wants to eat whatever is available. We were also surprised when we saw that why is it doing this? But we have to understand that nitrite is finally feeding into formation of amino acid too. So maybe the levels that we are seeing are still not toxic, and most of it is diverted to the formation of glutamate.
Yeah, it's interesting to know that. Thank you.
Yeah.
So we have a question for Debyanshi. So it's just a curiosity. So this bacterium causing disease, Ralstonia causing disease mainly in the shoot region. It is entering to the root, but the disease we mostly observe in the shoot, like wilting and all. So it is this is our interest also to understand why it actually entering through the root. There are many bacteria which are infecting, like Xanthomonas. It infects in the leaf part and it goes by different. So is it like when it is nodulating in the root, it is entering through the root. What was the last stop? But then why it is entering to the root? Why it is not?
I think,
yeah, please.
so generally from what we know is, you also probably know, rhizosphere is rich in nutrients as compared to phyllosphere or whatever is found inside for the endophytes. So it has chemotaxis ability. It can sense diverse nutrients and hence can swim towards those nutrients and can enter. So I would say it's an opportunist, because it can only enter through wounds or through the emergence of the root hair, where it's the lateral roots where they're emerging. Those are the points where it can access or can gain entry into the host. So I think the root exudates and those opportunities to enter make favorable for the pathogen to enter through the roots.
Okay, this just this is actually the questions are coming because of your such an elegant
Debyanshi,
sure.
Debyanshi, to continue this discussion further, as you said, during saprophytes, they are basically less abundant and less cell number, and during pathogenic stage, they grow more. So the PSC mutant, the one of the main attractive force for the bacterium to attach to the plant is that root exudates. So what happens to this mutant with respect to its chemotaxis towards root exudates? Whether it is more chemoattractive to root exudates?
Yeah, we have not functionally characterized chemotaxis behavior in lab, but we did an attachment study on tomato seedlings, and we found that, yeah, that's what I showed, and we were surprised it was 9 to 10 times fold higher, like highly attaching as compared to wild.
There are two different phenotypes. One is attraction, and then that is attachment. So attachment you have shown, maybe how it is attracted. First thing is it has to be attracted towards plant. So that maybe you can explore. Yeah.
Yeah, I think because PHCA is good at consuming so many different kinds of food, so it would, their chemotaxis ability, I'm hypothesizing, we have not tested, but would be much better as compared to wild type.
That will be very good. Another thing to add on to Suven's question, maybe in the basically many bacteria may try to enter through stomatal opening and things like that. Okay, Ralstonia does not do that. So maybe there is something called stomatal immunity and things like that. So maybe those immunity might be playing a role against Ralstonia.
Yeah, I don't know if we just put some drops of bacterial suspension on leaves and you will see disease. But yeah, that's a good hypothesis that even they have a chance to enter through stomata, and if it can still not enter, then this hypothesis would be very interesting. Yeah.
Actually, on chickpea, we did vacuum inoculation, right? I think Suven knows this. So, but then even after vacuum inoculation, it was not successful. But there is something beyond even stomata, something that exists that probably, and I think there is no particular host that actually allows this bacteria to enter aerially. And it's probably exclusively bound. I think it has over 400 host range, but I think none of the hosts actually allows it to enter through the aerial parts.
Yeah.
And in your studies, the inoculation was stem injection, right?
Yes, because PHCA, if you try to pour it in soil, it will just not enter the stem.
Or is it less survival in the soil, or it's just that it cannot enter through root?
So I showed you two, so less spreading and increased attachment. So if it cannot spread, even if we want to collect plant samples for our transcriptomics, it would have been very challenging. So for our convenience, we put it in the stem so that we can get some samples to understand the transcriptome.
Okay, so that means that that's why the chemotaxis part could not be answered much of. It's only a hypothesis. Okay.
Yeah.
So Divanshi, so this regarding the different species of Ralstonia. So all they enter through roots, or there are some species like Saijiji, this Indonesian species, they also enter through roots or through some other vector like insect.
Yeah, so in phylotype 4 strain, it has been shown that insects help in their transmission too.
So I think they have very highly reduced genome also. So there was a comment by Suvendra, like why Xanthomonas do not enter through root. So if I am right, like Ralstonia have very bigger genomes, and I think they have that kind of capability to enter through root. Maybe genome size difference may explain. Suvendra?
I'm listening. I'm listening and learning.
If I am right, like they have these all beta Proteobacter, these have very large genome, so I think they can enter from any like root.
Very nice point. Yeah, very nice point. Yeah.
That's why if you see that Ralstonia Saijiji, if I'm telling correctly, so that has reduced genome and it is using insect as a vector, and I don't think it has any root lifestyle. I don't know much about Ralstonia, but
Gopal's work that the rice root cells, when he infected with Xanthomonas, it elicited a lot of reaction, and actually then it went to wound response, his work, but he started with actually rice root cells. Am I correct, Gopal?
Yes.
So that is very, actually now it is very nice opening actually. Alok.
Yeah, maybe we can first discuss with the presentation. So shall I take one question for Alok? Alok, your mutant was very interesting. Okay. But its application, Amit is also asking how to take the leads for field application. So I'm just concerned that mutant, just single gene mutant, if you start using as a biocontrol, may not be a good idea, because eventually we all know that revertant will come and will take over rest of the thing. So what I was thinking, maybe you can take help from Prabhu and see whether naturally occurring non-pathogenic that Xanthomonas have any Xanthomonas where these genes are mutated. It's not there. So maybe those strains can be good because your XO strain is pathogenic. It has everything. So type 3, type 2, everything. I'm sure it will revert and can start causing infection someday.
Yes.
But you take help from Prabhu and try to see whether there are xanthomonas where non-pathogenic xanthomonas where this is not there. Maybe that will be a
this
good...
particular regulator is not there.
Yeah,
right?
that's what I'm saying. That's what I'm saying.
Okay, fine.
That will be very good lead for biocontrol.
Yeah, thank you.
So that so that is one thing. Another thing I was telling that maybe this that I was telling you during the your discussion that maybe it is regulating some common regulators of T3SS, and those common regulators may have pleiotropic role in terms of T3SS functions as well as bacterial physiology like EPS, motility, and things like that. So it can be a very good research material for you to work on forever. So you can go into the depth of it. Yeah. So any questions for Alok?
Yeah, Alok, I think the plant determinants of HR, it will be very interesting because the HR pathway is very powerful, but very less is known about it. Although a lot of candidates are known, because you have already developed a nice system. And that too, a system with Benthiana and tomato will definitely work faster. So I think it should be very easy to do assays to identify the plant factors that contribute to the HR. There must be some common signals that I think you will be able to start with to knocking out the regular HR pathway things. And then if something happens to be a novel regulator, then it can come from some kind of a library screen or something else, which is very easy to do in Benthiana.
Right. I would like to discuss one point with you. Like, what would be the ultimate fate of type 3 secretion system like mutant? Because the cell number were not decreasing in the Nicotiana. So what would be happen actually ultimately with those cells?
I think maybe Gopal can answer this, but before that, like these are a lot of artificial. I mean, it's an artificial inoculated bacteria, right? I'm not sure whether you will dip inoculation. If you do it, probably the trend would be different.
Okay.
So because Alok, you are infiltrating into it, na? So you are anyway giving lots of things, and HR is not working. So there is no plant defense. So bacteria is ideally sitting for some days. So if you try to look it over a time, maybe you will not see the bacteria. Plant would have taken care of that.
Yes.
Right.
Both
so
over
maybe
a
it's
time.
an artifact because you have just infiltrated, and there is no HR, so which can kill those bacteria.
Right.
Okay, so maybe if you try to do it after a week or after 10 days, maybe it's something different. And what Senthil was telling you, naturally try to infect HR defective mutants epiphytically and see whether they cumulate to such number. They may not do such accumulation.
Okay. Fine.
Yeah. But another interesting thing is that even though you are not seeing visual HR in terms of your mutant, but your cell number is decreasing. So some sort of plant defense is happening there.
Yes, some sort of is happening.
Yeah, so it is a good model to tease out the component of HR. So some basal plant defense is there, and then a cumulative HR. So you can think of, and as Senthil said, due to advancement of Higgs, you can quickly do lots of things there.
Yes. Actually, so far the type 2 non-host resistance, which typically involves HR, only SGT1 has been proven to be SGT1 and NHO1. These are the two major candidates that have been known to elucidate as an exclusive for NHR dependent type 2 non-host resistance. So you have a lot of scope that any new candidate that you will identify will be a novel factor, which is a plant determinant of hypersensitive response, which will have a wide spectrum application beyond the pathway dissection. So maybe it is easy.
And actually, I mean, NHO1 and other one, what they detect actually, what they sense in the bacterial part, what they sense.
So NHO1, since it's a glycerol kinase, I think it senses one of the effectors. It's kind of a argine. It's not typical argine, but it's kind of a pathway that relates to this. But SGT1, it happens even with the host regular argine interaction also. If you put AVRR interaction, it happens with SGT1 also that compromises. So SGT1 is a different story, but NHO1, yes, it has a specific determinant from the bacterial side, Pseudomonas. So, but I think you will have something new to find if you have a screen or something like that. If you have an exclusive screen setup or something, that if you can do it.
Okay.
Until Ramesh here.
Yes, sir.
Yeah, remember Sohini Dev did some experiments and also Mahesh also. It looks like it is, and it I think started with, maybe it was Mahesh who started this experiment. It is the ZOPQ is a protein from XOO that's recognized by benzammiana to mount a defense response.
Yeah.
So there is an effector triggered immunity, and if I remember correctly, if you knock out ZOPQ, then it actually multiplies a little bit more than the wild type. So you have to actually worry about effector triggered immunity as one kind of non-host resistance, and the kind that Senthil is actually talking about that may not involve a classical resistance gene. Just take a look at it. Even the R gene that recognizes ZOPQ has been cloned.
Okay.
So there is one another protein, just like what Sir was mentioning, is there is EIX, ethylene-induced xylanase. There is one Israel group who has published a lot of papers in this area. So they have screened like that, and they could identify a lot of HR intermediary candidates which are specific to host, non-host. They could able to distinguish it. I mean, there are papers from that group that can also reveal more candidates onto this. I like more of a type 2 non-host resistance because it's more powerful, and if that can be exploited without much of harm to the plant by having a restricted, I mean, it should not be a runaway cell death. As long as we manage that, it should be a very potential, novel resource that we can bring into a host from a typical non-host.
So Alok.
Yeah.
So you are mentioning that this regulator WEM-RXO.
Yes, sir.
Orphan means you don't know receptor and the signal.
Yeah, I don't know actually. Yeah.
So is it possible it is using receptor from some other two component system?
Yeah, possibility is there. I mean, the cognate, there is no cognate sensor kinase. So we need to find what is the actual cognate sensor kinase.
Just like at least in case of OP4R, they find it get phosphorylated by some other two component.
These are possibilities are there actually. Even like one response regulator can be governed by multiple sensor kinases. Yeah.
So that part you can like explore. There is
yes.
And that is becoming, I think, little interesting in this field.
Yeah. So there are two things that can be done. Like first to find what is the cognate sensor kinase, and then second thing is what is the exact host signal.
But now, Alok, there are 500 genome. You see, you may find in one of the genome its partner, or if it is deleted in this strain. I hope it's not like it's in all strain. It's orphan only. But if you can see.
Shall I ask one question for Ashish? So Ashish, so as you said, you have also figured out some of the pathogen genes which may be helpful for pathogenesis. So did you standardize the mutagenesis, or still?
So so one we have done is host-induced gene silencing. So ultimately, having that gene in the plant itself. And one of the important virulence factor for fungus, especially Sclerotinia, is oxalic acid. So we have shown that if you do a host-induced silencing of a gene against oxalic acid synthesis gene, then it's able to control.
But that will be very interesting, because I think oxalic acid biosynthetic gene is not known, the pathway. So could you?
Oxaloacetate anhydrase, one gene is there. So we have also done this double standard RNA spray-induced silencing too, like where we have fed the fungus with this double standard RNA in a culture, and we saw that it's able to go in the fungus and it stops its growth. So, and we have also sprayed on the plant and then challenged with the pathogen, like in vitro, like you detached leaf assay. So it does look like those kind of things which can be applied in the field.
Yes. So any question for?
So Devanshi, for the species complex, let's say your mutant versus two other species, because when there is RSSC complex exists, that complex behaves much differently compared to, let's say, Ralstonia solanacearum alone. So how does this mutant in relation to another species complex will perform? Like if you co-inoculate, will there be same interaction that you would find with the plant?
Yeah, so I have discussed similar things with Tiffany Love Power. She's a faculty at UC Davis, and her lab is planning to mutate PHC and other phylotype strains and trying to see if what we saw in Asian strain, are we seeing similar things in other phylotype strains. We did do some competition assays where we tagged PHCA mutant with different antibiotic as compared to GMI 1000, the wild type strain. And when we did the competition, we found that the wild type was overpowering the mutant strain. So less number of PHCA cells and higher population of wild type. That's what we found just for phylotype 1 strain.
So Sejuti, I can ask one question to you again. So my question is that what are the structural difference of this cap protein if you compare the cap protein with the Medicago NCR protein?
Actually, Sarup, so there are two things. One is this NCR and defensin. So in Medicago, there are NCRs with 6 cysteine, and this defensin, there's short peptide with 8 cysteine. So that is dealing the endoreduplication. And the second, the PR1, they're not peptide, they're big protein. They are around 150 amino acid. The only commonality is cysteine, presence of cysteine, and but they do have this alpha beta fold structure, which is similar to like any other PR1 protein. So when I'm saying structure, it's like based on this we spot like more folding on available other template. So you're also trying the alpha 4 to see what are the structure, they're like different these caps and their structure. So there are two components. The first thing is this NCR versus defensive, and then cap is unique.
Okay, thank you.
Going back to the question that Suvendra was having about why XO doesn't infect through the soil and only infects through the leaf, maybe that's partly because the ability to survive in the soil is a very specialized trait. So a bacterium that can survive on a leaf surface like XOO may not do very well in the soil. So if I remember correctly, there were papers that said that it doesn't survive very well in the soil. So maybe if someone compares how XO, its population, how it drops off, or what's its survival rate in soil versus something like Ralstonia, then we would know. I would suspect that Ralstonia does much better in a soil than the plant does. So it survives there, multiplies there, and then it can find its way through the root. XO probably can't survive very well in the soil.
Yeah, just connecting with this. So Ralstonia, like recently people started seeing that how nodulation and pathogenesis, they are connected. So if you inoculate Ralstonia to nodule, it will do some infection, but it will restrict the infection inside the nodule. So like there also this immunity and like specialize this microbe and plant to discriminate between the pathogen and symbiont, it's taking place.
Yeah.
Yes. Gopal, if we are done with the panel, individual presenter's discussion, maybe this you can explain, or if you want me to explain, I can.
Senthil, you can explain. Yeah.
Sir is also there. So actually, I just spoke to Gopal and it just clicked my mind. This picture has been promoted by MPMI APS as such in a large number of forums. I've seen it in many forums. So they have started developing a group. I think it started sometime in 2009 or so when I attended that meeting. And in Glasgow meeting 2019, I think it happened. They have concluded and listed top 10 unanswered questions that the field should focus on. Our field should focus on. So I think most of the questions are known. I think this group is up because at least 4 questions are already we discussed from the presenters of this group. One, I think question 1 is whether the interaction between beneficial and pathogenic is simultaneous. You can read an article. There are several articles in this series that the MPMA has published if you go to their website. Second is what is the influence of different abiotic stresses on the plant microbe interaction. Especially they focus on how the disease triangle, the environmental part earlier, which is considered not as a stress, but just an environmental influence, namely RH and other stress. They've replaced that environmental part with an actual stress component, not replaced, slashed with the abiotic stress, which means another equally disturbing entity, which is an abiotic factor to the plant. If it comes, how this triangle will change. And I think the third point is translation to the field. I think this is something we know. We've been discussing. The fourth point was how about the microbe to microbe interactions, be it in the outside the plant, like philosphere or rhizosphere, or inside the plant. So either it is a phytobiome or a microbiome that sits in it, microbe to microbe interaction that is in large numbers it happens. I think they also opened up a journal that APS has opened up phytobiome and one other Frontiers journal which focus on this one. And fifth question they are asking is typically this MTA and DTI, whether it is distinct pathways or it's the same pathways. A lot of discussion goes on. I think this is one field they project that one must work on to explore more, to get more. And the sixth area that they are suggesting is the non-host resistance. And seventh is the cell death that is related. That's why I was excited with the Alok's presentation also, that the cell death component is unexplored and unexploited. And it's there is a lot of scope to understand with systematic assays. And eighth question that they are asking is about why so many or so few effectors depending on the pathogen what we are talking about. Why that particular pathogen either it's able to handle with so few effectors a large number of hosts, whereas the other pathogen which has what distinction that makes between these two. And the ninth point that they are saying is the evolution of virulence, and then the 10th point is about the ecological context. So I think I'll start with this. If there are any discussions that we can have to indulge this kind of areas into our research, or to look at our research areas in this angle, that can be done.
Senthil, if I can just add in the first part, the beneficial microbes, I feel there are 2 kinds of beneficial microbes. There are the specialists like Rhizobium, which can actually enter and grow to very large numbers. Okay. And I feel that there are other beneficial ones that are the typical endophytes. You never find them in such a high concentration within the plant. And so the plant immunity has to be, if you want to multiply to large numbers, then you have to be able to modulate the plant immunity. If you want to stay, if you are trying to stay below the radar and are providing some kind of beneficial functions to the plant, I think the plant will be able to tolerate it. So I feel that we have to separate out the beneficial microbes into these 2 classes.
Okay.
And how Rhizobium actually manipulates the host defense response versus how an endophyte that doesn't really grow to very large numbers, I think they are very different things. So
I agree. Yeah.
It's good if you keep those 2 separately, and both are very important, because Rhizobium is obviously a very successful successful lifestyle, but these other endophytes, they seem to have a lifestyle where they don't multiply to very large amounts. And because they provide some kind of services to the plant is tolerating them. So that's how, that's one way to look at the endophyte and the beneficial versus the pathogen. And
yeah.
yeah.
Sejati has this CAP class of proteins. Some of them are, I mean, one of them she described is, of course, related to her work, but many others are typical PR proteins which are immunity related. I think she is also addressing this area of converging this pathogen and its role of this class of proteins in that. Yeah.
Actually, my apologies. I couldn't attend many of the lectures because I was stuck in ICER, you know. We had a meeting where we are moving into a new campus and allotment of space to faculty and things like that. So I just couldn't avoid it. But I just got here as soon as possible and joined.
It must be an exciting thing happening, sir. Yeah.
Well, it's exciting, but this is something very exciting. So, you know, I would have, today I think I would have rather been here. In other days, I would have liked to have been there. But you know, you can't have everything in life. So
Yes.
at least I could join in and hear some of your discussions.
Will be
But I
exhausting.
think it's important, you know, we need to think in terms of, I think as a field, you know, we need to start thinking about the questions. You know, what are the larger questions? Which is the direction in which the field is going to be moving? You know, and then start looking at, positioning ourselves to start those. So many of these, the problem is there, but the question that how do we solve that problem? You know, those are not very clear. How do you decide that an endophyte or a beneficial bacterium that grows to very, very low numbers within a plant, you know, that it's staying below the radar of the plant. So those are, what I remember is there are some papers where if you reduce the innate immunity of the plant, you know, for example, maybe FLS2 mutants, these endophytic bacteria, you know, that were earlier growing to lower levels can now increase in their numbers. So there is some trade-off that is going on, you know, and also the number of cell wall degrading enzymes that they make. An endophytic fungus that is not going to grow to large amounts may not make very many large numbers of cell wall degrading enzymes, but closely related pathogenic variants of, you know, of the same genus but a different species that causes a disease. If I remember correctly, there are a lot more cell wall degrading enzymes that they actually produce.
Yeah.
So we need to actually pick the question. I mean, the problem is important. How do we actually identify, you know, how to tackle the problem? So what kind of questions we can ask? How do we develop a suitable model system in which we can actually address these? So these are, if we can actually come up with, that's where the challenge is, you know. And if we can come up these, brainstorm on those aspects, and then you know, we can actually start putting up experiments that we can actually do, you know, write proposals, get grants. And basically I feel part of the thing about Indian science is that you know, we actually don't get into the problem. We get to the problem after it has been defined by somebody in the West, and the initial methodologies have also been developed. So we end up doing, you know, we don't make the initial breakthroughs, and that's because of the fact that we are a little bit you know, laggard in our thinking.
Yeah.
I'm happy that Senthil actually put it up, because that gives us an opportunity to start thinking about some of these big questions, important unsolved questions, and how do we address them. For example, you are saying, how does virulence evolve? You know, how do you actually tackle something like that? See, these are things that we have to actually think about. I feel that there are experimental possibilities. You know, there are ways that we can design experiments to solve these. We just need to brainstorm, come up with those things, and then put up the grant proposals and get the money before, you know, I mean, sorry to Devanshi and Ashish, I'm considering you as Indians, you know, as part of
yeah,
our group.
we are.
Definitely.
There is no.
Okay, so I feel that as a country, you know, this is what we have to do. It's not just in our field, you know, it's just in every field you look at. But I think our field has an opportunity to be able to solve this, you know, to go one step ahead. We have a good group of people. and looking at diverse systems, because in some of these questions can be studied in one system, but maybe not in another, not in another system. So
sir, maybe actually we can take this group meeting and let's like a proceeding. We can try to have the Indian overview of this MPMI top 10 questions. How India is prepared and contributing to answer.
No, but how much of it we are actually
at least few, sir, 4 or 5 for sure we can.
No, we can solve many of them. We can address many of them. But we need to have. See, my opinion is 50% of a job done for a project. If a project is well thought out in the beginning, if you lay out the logic and if you lay out your experimental plans very clearly in the beginning, then 50% of the job is done. If that part is wrong, no matter how hard people down the line, the students or the postdocs work, nothing will actually come out. So if we define our problem, our question, and the experimental approach, then I think, so I feel that how we do it, I don't know, whether we need to have brainstorming sessions on each of these topics and then, come up with a set of ideas that, okay, this problem we can solve this way. And maybe some of us will work on that. Some others will work on another. But these are, very, very important, but very difficult to address. How does one, see, one of these things that Prabhu and I started discussing about these non-pathogenic xanthomonads, we thought that would give us an idea about what actually constitutes virulence. But it now turns out that these non-pathogenic xanthomonads are a distinct lifestyle actually. So, and they may have actually evolved as a parallel line to the pathogenic ones. So the answer may not come from those. The answer to, how virulence evolves, may not actually come from there. So we need to think through each of these requires, brainstorming and thinking about things. And I know groups in the US do that. A group like these, they will sit together, think, ahead, what is the problem? What is the major problem? How do we solve it? And then they start actually putting resources into that and go ahead.
So one similar thought, like what you said, Ralstonia solanacearum, about 60 years ago, if you look at some papers, they used it as biocontrol. I was surprised to see that. And then later on, I don't know whether what happened to the pathogen or whether that experiment that was done 60 years ago was on a particular plant species that was working, or is it because of the pathogen's virulence that got spread it over as a pathogen, it evolved and it spread over 400 species. So it's again, I'm not sure, depending if those results are correct and if there was indeed a good biocontrol and later became a pathogen.
No, it could have been a strain of Ralstonia, like, this Xanthomonas that is non-pathogenic that Prabhu has got, so they could be even in the Ralstonia, maybe there is something like that, So I don't think that, pathogenesis can evolve over 60 years, It's something that takes many, many years and, a lot of innovations from the, if we can call it that, a lot of innovations are needed to actually get there. And, how it started, when it started, all of these.
Sir, actually genome sequencing and characterizing lots of genomes are very helpful. So for example, for Rhizoctonia solani, we have been sequencing the genome. And what we have figured out that genome has duplicated. So basically during domestication of plants, when plants started domesticating, the pathogen also started domesticated to the plants. So host specificity was there. So around for Rhizoctonia solani, we have recently figured out that there was a major evolution around 44 million years ago when it started differentiating the below part and the aerial part actually. So some Rhizoctonia is root pathogen or some are foliar pathogen actually, sir. So we figured out that around 44 million years ago, the strain has diversified. And when we see there are transposons which has made the duplication of the genes, sir. So some genes has become secreted, some paralogs has become as a effector, other paralogs are now not being induced during pathogenesis and things like that. So we have characterized around 50 rhizoctonia genomes, and this is the recent insights which we are getting. That genome duplication, evolution of the secretion signal, effector paralogs are getting positively selected, and things like that are helping towards evolution of the strains.
Sure. No. So one way of actually, practically trying to make a difference from our side, as a group, if we think, okay, these are the 10 questions. Senthil has actually put them up that have come up after a lot of thought. Now, are there questions that now, yes, we can actually pick each of these if we brainstorm and say, okay, is this the way to do it? Is this the way to do it? Just keep, one by one. Maybe, we need to spend time on some of these things. Maybe not now, but another time we can actually have where we devote to brainstorming about each of these problems, So one of us actually just lays the problem that this is like these 10 questions that Senthil has said. Maybe in each session, we can have 2 such questions that we look at. And then we just brainstorm, put our ideas together, and then write up small things. And there will be enough for so many people actually to work on. And lots of young people, all of you are young, and you have your young students. So we need to define the problems, make the problems actually even more interesting, so that we can compete with, the people who work on, pathogenic bacteria that affect humans. So our thinking must be smart. That's what I feel. So we have these 10 questions, and maybe we can devote, over the next 4-5 months, okay, we decide, all right, this day we'll discuss about a couple of things. Those who can join in the group can actually join and do an online discussion like this, and come up with some sort of an action plan. I mean, just a thought. You guys can discuss. As I said,
yes, sir, it is completely correct, sir. Also, actually, the MPMA has already started doing it. I think they are currently in 6th question or something like that. So every question they have taken it up. There is a lead talk that is given by a very eminent expert in that area, and then there is a discussion followed by, they are formulating special issues also for each of this. So they are systematically addressing that, and then they are giving every month one talk to a young first author who publishes that. Like that, they are proceeding. Like, okay. But yeah, from Indian context, we should do that.
So what we do it, and we decide. I mean, those of you who may have actually read some of those, we can take those. We don't have to reinvent the wheel and summarize it. Maybe we have a recording of that. We can actually hear it.
Yes, sir, they have a recording of all of them.
Yeah. So we can do that. Then we can actually think through and see what is it that that we can do. So MPMI is doing it. I mean, MPMI is great because they actually are the ones who. It's ISMPMI that actually led to all of these breakthroughs that we are actually been a part of, and they have actually benefited from those. Yes,
and these talks, these talks are open for everyone. So this talk is international, and they are like, I have attended few of them. Okay. So
it's the recorded version is available, sir.
Also available. So
yeah. So those maybe we start with that, and if somebody so that's already played, then we can start discussing, brainstorm. Maybe every one of those problems we may not be able to tackle, but if we can start a few that where we think that we have an advantage, then we can actually move forward on that. So, difficult problems, but some of them are experimentally tractable. How did virulence evolve? I mean, that's something that is very, very difficult to get at, but for everything there is a way you can actually tackle.
Sir, as you mentioned, sir, I think some Indian specific from Indian context, if you can,
that's the advantage we can have. We can try and see if we have an advantage that we can leverage, either the pathogen or the plant, or maybe some resource that we have. If we actually put up a good proposal, money is not really that big a problem. You can get reasonable amounts of money, but we must have really cutting edge ideas where we can actually get huge, fairly large amounts of money. So it's up to us to come up with those sorts of things. And the problem is that for each one of us, we are busy with mundane things that we have to take care of. A grant proposal we have to write, a grant proposal we have to review or sit on some other committee, go and teach a class, various things, go give a talk somewhere. But we never give enough time to the brainstorming part, where we have to try and frame the big question, sort of contemplate the problem. These are things if I remember, Suvendra and Prabhu in the lab are actually really good at it. And I've seen Senthil do it in NIPGR. He would come and talk to me about some of these things. I must say that I myself haven't had much time to think of many of these things over the last few years. But as more and more I see, and I see how science actually works and how people, bring teams together and get the resources that are needed to do, to address problems, because one is defining the problem and the other is getting the resources. So I feel in India, our main problem is not the second one. Many of us think it's the second one. That's not the problem. The problem is being able to come up with a world-class, some sort of a world-class program that I'm not saying that we will get it, but let's at least give it a try and do it. So maybe we can think of, this meeting itself, I think, has been quite exhausting for people. So maybe we can give it a break and then pick, one or two, and then start have one Saturday evening discussion, or some time we can find where that's convenient to people in the US, people here, and then start discussing these things.
Yeah. And
anyway, that we can discuss
tomorrow also, sir. Actually, during
we can discuss that tomorrow.
Yes, sir.
Usually we do that on the last day. Actually.
Yes, sir. Yes, sir.
The last day of MIPAM we do that.
I think now we can close.
Yeah, we can close for today.
Yes, sir.
Yes. Just one thing I wanted to add. As an international collaborator, just keep me in your, if you think that there can be anything, I can be a collaborator, I'm open to that. And science does not need boundaries. Science is open.
No, we know that. We know that. Don't worry about it. We'll discuss and see what is the best way to, first is finding the problem, then the question, and then where we have an advantage as compared to what somebody else is doing. Okay.
Yes. So then I take this privilege to thank all the speakers and and great discussion at the end. So tomorrow, I think morning, what time we are meeting? 9 o'clock, Sukhdeep bhai?
9:30.
9:30?
Yeah.
So we'll see you then 9:30. Thank you. Thank you, sir. Thank you.
Bye,
thank you.
Bye, everyone. Bye.
Thank you, everyone.
Yeah. Bye.
Thanks.
Bye.
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 Gopal.
और ये प्रभु आई कॉल करती है। हेलो